If you're studying for the AP Bio exam and you're feeling nervous or anxious, that makes so much sense. The AP Bio exam is tough. But don't worry, in this video we're going to go through the entire course, every unit, every topic. You'll learn everything that you need to know so that you can crush it on the AP Bio exam. My name is Glenn Wokenfeld, also known as Mr. W, and I love teaching Biology. To help you study, I put together a checklist. It includes everything that you need to get ready to study for this test. You can download it at apbiosuccess.com/checklist. Unit 1 focuses on the chemistry and properties of water and hydrogen bonding, the elements of life, and the 4 biomolecule families that all living things are made of: carbohydrates, lipids, proteins, and the nucleic acids, DNA and RNA. First thing to note is that water is a polar molecule. There's unequal electron sharing between oxygen and hydrogen, and so there's a partial negative region over here, there's partial positive regions over here. Note that hydrogen bonds are intermolecular bonds. They're between molecules, not within molecules like covalent bonds or ionic bonds or anything like that. What's happening is that the oxygen is partially negative, the hydrogen is partially positive, and the hydrogen bond is a weak bond that forms between those two areas. Hydrogen bonds are much weaker, much weaker than covalent bonds, ionic bonds, any of those intramolecular bonds. Using the diagram below as an example, describe how hydrogen bonds can form between molecules besides water. The key idea is that hydrogen bonds are everywhere. They're not just within water. They're essential in biology in general. So in this example, there are 2 hydrogen bonds, and here's one oxygen to hydrogen over here. Here's another one between nitrogen and hydrogen. This is between the nitrogenous base adenine and thymine. And these are the hydrogen bonds that hold together DNA. Hydrogen bonds, again, are essential. They're key to the structure of DNA, RNA, proteins. You'll meet them again and again throughout your biology course, and they're important to know for the AP Bio exam as well. So now let's look at some of the consequences. So describe cohesion, adhesion, and surface tension, and explain how these key properties of water result from hydrogen bonding. Cohesion, that's hydrogen bonds between water molecules. So this is cohesion right here. And it's responsible for many of water's very peculiar properties. It's a very small molecule, but it has a very high heat of vaporization. It takes a lot of energy to get water to evaporate, a high specific heat. It can hold a lot of heat, and it has high surface tension, which we'll talk about below. Adhesion is water sticking to other stuff. So like, for example, here you see hydrogen bonds between water molecule and the cellulose walls that make up the conductive tubes of plants, which are also called xylem. A phenomenal thing is called transpiration, which is how water gets pulled up to the top of trees, and that's all based on water's ability to cohere to other water molecules and to adhere to the sides of the conductive tubes of plants as water evaporates from the top. And finally, surface tension. Here we see a paper clip that's resting on a net of water molecules, and of course that's ridiculously out of scale. You can see how that's keeping this paper clip from sinking down, and here it's the force exerted by water molecules on the surface of a body of water creates a kind of web or net upon the surface. In terms of hydrogen ions, hydroxide ions, and pH, describe the difference between an acidic and a basic solution. First thing to note is that acidic solutions are solutions that have more hydrogen ions, here's a hydrogen ion over here, than hydroxide ion. That's represented by OH over here. So if you dissolve this in water, you wind up having lots of hydrogen ions, and that pushes the pH down, all right? The pH is below 7. In bases, there are more hydroxide ions than hydrogen ions. So if you dissolve sodium hydroxide in water, you wind up with a lot more hydroxide ions, and that pushes the pH in this direction. pH is above 7. And one thing that I want to emphasize is that you don't really need to know about pH for the AP exam directly. There won't be a question about it, but it might be part of an FRQ or a multiple choice question. So it's an essential underlying concept. Elements of life, topic 1.2 of AP Biology. So what do you need to know about the elements of life? Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, sometimes referred to as CHNOPS. Carbon is the central element in all of the molecules that make up living things. Hydrogen, often used in energy exchange. We have molecules like NAD and NADH, which show up in cellular respiration. NAD plus is the low energy form, NADH is the high energy form. You see stuff like that over and over again. And also, hydrogen as an ion, as a hydrogen ion, a proton, is often pumped around to create energy gradients. It's very important in the synthesis of ATP. It's the basis of acidity and alkalinity, which we just talked about. But the main thing is to know about these atoms in context. Phosphorus is in phosphate groups, which is found in ATP. That's the kind of interconnected cross-topic knowledge that you want to have for success in AP Bio. Now on to topic 1.3, monomers and polymers and functional groups. So what are monomers? What are polymers? The basic idea is that 3 of the 4 groups of biomolecules, carbohydrates, proteins, nucleic acids, are built from smaller building blocks that are called monomers. So here's a glucose monomer over here. Living things build macromolecules, the big molecules, proteins, nucleic acids, polysaccharides, that have specific 3-dimensional shapes, and shape determines function by combining these monomers into polymers. And a great analogy, if you hadn't already thought of it, is that the monomers are like Legos. You can combine them in any way to create other structures. And these big structures, the Millennium Falcon that you might have built as a kid or something like that, those are the polymers. And here's a note about structural formulas. If you see something like this, you might wonder, like, what's going on at these angles? Every unspecified angle vertex has a carbon atom. And that's why this is a C6H12O6, because there's carbons here, 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 here. Carbon is so central that it's just understood. How do you put monomers together to make polymers? It's a process that's called dehydration synthesis. Everything in biology is run by enzymes, as we'll see in Unit 3. And what enzymes do is they pull a hydroxyl group over here off one monomer, and it pulls a hydrogen off the other one. So here's the hydroxyl, here's the hydrogen. That water is pulled out, right? Because this is H2O, the water gets removed, and that creates a bond that's right over here. So here we see that between 2 amino acids, and same thing. So 2 monomers becoming a larger molecule. Dehydration, synthesis, easy to remember. Synthesis is how you build things, and dehydration, when you're dehydrated, you lack water. So dehydration, synthesis is pulling out a water. Now, what about hydrolysis? Hydrolysis is the opposite of dehydration synthesis. In biology, the suffix lys is very important, and it involves breaking. So what happens in hydrolysis is that an enzyme, which isn't shown, inserts a water molecule in between the 2 monomers making up the polymer. And what that does is it breaks them apart. Here we have lactose, which is a disaccharide. I'll review that in a minute. It's a sugar that's made of 2 simple sugars that are bonded together. You add a water molecule and you get galactose and glucose. What do you need to know about functional groups? In one sense, not a lot. They're not going to directly appear on the AP Bio exam. But in another sense, they're very important in terms of you decoding what's happening with the molecules in biology, which means the molecules that might appear on the AP Bio exam or in your course. So let's go through a couple. Phosphate groups, number 1 over here, they're key for energy exchange. ATP, adenosine triphosphate. Phosphates are also found in DNA, and they're what energize DNA monomers as they're put together. This methyl group over here at number 2, it's used to silence DNA. It makes molecules non-polar or hydrophobic. There are a couple of polar functional groups to know about. There's the hydroxyl group at 5, the carbonyl group at 7, and they make molecules hydrophilic or water soluble. The carboxyl group, that's at number 3, and the amino group, that's at number 4, those are essential in amino acids, which obviously have an amino group over here, have a carboxyl group. The sulfhydryl group over here at number 6 is very important in terms of protein structure. It creates a stabilizing bond that holds proteins in a specific three-dimensional shape. And the acetyl group at 8 is used to activate DNA through a process called acetylation. So it's kind of the opposite in terms of function from the methyl group that we talked about. Carbohydrates and lipids. So what are the 4 types of macromolecules that make up living organisms? What can you identify from this diagram? So what you have here are carbohydrates, represented by this disaccharide over here. We have a lipid, represented by a phospholipid, a key molecule in membranes. We have a protein represented by hemoglobin, and a nucleic acid represented by DNA. So those are the big 4. What do you need to know about carbohydrates? Carbohydrates, the monomer, or monosaccharides, those are simple sugars, and some of those are all important in biology, like for example, glucose, which is essentially the fuel of life. Disaccharides are going to show up less in the course, but you might have a question that's about lactose and lactose intolerance. Well, it really doesn't make sense unless you know that lactose is a disaccharide composed of 2 linked monosaccharides. And then you have polysaccharides, molecules that are used for energy storage, like starch, which is in plants, glycogen, which is in you and other animals, and then polysaccharides that play important structural roles, like cellulose, which makes up the cell walls of plants. While humans and other animals can't digest cellulose for food energy, a few animals such as ruminants and termites can. Explain. Most animals can't hydrolyze the bonds that connect glucose monomers in cellulose. So here's cellulose, it's a polysaccharide, it's a bunch of linked glucoses, but it's linked in a way so that you don't have the enzymes that can break this bond, freeing up the glucose monomers. So essentially, you could eat lettuce, celery, these are high cellulose foods all day. You'd never get enough calories to really power your life processes. You do have the enzymes that can break this bond in starch. So over here, and over here, and over here, and that enables you to convert starch into glucose, and that enables you to use that glucose to power cellular respiration. There are a couple of animals, such as termites and ruminants. Ruminants include cows, sheep, goats, deer, other animals, other mammals. And what they did is they involved symbiotic relationships with microorganisms that can hydrolyze this bond and thereby break this bond over here, break this bond over here, break this bond over here. And that releases these glucose monomers, making food energy available. Let's end our review of carbohydrates by looking at an issue in relatively recent human evolution related to carbohydrates. And that's about the biology of lactose tolerance and intolerance. So here's what you need to know. Lactose is the sugar in milk. Here it is, it's a disaccharide. Lactase is the enzyme that hydrolyzes lactose into monosaccharides. And here you see that reaction happening. Most mammals only produce lactase during infancy while they're suckling, because that's the only time that most mammals ever drink milk. So that makes sense from an evolutionary point of view, because when you're an adult, you don't produce lactase. Why should you produce an enzyme for something that you don't eat? But what happened in human evolution is that some human groups that were pastoralists, herders, these are people who were associated with cows and sheep and goats and so on and so forth. They had access to all these milk products from the cows and the sheep and the goats, and some of them developed a mutation that enabled them to continue to produce the lactase enzyme into adulthood. And that opened up a whole niche of food exploitation that wasn't otherwise available. Now, this didn't happen all over the world. This happened in a couple of hotspots. Here's one in Africa, here's one in Europe, and here's one in Saudi Arabia, current Saudi Arabia, and here's one that is in the Indian subcontinent. And in those areas, lactase persistence, production of lactase into adulthood, became widespread. But in large areas of the world, that never happened. And there are many humans, the majority of humans, who are lactose intolerant as adults. But if that's true of you, as it is of me, then we have products like lactate. Lactate is essentially lactase, the enzyme that you can use as an additive to food, and it'll break lactose down into galactose and glucose, and you can buy lactate milk and so on and so forth, and that's how that problem, if it's even a problem, is solved. Now let's move on to lipids. Let's talk about lipids and what are their functions. So here we have 4 different lipids, and what makes a lipid a lipid? First of all, lipids are molecules that are wholly or partly non-polar. So for example, like you see all these hydrocarbons over here. Those are all completely non-polar. They don't dissolve in water. They're hydrophobic. They also are characterized by the fact that unlike the carbohydrates that we've met and the proteins and the nucleic acids that we will meet, they're not composed of repeating monomers. They might have subunits, but they don't have those subunits repeated hundreds or dozens of times. So what are their functions? Well, over here we have a fat, a triglyceride, and that's used for energy storage. That's true in both animals and plants. In animals, those fats are usually solid. In plants, those fats are usually liquid. We call them oils. Here's a wax. Waxes are used for waterproofing. This molecule is a phospholipid. It makes up cell membranes. We'll talk about that later. And then we have a steroid hormone like estrogen or testosterone that's used for signaling. So what's the relationship between phospholipid structure and membrane structure? Well, here's what you need to know. Phospholipids have a hydrophobic nonpolar tail. That's this over here. And they have a hydrophilic or polar head, and those two parts are connected by a molecule of glycerol. And the key thing is that when you mix this kind of molecule in water, they spontaneously form an orientation where the heads will interact with water and the tails will avoid water. Hydrophilic over here, hydrophobic over here. And so if you can think about that arranged in a kind of spherical way, you wind up having a structure that is a bilayer, 2 layers of phospholipids. And that's the structural framework of cell membranes. We'll talk much more about that when we do Unit 2. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the amount of vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or 5, you need an easier way to study, and that's why we created learn-biology.com. It's got quizzes, it's got flashcards, it's got interactive tutorials about every topic in the AP Bio curriculum. It's got a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence to ace your course and to crush it on the AP Bio exam. Here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. Sign up. You won't believe how much you'll learn. Let's review proteins. So the monomer is an amino acid, and it has a central carbon over here, and connected to that carbon is an amine group over here that makes this basic in its structure, but over here on the other side it's got a carboxyl group and that makes it acidic. So therefore it's an amino acid. There's a hydrogen atom attached to the central carbon, and then there's a variable group or an R group, and there are 20 variations, and that's true of all life. All life is built of the same 20 amino acids. That R group is also called a side chain, and it can be polar, nonpolar, acidic, or basic. There are 4 levels of protein structure. This is a super important topic. We're going to do an overview and then we're going to walk through all 4 of these levels. Primary structure is what's shown over here in A. It's a linear sequence of amino acids. It's genetically determined. The secondary structure, which is shown here and here, those are interactions that involve what's called the polypeptide backbone. When you do a dehydration synthesis and connect one amino acid to the next, to the next, to the next, that chain of carbon, carbon, nitrogen, carbon, carbon, nitrogen, that's the polypeptide backbone. The next level is called tertiary structure, and those are interactions between those R groups. And then finally, there's quaternary, fourth level structure, and that involves interactions between multiple folded tertiary peptides. Okay, let's talk about primary structure. In this diagram, A1, A2, A3, those all represent different amino acids. So the sequence of amino acids that make up a polypeptide, that's what you call multiple amino acids linked together, that's the primary structure. Proteins aren't really built by enzymes in the way that everything else is. They're built by ribosomes. The amino acid, here's one, here's another one, they're connected to one another by peptide bonds. So I was saying before, nitrogen, carbon, carbon, nitrogen, carbon, carbon. That's the polypeptide backbone. All of those amino acids linked together. That's primary structure. What's secondary structure? Well, here you have a nice diagram that shows you what that polypeptide backbone is. And the secondary structure emerges as interactions between the carbonyl groups over here and the amino or amine groups over here within the polypeptide backbone. Now, what happens is that interactions between these amine groups and these carbonyl groups, they form hydrogen bonds and they stabilize certain shapes. One of the shapes to know about is called an alpha helix, and that's kind of a corkscrew over here. So you can see that there's a hydrogen bond that's stabilizing this, hydrogen bond, hydrogen bond. So that forms that shape. Now the other thing that happens is if the parts of the polypeptide chain are either parallel to one another, like this, or anti-parallel to one another, like this, then carbonyl and amino groups can again interact and form hydrogen bonds, and that can lead to a form called a pleated sheet. And that's what we see over here. Tertiary protein involves interactions between the side chains or the R groups. And there are a couple to know about. First of all, there are hydrogen bonds shown at number 2. There are ionic bonds that are shown at number 5 over here. There are covalent bonds, which are shown at number 3. That was between two sulfhydryl groups, another one of the functional groups. And this is a covalent bond that's very important in really tightly holding that protein into a specific shape. And then finally, you have what's called hydrophobic clustering, where non-polar side chains will cluster together, avoiding water. Down here, you see myoglobin, which is an oxygen-storing protein that's found within muscle tissue. That's a tertiary protein folded into a specific shape. And you can see, like over here, there's a whole bunch of alpha helices that are in that structure. Quaternary structure involves multiple polypeptides that interact with one another to create the final form of the protein. So those interactions might be hydrogen bonds, they might be ionic bonds, they might be hydrophobic interaction. In this diagram, actually, you can see all 4 of the levels. So here's the primary structure. Here's an alpha helix, secondary structure. This hairpin turn is actually part of the tertiary structure over here. And then you have multiple polypeptides interacting. This molecule looks a lot like hemoglobin. This is kind of cool in light of our recent history. The spike protein that's on the outside of SARS-CoV-2, and in fact all the SARS viruses, is also a quaternary protein that's made of multiple folded polypeptide chains. An application of what we learned about proteins is this question. Describe the structure and function of hemoglobin and explain the molecular cause of sickle cell disease. So sickle cell disease is an inherited blood disorder. It was one of the first molecular genetic diseases that was really well understood. The molecule that is in question here is hemoglobin. And hemoglobin, its function is to transport oxygen in our red blood cells. The structure, it's a quaternary protein. It's made of 4 polypeptide chains. Sickle cell disease is caused by a recessive mutation. We'll cover that in Unit 5. But the key idea is that there's a mutation that causes the amino acid valine over here to substitute for glutamic acid. And that's an important mutation because glutamic acid is acidic, whereas valine is non-polar. The result of that is that when blood becomes deoxygenated. Those mutated hemoglobin molecules, they form hydrophobic bonds with one another. Well, why? Because they have a hydrophobic amino acid sticking on the outside, and that causes them to do this. They cause fibers to develop within the cells, and that causes the cells to become spiked like this. They're mutant cells, and those cells will then clump up within smaller arteries, and that causes these pain crises. It causes tissue damage. It's a debilitating disease. There are a few other things to know about sickle cell anemia. The first is that during the 30 years that I've been a biology educator, sickle cell disease has been transformed from a childhood disease with certain early mortality to a disease that can be managed with good medical care so that people can live into their 50s, 60s, and even beyond. Sickle cell disease is also a target for gene therapy, something that we'll talk about in Unit 6. And finally, there's a really interesting evolutionary thing where sickle cell disease evolved because having one copy of the gene is actually a benefit that gives you resistance to the tropical mosquito-borne disease, malaria, something that we'll talk about in Unit 7. And now for our last topic in this Unit 1 review, nucleic acids, DNA, and RNA. Note that we'll give an overview now, and we come back to nucleic acids when we cover gene expression in Unit 6. So we'll start by describing the biological importance of the nucleic acids. These are the molecules of genetic information. DNA is the molecule of heredity. It's what passes from generation to generation. It's the molecule that cells pass on as they divide and replicate within a multicellular organism. RNA has other functions. RNA is the hereditary molecule in some viruses, never in cells. And RNA's key role is information transfer, as in messenger RNA. So within a cell, here's DNA, the repository of genetic information. It has its information transcribed into RNA, and then that RNA goes into the cytoplasm where a ribosome will translate that RNA message into protein. RNA is a very versatile molecule. It's not in the form of a double helix the way DNA is. It can take many, many forms, and it can act as an enzyme catalyzing reactions. Ribosomes are essentially catalytic RNA. There are also other molecules called spliceosomes, microRNAs, that play a whole variety of functions. And I finally want to say that this typically isn't put into this unit, but ATP is a nucleotide monomer. It's one of the monomers of RNA, and it's the energy molecule of life. It's how cells get work done. What is the monomer of nucleotides? What's its structure? How are these monomers different in DNA and RNA? The monomers are called nucleotides. We just looked at ATP. All of these molecules have a 5-carbon sugar that's shown at number 2. They have a phosphate group that's shown over here at number 1. And then there's one of 4 nitrogenous bases. So the nitrogenous base doesn't have to have this structure. Note that the phosphate group is connected to a number 5 carbon, whereas the nitrogenous base is connected to the number 1 carbon. When you learn about DNA replication, you'll talk about things like DNA is replicated in a 5-to-3 orientation. And now you know that's about the 5 carbon over here and the 3 carbon over here. In DNA, the sugar is deoxyribose, and there are 4 bases: adenine, thymine, cytosine, and guanine. In RNA, the sugar is ribose, and the bases are adenine, uracil, cytosine, and guanine. So now let's talk about the structure of DNA. DNA consists of 2 nucleotide strands, so easier to see in this flattened out version. Here's one, here's the other. Within each strand, the nucleotides are connected by sugar-phosphate bonds. So here's a sugar, here's a phosphate, here's a sugar, here's a phosphate. But the strands connect to one another by hydrogen bonds. So here you see G connecting with C, A connecting with T, and those are rules to memorize. Adenine, A always bonds with T, C bonds with G. They have complementary shape. Their molecular dimensions are such so that they fit nicely within the helix. That's more of a story for Unit 6. And note that these 2 strands fit together in an anti-parallel orientation. In order for the nucleotides to form hydrogen bonds with one another, they each need to be upside down relative to the other. And that's what anti-parallel is all about. We'll talk about this again more in Unit 6, but just to lay this down for right now, DNA is directional. Notice that in a chain of nucleotides, these are RNA nucleotides, you can tell because there's uracil. The nucleotide sugar binds with a phosphate, and then there's a sugar, there's a phosphate, there's a sugar, there's a phosphate. Well, the enzymes that build DNA, it's called DNA polymerase, and these enzymes, like all enzymes, have an active site. They only can work in certain orientations, and they work completely by feel, and they can only add new nucleotides at the 3' end of a growing nucleotide strand. So that's what directionality is all about. All of these nucleic acids are built in the 5' to 3' direction. Your success in AP Biology starts here. Are you struggling with AP Bio? With Learn-Biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to Learn-Biology.com to find out how you can master your biology course and crush the AP Bio exam. Cells and introduction. Describe the basic structure and fundamental parts of cells. Cells are the basic unit of life. They're the basic unit of structure and function in every organism. In terms of their own structure, they have a membrane which separates the cytoplasm, all the living material inside the cell, from the cell exterior. They always have genetic information in the form of DNA, the double helix, and there are systems that maintain and replicate the cell as a whole and the cell's genetic information, its DNA. One of the most important systems is a system by which the genes in DNA get transcribed into messenger RNA, another nucleic acid, and that messenger RNA is read by a particle called a ribosome and translated into proteins. Proteins are all important. They're the workhorse of the cell, and one of the most important proteins are enzymes. Enzymes control the cell's metabolism. Other proteins might be embedded in the cell's membrane, and others might even be exported from the cell. Compare and contrast prokaryotic and eukaryotic cells. Among living organisms, there's a great divide between the prokaryotes and the eukaryotes. Here's what it's all about. Prokaryotic cells are small and relatively simple. They have no nucleus. Their chromosome is circularized. And what that means is that there's a loop. The beginning and the end meet up. They contain extra chromosomal pieces of DNA that are called plasmids, and they're found in 2 of life's 3 domains, the archaea and the bacteria. Eukaryotic cells are larger and more complex. They're found in the domain that has their name, the eukarya. They have a nucleus. They have multiple linear chromosomes. So here they're shown over here, and you can see the chromosomes here. They have a beginning and an end. And those chromosomes have DNA that's associated with proteins, which is different from the simpler DNA packaging that is found in the prokaryotes. Eukaryotes have mitochondria, and the presence of that organelle is the defining feature of the eukaryotes. And they have many membrane-bound organelles. Membrane-bound means surrounded by a membrane. AP Bio topic 2.3, cell size, an essential topic. Use the relationship between surface area and volume to explain why cells are small. Cells need to have enough membrane surface area to allow for diffusion of substances and molecules in and out. Everything that gets into a cell gets there through the membrane. And it might be counterintuitive, but a small cell has much more surface area relative to its volume than a large cell does. Let's look at the math. If you have a cell that's 1 micrometer, 1 micron on the side, that's its length. Then its surface area, if it's cubic, would be 1 times 1 times 6. Think of the formula for surface area for a cube. Its volume is 1 times 1 times 1, and therefore its surface area to volume ratio is 6 to 1. 6 units of surface area to every 1 unit of volume. Now, if you have a larger cube, one that's 10 micrometers, then its surface area is 600. Wow, that seems like a lot. But its volume is 1,000 because volume's a cubic function and it's going up much faster. So if you look at the surface area to volume ratio, it's 0.6 to 1. As an object gets larger, its amount of surface area relative to its volume decreases. And in this example, the larger cell's surface area to volume ratio is 1/10 that of the smaller cell. And a large cell can't efficiently use diffusion to get the nutrients it needs in and to get waste, carbon dioxide and metabolic wastes out. Why do organisms need to increase surface area in certain tissues? How do they do that? The why is in order to increase the surface for diffusion of molecules or heat. How? You can have thin sheets of tissue, like these thin sheets that make up the gills of a fish that have been dissected out. That's for increasing the intake of oxygen and the diffusion out of carbon dioxide. Or if you think about the ears of elephants, those are big, flat sheets of tissue that enable the elephant, which has very little surface area relative to its volume and lives in a hot place, to get heat out of its body. You can also have highly folded surfaces, like the internal membrane of the mitochondria or the structures that are called villi that make up your intestine. And there's actually folds within folds. Explain in terms of surface area why there are no small mouse-sized marine mammals, and why some marine mammals, like the whales, the largest organisms to have ever evolved, are really huge. Mammals are warm-blooded. Ocean water is cold. Increased size decreases the surface area to volume ratio, so there's less heat lost to the environment. Think about a mouse. It's small. It has a lot of surface area relative to its volume. It would be over here on the graph. It couldn't survive because it would experience so much heat stress in the water. The smallest marine mammals are the otters, and they have a thick coat of fur that helps them maintain their body temperature. Whales have evolved to be larger and larger for a variety of reasons, but one of which is that by being big, they lose relatively less heat to the environment than a smaller animal would. It's all about the surface area to volume ratio. Cellular compartmentalization and the endomembrane system, how eukaryotic cells are organized. What is cell compartmentalization? What are its advantages? Compartmentalization means internal division of a space into sections. Think about the overhead luggage compartment in an airplane. What are the advantages? The first is that it allows a cell to have regions with an internal chemistry that are distinct from the cytoplasm. A great example of that is the lysosome, shown at G over here, and number 7 over here. Lysosomes contain hydrolytic enzymes. Those are enzymes that break things down. Those can't be released into the cytoplasm, or they would break down the cytoplasm, so they are compartmentalized. They're kept in a membrane-bound space. In addition, compartmentalization provides a lot of internal surface area, and that's important for membrane-bound enzymes that are found in the smooth ER or the Golgi that can only do their job when they're embedded in a membrane. Compartmentalization makes that possible. And that's also true of the ribosomes that are found in the rough ER. Compare compartmentalization in prokaryotic and eukaryotic cells. Prokaryotic cells have few compartments, though there are internal regions with specialized structures and functions. Here's a prokaryotic cell. You can see that its internal structure is much simpler than that of a eukaryotic cell. But one notable compartment is called a thylakoid, and it's this over here in a cyanobacterium. Cyanobacterium evolved into chloroplasts. We'll talk about that in a moment. And they have these internal compartments called thylakoids that are essential for the function of photosynthesis. Eukaryotic cells are highly compartmentalized. There are many internal membranes that divide the cell into regions with distinct structures, distinct chemistry, and functions. And examples include lysosomes, the endoplasmic reticulum, the Golgi complex, and vacuoles like this large vacuole in a plant cell. What is the endomembrane system? It's a dynamic connected system of internal membranes and compartments. It includes the nuclear membrane, the rough ER, the smooth ER, the Golgi, lysosomes, and all the little vesicles that interconnect them, membrane and material flow from one compartment to the next. So the membrane that's part of the rough ER might become the same membrane, the same phospholipids that are part of the smooth ER, and those might become parts of vesicles which flow into the Golgi. So those phospholipid molecules are the same. It's dynamic, there's constant exchange. Explain the origin of mitochondria, cellular compartmentalization, and chloroplasts. What we're explaining is the origin of the eukaryotes. When did this happen? About 1.8 billion years ago. Before that, there was nothing on Earth but prokaryotes. How did this happen? It arose as a mutualistic endosymbiosis. Symbiosis means living together, two species living closely associated with one another. Endosymbiosis is when one of those species lives inside the other. We normally think of that as parasitism. That's a negative kind of endosymbiosis, but there's also positive endosymbiosis. That's a mutualistic relationship, and that is what gave rise to the eukaryotes. So what happened is that here we have representatives of the two prokaryotic domains, and an archaeal cell took up a bacterial cell. Now I want to emphasize that there are many scenarios and many diagrams that represent how this happens, and this one is the one that makes most sense to me. So the bacterial cell entered inside the archaeal cell and it evolved into a mitochondrion. That mitochondrion, still kind of the bacteria, secreted vesicles, and those vesicles became the nuclear membrane, shown over here, and it started to develop structures that are part of the endomembrane system, like the endoplasmic reticulum, which is shown over there. So that's how we have eukaryotes. Then later, a second endosymbiotic event led to chloroplasts, which were free-living cyanobacteria, entering into a eukaryotic cell, and that set the stage for algae and plants. What's the evidence that chloroplasts and mitochondria were once free-living bacterial cells that arose through endosymbiosis? What's the evidence for this idea? Like bacteria, both of these organelles, mitochondria and chloroplasts, have their own circular DNA. They replicate themselves through binary fission. That's how prokaryotes replicate themselves. They use their own ribosomes to produce some of their own proteins. These ribosomes resemble bacterial ribosomes in terms of their rRNA sequence and structure, and they have two membranes. The outer one, so the outer bacterial membrane, let's consider it over here, is a vestige of an endocytotic vesicle. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or a 5, you need an easier way to study, and that's why we created learn-biology.com. It has quizzes, it has flashcards, it has interactive tutorials about every topic in the AP Bio curriculum. It has a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence that you'll need to ace your biology course and to crush it on the AP Bio exam. So here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. You won't believe how much you'll learn. Eukaryotic cell parts and functions. Describe the structure and function of the nucleus. The nucleus's function is to store and protect genetic information in the form of DNA. That DNA is wrapped around proteins to form chromosomes. This representation here is only present during mitosis or meiosis when cells are dividing. Otherwise, the DNA is more diffuse and spread out. It's still in chromosomes, but we call the form chromatin. The nucleolus is this dark part in the center of the nucleus, and it assembles ribosomes. The entire nucleus is surrounded by a membrane that separates the chromosomes from the cytoplasm, and the nuclear membrane has pores that allow molecules to enter and leave the nucleus. Important among those are messenger RNA, which leaves the nucleus, goes into the cytoplasm, where it's transcribed into proteins, and various transcription factors that come into the nucleus to turn genes on or to turn them off. Describe the structure and function of ribosomes. Ribosomes are particles composed of ribosomal RNA and protein. They consist of a large subunit and a small subunit. Their function is to read a genetic message encoded in a sequence of messenger RNA and to translate that message into a sequence of amino acids that make up the primary structure of a protein. The details of translation, protein synthesis will occur in Unit 6. What are two locations within a eukaryotic cell where ribosomes can be found? Ribosomes can be free or bound. Free ribosomes, shown at 3, float freely within the cytoplasm, which is number 5. Bound ribosomes, shown at 4, are connected to the membrane of the rough ER. All ribosomes start out as free, though through a process called protein targeting, they migrate to the ER to become bound. They do that if the proteins that the ribosomes are creating are destined to be put inside a vesicle so they can go to the Golgi or the membrane or a lysosome. Describe the structure and function of mitochondria. Mitochondria are shown here in the context of an animal cell. Here's a larger image. The function is converting food energy into ATP. That's the molecule that cells use to get work done. The key structures, there's a chromosome that consists of DNA. There are also ribosomes, which aren't shown in this diagram. Evidence of endosymbiosis. There's an inner membrane. It's highly folded that increases surface area. And that's because there are many membrane-embedded enzymes and proteins, one of which is shown at 5, that are involved in the process of ATP synthesis. The mitochondria, as a former independent cell, has its own cytoplasm. It's called the matrix. It has enzymes for the Krebs cycle and enzymes that perform other functions. There's an intermembrane space, which is an important cellular compartment that enables the mitochondrion to produce ATP. We'll see how that works in Unit 3. And there's an outer membrane that is a vestige of endosymbiosis. Describe the endoplasmic reticulum, the ER, and list its two forms. The ER is an interconnected series of channels found between the nuclear membrane over here and the Golgi body in eukaryotic cells. So here's the ER, and it consists of two forms. There's the rough ER and the smooth ER. Describe the structure and function of the rough and smooth ER. Well, the rough ER is studded with ribosomes. That's what all these little dots are over here. And using those ribosomes, the rough ER synthesizes proteins for inclusion in lysosomes, in other organelles, in the membrane, or for export from the cell. The smooth ER is on the outer side of the ER network, always towards the Golgi, and it lacks ribosomes, but that doesn't mean that there's nothing in that membrane. It has many membrane-embedded enzymes, and the functions of the smooth ER vary by tissue. So it can include synthesis of lipids, converting toxins into soluble forms that can be excreted from the body, carbohydrate breakdown, and synthesis. Describe the structure and function of the Golgi complex. The Golgi is a series of membrane-bound flattened sacs, and you'll remember that flattening increases surface area. The Golgi receives vesicles from the rough and smooth ER and chemically modifies their contents. So here's the rough ER, here's the smooth ER, here's the Golgi, and here's a vesicle bringing something from the ER system to the Golgi. The Golgi packages those modified proteins into vesicles, shown at 6, that are sent to organelles like lysosomes over here at 7, to the cell membrane, or exported from the cell. Describe the structure and function of lysosomes. Here's a lysosome over here, here's a lysosome over here, and one thing that I rarely say, but I'll let you know that you will almost certainly never have to identify a lysosome on a diagram on the AP Bio exam. It's just a big vesicle. Lysosomes are membrane-bound organelles that contain hydrolytic enzymes. They're only found in animal cells. They carry out intracellular digestion. So like, for example, Here you see a particle. It actually looks like a cell that's being engulfed by the membrane. It'll be packaged into a vesicle, and that vesicle will be sent to a lysosome where hydrolytic enzymes will break this down. The lysosome also recycles worn out, damaged, or excess organelles and molecules, and it plays a key role in a process called apoptosis. The P is silent. That's programmed cell death, and we'll talk about that later in the course. The cytoskeleton is a dynamic network of protein fibers, and those fibers make up the bones, the skeleton of the cell. In the same way as our skeleton, our bones, working with muscles enables us to move, the cytoskeleton is a dynamic network that enables the cell to move, both internally, so it enables cells to move materials and organelles within themselves, and it also enables the cell to move their membrane so that they can do things like endocytosis, where cells can engulf things by moving their membrane, or also moving their entire bodies. This is an organism called an amoeba, and it moves by extending parts of the membrane over the surface, enabling it to crawl. How does it do that? Through the cytoskeleton. What are centrosomes and centrioles? Centrosomes are the organelle. They contain 2 centrioles, and the function is creating spindle fibers, that's what these are over here, that separate chromosomes during mitosis and meiosis. The parts that we've talked about so far have all been represented within animal cells, but most of those parts are also found in plant cells. The one exception is the lysosome, unique to animal cells. Let's describe the function of the central vacuole, this large structure right here. It's only in plant cells. Its functions include water storage, storing and releasing macromolecules, sequestering waste products, and maintaining turgor pressure. We'll talk about this in the context of osmosis, the movement of water, but as water flows into a cell, it creates outward pressure that keeps plant cells full, firm, upright, they're not wilting. Describe the evolutionary origin and function of chloroplasts. Here are chloroplasts over here. Here's a single chloroplast. Chloroplasts are the endosymbiotic descendants of free-living photosynthetic bacteria, and that's why they have their own DNA, their own ribosome. That's why they have 2 membranes. Their function is creating carbohydrates through photosynthesis. We're going to leave their detailed structure and how they work for Unit 3 when we discuss photosynthesis. Describe the chemical composition and the function of the plant cell wall. The cell wall is composed primarily of cellulose, which is a polysaccharide. It's that polysaccharide that we can't digest, but which ruminants, cows, goats, can digest because they have these symbiotic relationships that enable them to break that down and release food energy. The major function of the cell wall is it acts as a pressure vessel that prevents overexpansion in response to inward flow of water, osmotic pressure. So here we see as water flows into a plant cell, the central vacuole will expand and it'll look like this. The cell will be full of water, and that water will push against the cell wall, keeping the plant cell full and firm, avoiding wilting. So that's a good thing for the plant. And the cell wall is the primary component of wood and the water conducting tubes in plant stems. Membrane structure and function. What is the function of the cell membrane? The cell membrane separates the highly organized contents of the cell from the cell's environment. Cells are open systems. They need to let things like food in. They need to let things like wastes out. The membrane is how that's controlled. That's because the membrane is a selectively permeable boundary. It allows the passage of some substances and particles through, but not others. That selectivity is really the key to life. Describe the structure of phospholipids and their role in cell membranes. Phospholipid structure, there's a hydrophobic nonpolar tail region. Here's the structural formula. Here's a representation. There's a hydrophilic polar head. Notice that the head has this phosphate group over here with its negative charge. The whole thing is held together by this 3-carbon molecule called glycerol. In solution, when phospholipids are mixed with water, the heads of the phospholipids bond with water molecules while the tails form a water-free zone. So interacting with water, water-free zone over here. That creates a bilayer. That's the basic framework of the membrane to which proteins and other molecules are added. We'll see that further on. And the phospholipid bilayer is stabilized by weak bonds, van der Waals bonds, that occur between the tails. Describe the fluid mosaic model of cell membranes. This model posits that the membrane is composed of phospholipids plus proteins plus cholesterol in motion. So why is it called fluid mosaic? Well, it's fluid because the components are moving around laterally within the plane of the phospholipid bilayer, which you can see here. It's a mosaic because it's composed of a variety of pieces in the same way as a mosaic tile is composed of different tiles with different colors that get put together to form the art. Within the phospholipid bilayer, you have phospholipids that are at 4 and 9. You have proteins that are at 1, 3, 5, and 6. And you have molecules like cholesterol, which are shown at 7. And then inside and outside the phospholipid bilayer, you might have additional molecules attached to proteins or phospholipids. And those might be glycoproteins, which are not shown, but at 8 you have a glycolipid. Glyco means it's a carbohydrate and it's attached to these fatty acids right over here. Describe how proteins fit into the cell membrane. There are 3 ways that proteins can fit in. And if you think about hydrophobic, hydrophilic, what binds with what, it makes a lot of sense. So there are transmembrane proteins. That's shown over here and over here. Here, notice that beautiful alpha helix that you should remember from our Unit 1 review. There's a hydrophobic core that fits into the nonpolar inner portion of the membrane. So these would be hydrophobic amino acids over here and over here. And there'd be hydrophilic regions that extend into the cytoplasm and the membrane exterior. There are integral proteins, such as the one at 5, and these proteins would have a nonpolar region that extends into the hydrophobic membrane middle and a single hydrophilic region that juts into the cytoplasm, as this one does, or into the cell exterior. And then finally, you have peripheral proteins, like this one that's shown at 3, and they attach to phospholipid heads that are either on the cytoplasmic side of the membrane or in the cell exterior. These terms, transmembrane, integral, peripheral, they're not important in and of themselves, but you should be able to understand the concept of how things fit into the membrane. Membrane transport, how things get across the cell membrane. Diffusion is the tendency of molecules to spread out from where they're more concentrated to where they're less concentrated. So here you have some substance, more concentrated over here, less concentrated over here. Here's ink going into a beaker, more concentrated, and then less concentrated. This happens spontaneously. It's based on the kinetic energy of the molecules. And a good way to envision this is to think of molecules as flowing down a concentration gradient from higher concentration to lower concentration. Explain how cells control what diffuses across their membranes. Diffusion is called passive transport, and that's because it requires no cellular energy. There are 2 forms. The first is simple diffusion across the phospholipid bilayer. That's what's shown up here. There are 2 things that can do simple diffusion. The first are any small non-polar molecules such as oxygen, nitrogen, carbon dioxide. The second are non-polar substances such as steroid hormones or fats. Facilitated diffusion is for polar molecules and ions that can't diffuse across the phospholipid bilayer. So they require protein channels. And these are transmembrane proteins, they go all the way through, that only let specific molecules or ions pass. So over here you see one for glucose, over here you see one for amino acids, those are both polar molecules, and here you see one for an ion such as calcium. There'd be others for ions such as sodium or potassium. Compare and contrast active and passive transport. As you do, explain what powers each process. Passive transport is what we just discussed. It's when molecules or ions flow down a diffusion gradient. Here, more concentrated, to less concentrated. And that's what it's all about. It's diffusion from high concentration to low concentration. It relies on the kinetic energy of the diffusing molecules or ions. It doesn't require cell energy. As you can see here, molecules are flowing down their concentration gradient. That's different from active transport, where we're pumping a molecule or ion up its concentration gradient from lower concentration to higher concentration. Over here, lower concentration to higher concentration. That requires an energy expenditure on the part of the cell. Usually that's ATP that's being converted into ADP and phosphate to power the pumping process, but also the flow of electrons can power the pumping of protons, and that's important in the creation of ATP itself. Compare and contrast endocytosis and exocytosis. These are both forms of bulk transport. In endocytosis, the membrane pinches in to surround a particle or some extracellular fluid, creating a cavity that can become a vesicle. In exocytosis, a vesicle containing something fuses with the membrane, and then that contents is dumped outside the cell. Both of these processes require energy on the part of the cell, and they both require the involvement of the cytoskeleton, which is changing the shape of the membrane. What is membrane potential? How do cells create membrane potential? How is membrane potential used? Membrane potential is an electrical charge across a membrane that creates a voltage difference. How is it created? Through cells expending energy to pump ions across their membranes. A key example, and this is a preview of a topic that we'll get into in Unit 3, is how mitochondria pump protons from the mitochondrial matrix to the intermembrane space. That gives the intermembrane space a positive charge relative to the matrix. It has all these protons inside of it. And that creates an electrochemical gradient, a voltage difference. And that pulls protons through this channel, it's called ATP synthase, back into the matrix, and that powers the creation of ATP. Some other examples, chloroplasts do pretty much exactly the same thing. And there's also a 70 millivolt charge across the membranes of nerve cells. That makes nerve impulses possible, and that's how I am able to think and teach biology, and how you are able to learn it. Is biology mind-blowing or what? I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Tonicity and osmoregulation. That's the College Board's term. I wish that was just called osmosis and its consequences. So let's start by defining what osmosis is. It's the diffusion of water. Water diffuses like everything else, and that means that water is going to move from higher to lower concentration. Water is the solvent of life, so we have to pay attention to how it moves. Water flows down its diffusion gradient, just like everything else, from hypotonic to hypertonic. Those are essential terms to know. Hypotonic means relatively more water and relatively less solute. And these terms are not absolute. They're always relative to another solution. So in this diagram, this side is hypotonic. It has a higher percentage of water molecules. It has a lower percentage of solute molecules. This side is hypertonic. And that means that it has relatively less water, a lower percentage of water molecules, and a higher percentage of solute molecules. But that's this compared to this. Now, what is that going to do? That's going to have water move down its concentration gradient, which means it'll move from here to here. After osmosis takes place, it'll look like this over here. This level will go down. This level will go up. The water actually is getting pushed up on this side. That's called osmotic pressure. It has enormous consequence. And the thing for you to memorize is that water diffuses from hypotonic to hypertonic. Here's a fabulous and fun example. A student places a gummy bear in a cup of water overnight. The next day, the gummy bear has expanded. Explain. Well, the gummy bear is mostly sugar. The water in the cup is 100% water. Water always flows from hypotonic to hypertonic. So water is going to flow from the hypotonic solution in the cup into the gummy bear. The gummy bear will expand because of osmotic pressure. Osmosis in plants. Use the principles of osmosis to explain each of the images below. On the left side, the cell is hypotonic to the environment. Water leaves the cell, the membrane peels away from the wall. That's also known as plasmolysis. The vacuole shrinks and the plant wilts. How could the environment be hypertonic to the cell? How could the cell be hypotonic to its environment? This environment has to have, for example, a lot of solutes dissolved in it so that it has a lower water concentration than is in the cytoplasm. That'll cause water to go from the hypotonic cell to the hypertonic environment. In the middle, the cell is isotonic to its environment. That's a new term. Isotonic means that the solute concentration and the water concentration are the same on both sides of the membrane. So water enters and leaves and enters at the same rate. And on the right side, the cell is hypertonic to its environment, so water flows into the cell. That osmotic pressure in the case of plants is called turgor pressure. It causes the vacuole to expand. It pushes the membrane against the cell wall. And this is a good situation for plants to be in. It makes them full, firm, happy. They're not wilted. Osmosis in animal cells. Use the principles of osmosis to explain each of the images below. Cell number 1. The cell is hypotonic to its environment. Water leaves the cell. Water always flows from hypotonic to hypertonic, so the cell shrivels up. In situation number 2, the cell is isotonic to its environment. Water enters and leaves the cell at the same rate. This is an important condition for animal cells because they have a membrane but not a wall. Cells that are kept in a tissue culture need to be kept in an isotonic solution. And in situation 3, the cell is hypertonic to its environment, and therefore water is flowing into the cells, and because there's no cell wall, the membrane can't really stop the inward force, and the cell will ultimately burst. Explain the function of the contractile vacuole in freshwater protists such as paramecia. A protist is a eukaryote that's not a plant, an animal, or a fungus. Protists in freshwater are hypertonic to their freshwater environment. That means that the cytoplasm has more solutes dissolved in it than the lake or stream that they're swimming in. And as a result, water moves into these cells by osmosis, flowing from hypotonic to hypertonic. To osmoregulate, that's a new term. Osmoregulate means to regulate your osmotic balance. The contractile vacuole, that's this organelle over here, fills with water and then contracts to expel water from the cell. If the environment becomes more hypertonic, then the cell can adapt by decreasing its rate of contractile vacuole contraction and do the reverse in more hypotonic environments. Describe the structure of leaf stomata. Stomata are pores that are on the underside of a leaf. If you took a leaf and you did a microscopic cross-section, you'd see layers of cells. The top layer and the bottom layer are responsible for waterproofing the leaf, and they're covered with wax. But nevertheless, the leaf needs to be open to its environment in order to exchange gases and other material. Each stoma, which is singular, stomata is plural, is formed by 2 guard cells. So here's one guard cell, here's the other guard cell. And when there's sufficient water, these guard cells buckle outward, like that, and that creates a pore that allows carbon dioxide to enter the leaf for photosynthesis, but also allows water vapor to escape. These stomata can be regulated. They can close in response to environmental cues, including water stress, and then open when water is abundant. Explain how guard cells are regulated in order to open and close the stomata. When water is available, cells adjacent to the stomata pump potassium ions into the guard cells. So we're talking about these cells and these cells, the ones that are adjacent to the guard cell, and they're represented like this over here. So the pumping of potassium makes these guard cells hypertonic to the adjacent cells. Water follows by osmosis, causing the guard cells to buckle and open up. When water is scarce, the pumping stops, potassium ions flow out of the guard cells, and water follows by osmosis, causing the stomata to close down, to revert back to this form. What is water potential? Water potential is a concept that's really closely related to osmosis and osmotic pressure, but it's more quantitative. The symbol for water potential is this Greek letter psi. So water potential is a measurement of water's tendency to move from where it is to where it's not. Adding solute decreases a body of water's water potential. Adding pressure increases water potential. And water will flow from areas of higher water potential to areas of lower potential. So if you look at this device over here, it's called a U-tube, and these two parts are separated by a membrane that's permeable to water, but not to the solute. So here we are, we're adding solute to this side of the U-tube. That makes this side hypotonic, but that's another way that you can say it has higher water potential, and water is going to move from higher water potential to lower water potential. That's going to force up the level of water over here. That's going to lower it over here. Could you have explained this entire thing with just hypotonic and hypertonic? Well, I just did, but you'll see that water potential is more quantitative, and it allows for other factors to be included, such as pressure. Explain the formula for water potential. Here it is over here. In plain English, it's water potential equals solute potential plus pressure potential. So psi is water potential, psi sub s is solute potential. And remember that adding solute to water decreases its water potential. Psi p is pressure potential, and adding pressure, like if you pressed on a syringe, that increases water potential. So let's look at these two examples over here. So we've added solute to this side of the U-tube. Pressure potential is 0. There's nothing pressing down on either side, but adding solute lowered the solute potential over here to negative 0.23 megapascals, and that's the unit that's used for pressure. So the total water potential over here is minus 0.23 megapascals. The water potential over here is just 0. There's no pressure, there's no solute, so water moves from higher water potential to lower water potential. That's why it's moving from this side of the U-tube to this side. In this example, we have a potato cell or potato tissue that's been placed in water. And so again, there's no pressure on either side, it's just an open container. But the cell has a negative 1.0 megapascal solute potential, so the total water potential is negative 1.0. This is just water, so the water potential is 0 over here. Water moves from the beaker into the cell, which has a lower water potential, and that's causing water to flow into the cell. It's causing the cell to expand. All of these things related to the cell wall. In a typical lab in AP biology, you put potato tissue into water or solutions of various concentration, and you can see what happens, and you can actually quantify it based on water potential. That's what water potential is used for. Your success in AP Biology starts here. Are you struggling with AP Bio? With Learn-Biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to Learn-Biology.com to find out how you can master your biology course and crush the AP Bio exam. Topics 3.1 to 3.3, Enzymes. To help you study, I've put together a checklist that you can download at apbiosuccess.com/checklist. Describe the key properties of enzymes. Enzymes are usually proteins. There are some RNAs that act like enzymes that catalyze reactions in cells. They lower the activation energy of the reactions that they catalyze, increasing the rate of those reactions. So in this diagram, you see a reaction that's catalyzed by an enzyme, number 2, and a reaction that's not catalyzed at number 1. And the difference is that the activation energy for the enzyme catalyzed reaction is much less than the non-enzyme catalyzed reaction. Enzymes are highly specific because their active site complements the shape and charge of their substrate, which is the substance that an enzyme acts upon. Here's an active site. Here's the substrate. This is the enzyme as a whole. It would be a large protein. Here's the enzyme interacting with the substrate. And here we have the products. Enzymes are both highly specific and have a narrow set of conditions where they can function at or near their optimum. Explain. Enzymes are proteins with secondary, tertiary, and quaternary level structures that involve hydrogen bonds, ionic bonds, and hydrophobic clustering. Changing pH, temperature, or ion concentration interferes with these bonds, changing the shape of the active site, keeping the enzyme from binding with its substrate. Enzymes therefore have a pH, ionic, or temperature optimum at which the shape of their active site best fits their substrate. Environmental change can cause denaturation, a change in the shape of the enzyme that lowers or completely negates enzyme function. Describe how enzyme activity is affected by changes in the pH of its environment. Most enzymes have a pH optimum where they operate at peak efficiency. Here's the optimum right here. As pH moves above or below the optimum, enzyme performance drops. And this is the rate of enzyme activity, and you can see that as the pH drops, it goes down. As the pH increases, it goes down. Why? It's for all the reasons we talked about in the previous slide. It's that enzymes are proteins. If you change the pH, you disrupt the bonds that hold that protein in its specific shape. The result is denaturation and less good fit between the enzyme and its substrate. Describe how enzyme activity is affected by changes in the temperature of the enzyme's environment. Up to a certain point, enzyme activity increases with temperature, and that's because there's more kinetic energy that increases molecular motion, and it increases the chance that the enzyme will bind with its substrate and therefore be able to catalyze the reaction. But at a certain temperature, beyond 2 in the graph, the enzyme will denature. It'll change its shape, reducing the enzyme's catalytic abilities because it'll no longer be able to bind with its substrate. What's the difference between reversible and irreversible enzyme denaturation? Reversible denaturation is where the restoration of optimal conditions restores the enzyme's function as it regains its optimal shape. If you can imagine that an enzyme has an optimal shape at 7, you move it up a little bit, the enzyme starts to denature, but then if you restore the pH, the enzyme shape might go back to its previous form, thereby going back to its optimal rate of efficiency. But irreversible denaturation is where the enzyme shape is permanently changed and its catalytic ability is destroyed. And this isn't exactly with enzymes, but if you think about what happens when you cook an egg, the egg white goes from clear to a solid, and it'll never go back, even once you cool it back down. That's irreversible denaturation of a protein. Imagine the same for an enzyme. Explain how enzyme activity is affected by substrate concentration. With low substrate concentrations, the probability of the enzyme meeting its substrate is low, and the product is produced at a very low rate. As substrate concentration increases, the collision and the reaction rate will also increase, but at a certain point, you get to a saturation point. And at that point, all the enzymes have their active sites interacting with substrates, so there's a peak in the rate, and you don't go any higher. Compare and contrast competitive and non-competitive inhibition. In competitive inhibition, a foreign molecule, one that's not part of the cell or the organism, that's not the enzyme substrate, so this is number 4 over here, blocks the enzyme's active site. And that keeps the substrate from binding. Here's the substrate, and that inhibits the rate of the reaction. It's inhibiting by competing for the active site. In non-competitive inhibition, which is shown over here, a foreign molecule, not one that's part of the organism, binds away from the active site at a region that's called the allosteric site over here. So here's the allosteric site, here's the allosteric site that's occupied by this foreign molecule. Binding at the allosteric site causes a ripple effect throughout the protein that causes a change in the shape of the active site. Therefore, the substrate can no longer bind at the active site, and that diminishes or blocks enzyme activity. Topic 3.4, cell energy. I'm Mr. W from learn-biology.com, where we believe that successful learning requires interaction and feedback, and we're so sure of that that a subscription to our website comes with a money-back guarantee. What is a metabolic pathway? A metabolic pathway is a linked series of enzyme-catalyzed chemical reactions occurring within a cell. So here you can see that these are all separate reactions, but they're linked together. Above, 1, 2, and 3 are all enzymes. A is the initial reactant. B and C are what we call intermediates, and D is the final product. Examples of metabolic reactions that you'll come to know well include glycolysis, the Krebs cycle, the Calvin cycle, all of which we'll deal with in this unit. And these reactions can be linear. So glycolysis is a linear reaction. You have a beginning point and an end point, or they can be cyclical, like the Krebs cycle and the Calvin cycle. So for example, in the Krebs cycle, this compound over here, oxaloacetate, it's the beginning compound, gets modified, and it also comes back and it's the ending compound. A similar thing happens in the Calvin cycle. What are autotrophs? Compare and contrast photoautotrophs with chemoautotrophs. Autotrophs are organisms that can produce their own food. We are not autotrophs, but plants and certain bacteria and archaea are. So photoautotrophs include plants and cyanobacteria. They use the energy and light to create organic compounds that they need to survive through photosynthesis. Whereas chemoautotrophs include some bacteria, some archaea. Archaea is that third domain. The energy for their life processes comes from a process called chemosynthesis, and that involves oxidizing inorganic substances, including iron, sulfur, or hydrogen sulfide. What are heterotrophs and how do they get the energy and matter they need to live, grow, and reproduce? Heterotrophs capture the energy present in organic compounds produced by other organisms. They can be ecological consumers, decomposers, or parasites, and they get their energy and matter by metabolizing the organic compounds in organisms that they eat or absorb, or in the dead remains of other organisms. Here's a heterotroph, and here's another heterotroph. What's the difference between an exergonic reaction and an endergonic reaction? Exergonic reactions release energy and increase entropy. So here's an exergonic reaction over here, and the energy of the reactants is less than the energy of the products. And you can't see this, but like for example, if you burn a piece of paper or a piece of wood, you start with cellulose over here and you wind up with many unorganized atoms of carbon dioxide and water. So that's an increase in entropy because you've decreased organization. Cellular respiration, most hydrolysis reactions are exergonic reactions. Endergonic reactions require energy and decrease entropy, and examples include photosynthesis or almost any dehydration synthesis reaction. Describe the structure and function of ATP. Explain how ATP can be used to store and release energy. The structure of ATP involves a 5-carbon sugar that's called a ribose, a nitrogenous base that's called adenine, and 3 phosphate groups. The function of ATP is to power work within cells. Every cell makes its own ATP. There's no sharing of ATP between cells. And to store energy, what happens is that cells take energy from food during cellular respiration or light during photosynthesis and use that to combine ADP and a phosphate group into ATP. And to release energy for work, cells remove a phosphate group from ATP. They break off this terminal phosphate group, and that creates ADP and phosphate, and that makes energy available to do cellular work. What is energy coupling? Energy coupling is the linking of an exergonic reaction to an endergonic reaction, and that linking drives the endergonic reaction forward. Here's an example. Cellular respiration, which is exergonic, drives the formation of ATP from ADP and phosphate. So this exergonic reaction is driving this endergonic one. And example number 2 is the exergonic conversion of ADP and P is coupled to endergonic processes such as active transport. We discussed that in our Unit 2 review video. That takes energy. So ATP to ADP and phosphate makes this endergonic reaction possible, or muscle contraction, also an endergonic reaction. It's made possible by the breakdown of ATP to ADP and phosphate. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or a 5, you need an easier way to study, and that's why we created learn-biology.com. It has quizzes, it has flashcards, it has interactive tutorials about every topic in the AP Bio curriculum. It has a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence that you'll need to ace your biology course and to crush it on the AP Bio exam. So here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. You won't believe how much you'll learn. Photosynthesis, the big picture. What happens during photosynthesis? What's its chemical equation? Is it endergonic or exergonic? In photosynthesis, using light energy, here's the sun, photoautotrophs like plants combine carbon dioxide and water to create carbohydrates. That's what the plant is made of. Oxygen is released as a waste product. It's the source of biomass and the base of almost every food chain. The formula is 6 CO2, 6 carbon dioxides, plus 6 H2O, 6 waters, with light energy to power the reaction are combined into glucose, C6H12O6, and 6 oxygens. This is an endergonic reaction for 2 reasons. It takes 2 low energy inputs, carbon dioxide and water, and converts them into a high energy product, glucose. It reduces entropy. That means it increases organization. And you can sort of count that out. So there are 12 molecules on this side of the equation, and there are 7 on this side. So we've taken something that was disorganized, made it into something more organized. A highly unorganized carbon dioxide, it's diffuse, it's a gas, and it's made into solid matter. And that's a huge decrease in entropy. When did photosynthesis first evolve? What are some of the consequences of photosynthesis? In terms of when, based on fossil and chemical evidence, about 3.5 billion years ago, that's relatively soon after the emergence of life, 3.8 billion years ago, its consequences were vast. First of all, when the Earth first formed, there was no oxygen in the atmosphere. It's photosynthesis, which splits apart water to release oxygen, that created the oxygen-rich atmosphere that made our aerobic metabolism possible. And it also created an ozone layer. That's a protective layer in the atmosphere that shields us from ultraviolet radiation, and that made life on land possible. So we owe everything to photosynthesis. What are the 2 phases of photosynthesis and what does each accomplish? We start with the light reactions. That's on this side of this diagram, and it converts light energy into chemical energy, and that chemical energy is in the form of ATP and NADPH. You already know about ATP. NADPH is like NADH. It's an electron carrier. The Calvin cycle is the second phase of photosynthesis, and it converts the chemical energy that's in NADPH and ATP into carbohydrate, and it does that by using carbon dioxide as an input, and it fixes that low energy gas into high energy sugars. Describe the role of chlorophyll in photosynthesis. Explain the absorption spectrum of chlorophyll and other pigments. Chlorophyll is the pigment that absorbs light energy in photosynthesis. And here you can see its structural formula. It's got a hydrocarbon tail that enables it to fit into the phospholipid bilayer of the thylakoids. And it's really this structure over here, this nitrogen ring with a magnesium in the center, that enables chlorophyll to help plants convert light energy into electrical energy, as we'll see in a little bit. An absorption spectrum shows the amount of light absorbed at different light wavelengths by a pigment, by a substance that absorbs light energy. And chlorophyll has two forms. They're different based on this functional group. Here's chlorophyll B, here's chlorophyll A, and you can see that they both absorb most energy in the blue part of the spectrum and in the red part of the spectrum, but very little in the green part of the spectrum. And that's why leaves are green, because leaves are reflecting green light, whereas they're absorbing other light wavelengths. There are other pigments that are also involved in photosynthesis. One's called a carotenoid, and they absorb other wavelengths. What is the action spectrum of photosynthesis? The action spectrum, which looks a little bit different from the absorption spectrum that we just looked at, shows how various light wavelengths drive photosynthesis. And blue and red drive the most photosynthesis, and green drives very little. This was determined by the Engelmann experiment. Thomas Engelmann in the 1800s did a cool experiment where he grew a filament of algae under light from a prism that divided the light up into its various wavelengths. And aerobic bacteria grew around the filament best in the blue and the red part of the spectrum. And they were able to do that because that's where the most oxygen was being produced. You can, in the lab, and I actually hope that you did, recreate this experiment with the famous photosynthesis spinach leaf disc experiment, where these discs of spinach leaves will rise based on the amount of oxygen that they produced. And you can set different variables like the intensity of the light or the wavelength of the light. Connect the structure of chloroplasts to the reactions of photosynthesis. So, chloroplasts, where do you find them? So here they are. This is a cross-section of a leaf. These are cells within the top part of the leaf, and these are chloroplasts. There are many per cell. There's only a couple shown here. And in terms of the structure of the chloroplast itself, it has an outer membrane and an inner membrane. The outer membrane is a vestige of the evolutionary origins of chloroplasts. There's DNA, which is a vestige of the fact that this was once an independent living cell. There's also ribosomes. They're there for the same reason. And then there are thylakoids, shown at number 5 over here. Those are membrane-bound sacs, and they contain the membrane-bound photosystems and chlorophyll for the light reactions of photosynthesis. They're organized into these stacks called grana, and surrounding them is the stroma, which is essentially the cytoplasm of the chloroplast. It contains DNA, it contains ribosomes, and it's where the Calvin cycle occurs, so that's where carbohydrates are actually created. The light reactions of photosynthesis, topic 3.5 of AP Biology. What do the light reactions produce? Where do these reactions occur? What are the inputs and the outputs? The light reactions convert the energy in light into the chemical energy of NADPH and ATP. NADPH is an electron carrier. It's like NADH in cellular respiration. ATP is the molecule that cells use to build things. It's the workhorse of the cell. Where does it occur? It occurs in the thylakoids. Oxygen is the waste product, as well as NADPH and ATP. And the inputs are light and water. The outputs of the Calvin cycle are the inputs of the light reactions. So NADP plus and ADP and P are the inputs. Those get fed into the light reactions. ATP and NADPH go out. What are the key structures involved in the light reactions? So we have a chloroplast over here, and then at N we have grana and a single thylakoid membrane. So this whole thing here is a thylakoid membrane. Within the thylakoid membrane there are photosystems. Those are complex assemblies of proteins, and they have embedded chlorophyll molecules. Those little green dots are chlorophylls. And those photosystems, those are the things that actually convert light energy, shown here at A and shown here at D, into a flow of electrons. This whole array is kind of like a solar panel that's converting light energy into electricity. They're also splitting water molecules, and that happens in photosystem 2. Let's just get this out of the way right now. In the organization of the photosystems in the thylakoid membrane, photosystem 2 comes before photosystem 1. For years, biology students have been memorizing that, and you have to memorize it too. So this is the electron pathway through which electrons flow, and at one point those electrons flow through proton pumps. They're labeled as cytochromes, but there's other stuff going on too. And what these do is they pump protons from the stroma into the thylakoid space. And over here's an enzyme called ATP synthase. And as the protons that are trapped here diffuse through this, they generate ATP. This is where NADPH is created. This is an overview. We're going to go through the details right now. Describe how the light reactions of photosynthesis create ATP. Photoexcitation of chlorophyll in photosystem 2 leads to a flow of electrons along an electron transport chain in the thylakoid membrane. That electron transport chain, that's an electrical current, and it powers a device. In this case, the device is a proton pump that's embedded in the thylakoid membrane, and that pumps protons from the stroma into the thylakoid space. So here's the stroma, here's the thylakoid space. We're pumping from the stroma into the thylakoid space, and that creates a chemiosmotic gradient. Chemiosmotic, what does that mean? Well, there are all these protons that are over here, and there are very few over here. It took energy to do that. And that gradient is a diffusion gradient, and it's also an electrical gradient. And that causes these protons to want to diffuse from the thylakoid space back to the stroma. They can't do it through any part of the thylakoid membrane except through this channel that's called ATP synthase. The ATP synthase channel is also an enzyme, and as protons diffuse through, the kinetic energy of those protons is used to power an endergonic reaction of taking ADP and phosphate and making it into ATP. Now note that there's also this water splitting complex that's part of photosystem 2. And what it does is it takes water molecules, splits them apart to create oxygen, that's a waste product, but also to create protons. And those protons accumulate in the thylakoid space. That enhances the gradient, and it powers additional ATP production. Describe how the light reactions create reducing power, NADPH, that can be used in the Calvin cycle. So we're going to start over here. We're looking at photosystem 1. Photoexcitation of chlorophylls in photosystem 1 is how the process starts. That creates a flow of electrons that's flowing through the electron transport chain of photosystem 1. And over here, those electrons flow to this enzyme. It's called NADP plus reductase, and that reduces NADP plus into NADPH. Reduction, that's a thing from chemistry. Reduction is gain of electrons, so that NADP plus is going to gain electrons and hydrogen, making it into NADPH. And why? Because during the Calvin cycle, that NADPH provides the electrons and hydrogens that reduce carbon dioxide into carbohydrates. Use the Z-scheme to summarize the light reactions. So the Z-scheme is a graphical representation of everything that happens in the light reactions. This axis over here, the y-axis, shows electron energy. So what happens? Light drives electron boosting from photosystem 2. Remember, photosystem 2 comes first. And so that electron goes to a much higher energy level. And at the same time, water is split apart into protons and oxygen gas. Then there's electron flow through the electron transport chain of photosystem 2, and that goes through proton pumps that power the synthesis of ATP from ADP and phosphate. Those electrons arrive at photosystem 1. They're relatively low energy at this point. You can see that by their position on the graph. But light comes in and it stimulates chlorophylls, and another electron gets boosted to a high energy level into what's called a primary electron acceptor that's in the thylakoid membrane, and that passes it off to the electron transport chain of photosystem 1. It flows to the enzyme NADP plus reductase, and NADP plus reductase creates NADPH from NADP plus and a proton. So we have the two products of the light reactions, ATP and NADPH, beautifully explained by the Z-scheme. Photosynthesis part 3, we've covered the big picture and the light reactions, and now we're going to talk about the Calvin cycle. What are the 3 phases of the Calvin cycle? So let's remember the Calvin cycle takes the products of the light reactions and carbon dioxide and uses it to create sugars. So that occurs in 3 phases. And the first phase is called the carbon fixation phase. Carbon dioxide gas is brought essentially into the biosphere. That is followed by the energy investment and harvest phase, where matter is actually pulled out, and that matter becomes part of the plant and ultimately part of you. And then finally there's the regeneration of the starting compound. This is a cyclical reaction. This compound, RUBP, ribulose bisphosphate, is at the end and it's at the start. Now we'll go into each phase. Describe what happens during the carbon fixation phase of the Calvin cycle. The fixation phase begins as carbon dioxide is combined with RUBP. That is a reaction that's catalyzed by the enzyme Rubisco. Fun fact, that might be the most abundant protein on Earth. It creates a 6-carbon product, which isn't shown. So think about this. RUBP has 5 carbons. Each of these black dots represents a carbon atom. CO2 has 1 carbon. You'd think that would create a 6-carbon product, but immediately that 6-carbon product dissociates into 2 3-carbon molecules. So that's how we end the carbon fixation phase. Describe what happens during the energy investment and harvest phase of the Calvin cycle. We ended the carbon fixation phase with this 3-carbon molecule, and that 3-carbon product is reduced and phosphorylated. So here's a phosphorylation. In other words, ATP contributes a phosphate to this molecule. This had 1 phosphate group. This one has 2 over here. So that's a phosphorylation. And reduced. So NADPH donates an electron to this molecule. So at the end, we have this molecule G3P, glyceraldehyde 3-phosphate. It's also called PGAL, phosphoglyceraldehyde. Those are both interchangeable names. And the key thing is that this has a lot more energy than this. Why? Because it was given that energy from ATP and NADPH. This molecule can now be harvested, taken out of the Calvin cycle, and used to build plants. This is the origin of the biomass in almost any ecosystem. It's the origin of your biomass because that's ultimately where it came from. Describe what happens during the last phase of the Calvin cycle. Before we do that, I want to talk about a way to think about the entire Calvin cycle that's really going to make you think about it in a more sophisticated way. And that way pays attention to the number of carbon atoms that are present at each stage of the cycle. So we talked about how during carbon fixation, RUBP is combined with CO2. But a more proper way and correct way to think about it is that it's 3 RUBPs. RUBP is a 5-carbon compound, so that's a total of 15 carbon atoms, get combined with 3 carbon dioxides. 15 plus 3 is 18. And we've talked about how the 3 6-carbon compounds immediately dissociate into 2 3-carbon compounds. So what we have over here at the end of carbon fixation is 6 3-carbon molecules. And that's a total of 18 carbons. Makes sense. 5 times 3 is 15, plus 3 is 18, and we've got 18 carbon atoms over here. Now, in the energy investment and harvest phase. During energy investment, we're just going to phosphorylate and we're going to reduce. We're adding energy, but we're not adding carbon. So what we wind up with over here is 6 molecules of G3P, glyceraldehyde 3-phosphate. 6 times 3 is 18. We had 18 over here, we have 18 over here. But what we're going to do in the harvest phase is we're going to pull one of those G3Ps out. So 18 minus 3, we're left with 15 carbon atoms, 5 times 3. Now during the next phase, these 5 G3Ps are rearranged by a variety of enzymes. That's why I have multiple arrows over here. And they're rearranged into 3 5-carbon RUBPs. And along the way, a phosphorylation occurs, and that's, again, energy from the light reactions that's invested over here. And RUBP is one of the substrates of carbon fixation along with carbon dioxide. But now we've accounted for all of our carbons. We start with these 15 over here, 15 carbons, and after the whole process is done, we again have 15 carbons. And if you can explain that, you're set up for an A and a 5 on the AP Bio exam. And that is now an appropriate time for me to say congratulations, because you've hung through one of the most difficult parts of this review. We're going to do cellular respiration next. You're a hero. Keep with it. You're going to get a 5 on the AP Bio exam. I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Cellular respiration, the big picture. What's the chemical equation for cellular respiration? Is it endergonic or exergonic? Where in eukaryotic cells does it occur? The equation C6H12O6, or glucose, plus 6 oxygens yields 6 carbon dioxides, 6 water molecules, plus energy in the form of ATP. It's an exergonic reaction. It releases energy and it creates disorder. It takes an organized molecule like glucose and creates much less organization over here. And it occurs in various phases. The first phase is glycolysis. That occurs in the cytoplasm. Then the link reaction brings the product of glycolysis into the mitochondrion. And in the matrix, we have the Krebs cycle. And then finally, the phase which produces the most ATP is oxidative phosphorylation. And that occurs based on enzymes and proteins along the mitochondrial membrane using the intermembrane space to create a chemiosmotic gradient. Briefly describe what happens in each phase of cellular respiration. Here we've got the whole process. Let's break it down. The process begins with glycolysis, so energy in glucose generates ATP and NADH. NADH, like NADPH that we met in photosynthesis, it's an electron carrier. And the end product is a 3-carbon molecule called pyruvic acid or pyruvate. 1 glucose, a 6-carbon molecule, becomes 2 pyruvates, which are each 3 carbon molecules. In the link reaction, what happens is that pyruvate enters into the mitochondrial matrix, and enzymes along the way convert it to a molecule called acetyl-CoA with 2 carbons. The carbon that's removed is released. That's 1/3 of the carbon that you exhale. And that conversion is powers the reduction of an NAD plus to NADH, and that's later going to be used in the electron transport chain. In the Krebs cycle over here, what enzymes do is they oxidize these 2 carbons in acetyl-CoA, and that powers the production of 3 NADHs, 1 FADH2, that's another mobile electron carrier like NADH, and it creates 1 ATP. That releases 2 carbon dioxides, and that's the other 2/3 of the carbon dioxide that you exhale. And note that for every glucose that enters cellular respiration, the Krebs cycle runs twice, as does the link reaction. And then finally, you have the electron transport chain, the ETC, and what that does is it takes these reduced products, these reduced mobile electron carriers, and it oxidizes them, and that creates electron flow that powers, through chemiosmosis, the production of ATP from ADP and phosphate. That's where most of the ATP during cellular respiration is produced. Cellular respiration, parts 2 and 3, glycolysis, the link reaction, and the Krebs cycle. What happens during glycolysis include phases, inputs, and outputs in your answer. I've got a great song about glycolysis that goes like, glycolysis, come on sugar for the breakdown. It occurs in the cytoplasm. It doesn't require oxygen, it's anaerobic, and it has 3 parts to it: investment, cleavage, and energy harvest. In terms of investment, what happens is that enzymes phosphorylate glucose. So glucose is a starting compound. Enzymes aren't shown here, but are implied by the arrows. Take a phosphate from ATP and plant it onto intermediate compounds. By the end of that process, you have fructose 1,6-bisphosphate. And that goes into the next phase where enzymes take fructose 1,6-bisphosphate loaded with energy and cleave it apart into 2 molecules of G3P. G3P is the same molecule that gets harvested from the Calvin cycle of photosynthesis. In the harvest phase, enzymes, again indicated by these arrows, do 2 things. They take G3P and they oxidize it. That means it's going to lose energy, lose electrons, and those electrons go to NAD+. It's a mobile electron carrier, which gets reduced to NADH, and that's later going to be used in the electron transport chain. In addition, there are other enzymes that take ADP and phosphate and phosphorylate it to ATP. They're using the chemical energy in G3P to bring that about. We're getting a gross energy yield of 4 ATPs. And that leads us to the next idea. What's the net yield of glycolysis? We get 2 ATPs. Why 2, even though there are 4 over here? It's because 2 were invested at the start of the process. So we net 2. And we also get 2 NADHs, 1 produced over here from this G3P, 1 produced over here. And we wind up with 2 molecules of pyruvate or pyruvic acid. That's still loaded with energy, and that's going to power the next phases of cellular respiration. What happens between glycolysis and the Krebs cycle? The answer is the link reaction. What we have ended glycolysis with is pyruvic acid, and that's transported across the inner and outer mitochondrial membranes. That's something that's represented by B in this diagram, into the mitochondrial matrix. As that happens, enzymes remove a CO2 from this pyruvic acid. You see this carboxyl group over here. It's basically a CO2. And that is 1/3 of the CO2 that you and any other animal that does cellular respiration, plants too for that matter, wind up releasing. Other enzymes oxidize the resulting 2-carbon molecule. So we had 3 carbons. Now we're at something with 2 carbons. It's actually called an acetyl group. And the oxidation of that acetyl group, oxidation is loss of electrons. Where do those electrons go? They go to NAD+, a mobile electron carrier. We just met it in glycolysis. And that NAD+ gets reduced to NADH, and that has electron energy that can get used in the electron transport chain. Finally, enzymes take that acetyl group and they attach it to a carrier molecule called coenzyme A, and the result is acetyl-CoA. That's the starting point for the Krebs cycle. Describe the Krebs cycle. The Krebs cycle, which I have a great song about, occurs in the mitochondrial matrix, and it's a cyclical series of reactions that generate NADH, FADH2, and ATP. It starts with enzymes, all of these are enzymes over here, enzymes taking this 2-carbon acetyl group from acetyl-CoA and transferring it from oxaloacetate, also known as oxalic acid, to citric acid, which is also known as citrate. The alternative names of the Krebs cycle are the citric acid cycle or the tricarboxylic acid cycle, TCA cycle, and that's for this carboxyl group over here, this carboxyl group over here, this carboxylic acid over here. So 3 carboxylic acids, TCA cycle. What happens next is that enzyme after enzyme oxidizes citric acid. And oxidation is loss of electrons. Those electrons get transferred to the mobile electron carriers NAD+, which becomes reduced to NADH. That happens over here, and over here. And there's also a reduction of FAD, another mobile electron carrier, to FADH2. Along the way, other enzymes over here power a substrate-level phosphorylation of ADP and phosphate into ATP. There's actually a complication involving GTP, but you don't need to worry about that for an AP Bio class. For each acetyl-CoA that enters the cycle, 1 ATP, 3 NADH, and 1 FADH2 are generated. And that's probably worth memorizing. And CO2 is released as a byproduct. There's one over here. That's the other 2/3 of the CO2 that you exhale. And oxaloacetate is the starting and ending compound. That's the Krebs cycle. Cellular respiration, part 4, the electron transport chain and oxidative phosphorylation. This is such a beautiful and important process. Let's go through it. In the previous phases of cellular respiration, glycolysis, the link reaction, Krebs, we've been creating NADH and FADH2. Those are mobile electron carriers. They've been accumulating in the mitochondrial matrix, and they diffuse over to the inner membrane where they're oxidized. Oxidation is loss of electrons. Those electrons now flow through an electron transport chain. That's this yellow arrow over here. And that's a series of membrane-embedded proteins that are in the mitochondrial inner membrane. So it's as if there's an electrical current that's flowing along the mitochondrial membrane through these various proteins. Notice that NADH comes in first. It has a little bit more energy in its electrons than FADH2, which drops its electrons off a little bit further on down the chain. Some of these electron transport proteins are proton pumps, and they pump protons from the matrix to the intermembrane space. Pumping is active transport that requires energy. Where's the energy from? It's from this flow of electrons. Notice that there's less protons over here and more over here. Again, active transport requires energy, and that creates an electrochemical gradient. There's more protons over here, fewer over here. There's more positive charges over here. There's fewer positive charges over here. It's also a pH gradient because the pH is much lower in the intermembrane space than it is in the matrix. All those protons are trapped. Oxygen acts as the final electron acceptor. And what it's doing is it's so electronegative that it's pulling electrons down this electron transport chain. And as it does, it absorbs electrons and protons that are available in the matrix, and that increases the gradient. This is why you need oxygen to do aerobic respiration, because it's the final electron acceptor in the electron transport chain. Now, all of these protons have been accumulated here. They can't diffuse through any part of the inner membrane except through one channel. That's called ATP synthase. It's the same that we discussed when we discussed photosynthesis. It's a channel and it's an enzyme. As protons diffuse through, so that's diffusion, it's facilitated diffusion, their kinetic energy is used to create ATP from ADP and phosphate. That's how the electron transport chain generates ATP. Cellular respiration can be used to generate heat instead of ATP. Explain. In newborn humans and other mammals and hibernating mammals, there are cells that are called brown fat cells. They're extremely dense with mitochondria. That's where the heat is generated. When body heat is needed, hormones induce a protein channel called thermogenin, or UCP. It's also called the uncoupling channel, to form in the inner mitochondrial membrane. So here it is over here. Now notice, in normal cellular respiration, protons that are trapped have to diffuse through ATP synthase, which creates ATP. But now there's an additional channel. So protons diffuse back to the matrix from the intermembrane space without passing through ATP synthase. But all of this activity in the electron transport chain here called the respiratory chain still has to happen. And think about the fact that when electrons move through a wire, that generates heat through resistance. Well, basically, you can think of the electron transport chain as a wire. The electrons that are moving through it generate heat, but in this case, they generate heat without generating ATP. And that's how cellular respiration can be used to generate heat instead of ATP. How is ATP generation in mitochondria and chloroplasts similar? In Unit 3, we've talked about these two great metabolic reactions, photosynthesis and the electron transport chain of cellular respiration. And there are deep similarities to the way that they work. And this kind of cross-topic thinking is essential to your success in the AP Bio exam. Both of these processes use an electron transport chain to pump protons to an enclosed space, creating a proton gradient. And in photosynthesis, here's the electron transport chain. We're pumping protons from the stroma to the thylakoid space. In cellular respiration, we're pumping protons from the matrix to the intermembrane space. And both use a subsequent process of chemiosmosis, the fusion of protons through an ATP synthase channel to generate ATP. And that's not a coincidence. As we'll see in Unit 7, where we'll talk about evolution, the similarities that are present between mitochondria and chloroplasts indicate that at some point in very ancient history, they had a common ancestor. ATP synthase evolved once, and then it became shared by the ancestors of chloroplasts and mitochondria. It's an incredible ride seeing biology and seeing evolution in process. Cellular respiration, part 5, anaerobic respiration and fermentation. Compare aerobic and anaerobic respiration. Aerobic respiration, oxygen is required. It involves essentially the whole shebang of respiration that we've talked about, glycolysis, plus the link reaction, plus the Krebs cycle, plus the electron transport chain, generate a lot of ATP, 32 ATPs approximately for every molecule of glucose that enters the cycle. Most of the ATP is generated over here in the mitochondria. There's a small amount that's generated through glycolysis. Anaerobic respiration occurs when oxygen is lacking or insufficient, or I should add, when the organism doesn't have the enzymes to do aerobic respiration. Glycolysis is really the key part of anaerobic respiration. So it involves glycolysis followed by fermentation, and it generates a total of 2 ATPs, and it occurs entirely in the cytoplasm. The mitochondria are not involved. What is fermentation and why does it occur? Fermentation is glycolysis that's followed by reactions that regenerate NAD+. So here we have one form of fermentation. It's called alcohol fermentation. Here we have lactic acid fermentation. But the key thing is that they produce NAD+. That's kind of the opposite of what glycolysis does. Why does fermentation happen? It's because if you're an aerobic organism, but you need to keep on moving and there's not enough oxygen available because you're working so hard, then you can continue to get 2 ATPs per every glucose. And that sure isn't as good as 32 or more that you get from aerobic respiration, but at that moment you just can't do it. Glycolysis, however, can only create those 2 ATPs if NAD+ is available. It's a substrate for one of the reactions of glycolysis. So 2 ATP are better than none, and that's why animals like us that do lactic acid fermentation perform it when oxygen is unavailable. Compare and contrast alcohol and lactic acid fermentation. Alcohol fermentation, really what we're talking about is ethanol fermentation, because there are other alcohol fermentations, occurs in yeast. When you're baking bread and you use yeast, you're doing an alcohol fermentation. Enzymes remove a carbon dioxide from pyruvic acid. Remember, that's the molecule that ends glycolysis. That produces acetaldehyde, and acetaldehyde is then by other enzymes reduced to ethanol. Well, reduction is gain of electrons. Something has to lose electrons, and NADH is oxidized to NAD+. That allows glycolysis to continue. Interesting and fun fact, this CO2 that makes up the bubbles in beer, which come about through an alcohol fermentation, that also makes up the bubbles that cause bread to be spongy and have its spongy form. Now, lactic acid fermentation occurs in muscle tissue under anaerobic conditions. It's a little bit simpler than alcohol fermentation. What happens is that pyruvate is reduced to lactic acid, and again, as that reduction occurs, something has to be oxidized, and NADH is oxidized to NAD+. That allows glycolysis to continue. There are all kinds of anaerobic sports like weightlifting or doing push-ups or things like that, and you're doing them at a level where muscle tissue can't really get enough oxygen. So during that time, they're doing lactic acid fermentation. You feel that lactic acid build up, and then eventually you have to stop exercising until you can recover aerobically. I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Topics 4.1 to 4.4, Part 1, Cell Signaling, the Big Picture. To help you study, I've put together a checklist that you can download at apbiosuccess.com/checklist. Cells are constantly communicating with one another. It's a basic feature of life. Cells are never really on their own. They're always in populations or they're in multicellular organisms. So there's direct cell-to-cell communication that you see here, where there's some kind of junction between two adjacent cells, and molecules can pass between those two cells, and that enables one cell to change the behavior of the other. There's also communication that happens through signals. So here you have a cell that's producing a molecule. It's secreting that molecule into the bloodstream or into the extracellular fluid, and that message is going to be picked up by a target cell. These signals are known as ligands, and there are basically two kinds. If the ligand, the signal, is traveling a long distance through the bloodstream from a gland to another part of the body, that's called a hormone. When cells are in relatively close proximity to one another, they produce local regulators, and that's for short distance cell-to-cell communication. Describe direct contact communication in plant and animal cells. Plant cells can be connected by plasmodesmata. These are bridges involving the cell wall and the membrane. These bridges allow signaling molecules, water, and other substances to diffuse from cell to cell. Animal cells have gap junctions. These are built of protein channels that connect adjacent cells. They allow a variety of substances, including signaling molecules, to move from the cytoplasm of one cell to the next. What are ligands? Ligands are signaling molecules. Many ligands are hormones, and we'll discuss some in depth in this review. What they do is they bind with receptors based on complementary shape. And here I've represented that very simply with a kind of circle and an arc. But because this is biology, you know that the shapes will be extraordinarily complex, like enzymes and substrates. Binding leads to a cellular response, and the mechanism by which that happens is going to be most of what we're going to talk about in this video. What is quorum sensing? This is a kind of cell communication that's seen in biofilm formation in bacteria, and if this seems out of context, these are films that can form, for example, on your teeth, leading to the buildup of plaque. What happens is that bacteria, so here's a single cell, be like one of these over here, what they do is they release signaling molecules, shown over here at number 2, and those bind to cytoplasmic receptors. So those are receptors that are actually inside the cell. They're not on the membrane surface. When the signal exceeds a certain intensity, so when there's a lot of cells around, that's a quorum. And those signals will be binding with the receptors, and that will activate genes. And that gene activation will lead to the expression of, for example, in this case, it looks like what these cells are doing is they're producing a biofilm, a polysaccharide that forms that biofilm. What's the takeaway? Twofold. The first is that all cells communicate, even bacteria, and the second is that you should brush your teeth so that you don't get a buildup of biofilm leading to plaque. Cell signaling involves 3 phases. List them. The 3 phases are reception of a ligand, that's what's happening over here. There's signal transduction, where the initial message gets changed into another kind of message that can go into the cytoplasm that often involves amplification of the signal. And finally, there's a cellular response. We're now going to expand on the material in the previous slide. What happens during the reception phase of cell signaling? The signal molecule, also called a ligand, as we've talked about before, binds with a receptor molecule. Here you can see a receptor that has a more realistic depiction, and you can see how it's embedded in the phospholipid bilayer of the cell membrane. That binding is based on complementary shape. What happens during the transduction and response phases of cell signaling? The receptor during the transduction phase interacts with membrane proteins to produce a second messenger. So something will be happening between here and other proteins in the membrane to bring about this second messenger. The second messenger with other relay molecules will bring that message to the cytoplasm, activating enzymes, or the nucleus, where we'll have the activation of genes. How is the mechanism of steroid, nonpolar hormones different from that of water-soluble hormones? The hormones we've talked about so far, the cell communication mechanisms we've talked about, have all involved water-soluble hormones. But what if the hormone is nonpolar, a steroid hormone like estrogen or testosterone? In that case, these hormones, which are nonpolar, can diffuse through the phospholipid bilayer. And once they're there, they can bind with a cytoplasmic receptor. That forms a receptor hormone complex, and that is capable of diffusing into the nucleus, where it acts as a transcription factor, something we'll talk about in AP Bio Unit 6. But all you need to know for now is that that can activate genes. So the DNA gets made into RNA. The RNA goes into the cytoplasm. It gets read by a ribosome and gets made into a protein. Water-soluble hormones, they're capable of binding with receptors, they interact with second messengers, and they bring about a cellular response. And in general, these responses are slower but more longer lasting. These responses are quicker. Topics 4.1 to 4.4 Part 2. Now that we've had an overview of cell communication, we're going to look in depth at an illustrative example, epinephrine and G protein coupled receptor systems. Before we delve into this, I want to let you know that I have a song that's a rap about cell communication, which covers much of this material.
Cellular communication works through phases 3.
The context for what we're about to review or learn is the fight or flight response, and that has effects throughout our bodies. One of the effects is the effect on the adrenal glands, which produce a hormone that's called epinephrine or adrenaline, and that acts upon the liver to get it to produce glucose that goes into the blood as part of the fight or flight response. Epinephrine is also known as adrenaline. It's a polar water-soluble hormone. You can see these hydroxyl groups over here and over here. Here's an amino group. This is not going to be able to diffuse into the cytoplasm. It's going to bind at the membrane. Note that epinephrine's effects are widespread but tissue specific. So epinephrine is going to get released from the adrenal glands into the bloodstream. It's going to go everywhere. It's going to touch every cell in your body, but only tissues with receptors are going to respond. That response will differ based on tissue types. So all of these are adaptations that are part of the fight or flight response. So over here we'll decrease digestion, because when you're trying to fight off some mortal threat, you don't need to be digesting at that moment. You want to increase your heart rate so that you can deliver more food and oxygen to your cells. Pupil dilation, more light, you're going to have better senses. Conversion of glycogen to glucose, that gives your cells, your muscle tissue, more energy to fight or flee. And bronchial dilation allows you to get more oxygen into your lungs so you can deliver more oxygen to the cells of the body. Epinephrine interacts with cells in the liver, and it induces changes that causes those liver cells to take stored glycogen, that's a polysaccharide, and to hydrolyze it into the monomers of polysaccharide, glucose. That glucose then diffuses into the bloodstream, and there it goes to the muscles of the body and to other organs as well, and that provides energy to fight or flee as part of the fight or flight response. The question for us is, how does epinephrine get liver cells to bring about this response? So we're now looking at the off state before epinephrine is released. And in this moment, the receptor is unbound. There's no epinephrine in the system. There's a nearby membrane protein that's called a G protein. A G protein is not a receptor, it's a membrane embedded protein, and it can oscillate between two states. Now it's off, it's inactive. Nearby the G protein is a membrane embedded enzyme, not a receptor, but an enzyme that's called adenylyl cyclase. That is actually the correct pronunciation. And it's also in the off state. And as a result, it's not activating the second messenger. What happens when epinephrine enters the system? First thing that happens is that epinephrine binds with a G protein coupled receptor. So here's epinephrine, and here it's binding with the receptor. This is a complicated protein, and we've talked about allosteric shifts in relationship to enzymes, where when something binds at an allosteric site, it can then change the active site. Well, the same mechanism is at work here. Epinephrine's binding over here, and that change kind of ripples through this protein, and it induces a change over here. Now, right at this moment, the nearby G protein is still dormant. It's still bound to GDP. GDP is a relative of ADP, and it's the low energy form. It can oscillate between this low energy form and a high energy form that we're going to see in a minute, and that happens when the G protein becomes activated. What's the effect on the G protein of epinephrine binding with the receptor? Well, the G protein is then able to interact with the receptor. We noted before that the receptor has changed on its cytoplasmic side, and that enables the G protein to interact with that part of the receptor, and that causes the G protein to discharge GDP, that's the low energy form, and to bind with GTP. That's the high energy form. And again, notice that this has 3 phosphates over here, just like ATP. This only has 2 phosphates over here, like ADP. The result is that the G protein now becomes activated. So what happens to the G protein once it's bound with GTP? It drifts in the membrane. It ultimately binds with adenylyl cyclase, this membrane embedded enzyme. That activates adenylyl cyclase, and adenylyl cyclase's substrate is ATP, and it converts it into a molecule called cyclic AMP, which is the second messenger in these G protein coupled receptor systems. Note that ATP is triply phosphorylated. This only has 1 phosphate, and the AMP stands for adenosine mono as in 1 phosphate. What have we done? We've taken our initial messenger and we've transduced it, creating the second messenger. So let's review what we've talked about so far. We've talked about reception. We have the ligand, which is epinephrine. It binds with the G protein coupled receptor. The receptor changes shape on its cytoplasmic side. It interacts with the G protein, causing it to discharge GDP, which is what it's bound to when it's dormant, and bind with GTP, which is what it binds with when it's active. The G protein then in turn can activate adenylyl cyclase, which takes its substrate, ATP, and converts it into cyclic AMP, the second messenger. What we're going to look at next is the cellular response. The second messenger, cyclic AMP, is going to activate a chain of relay molecules. These are called kinases or kinases. And this activation involves one kinase activating the next kinase, activating the next. I've only put 3 in this chain, but there can be many, many more, and that's called a phosphorylation cascade. How does that phosphorylation cascade work? The kinases are activated by phosphorylation, by gaining a phosphate. And once they're activated, what they do is they activate the next kinase in the chain. So here we have protein kinase 1 that acts upon protein kinase 2 by phosphorylating it. So now protein kinase 2 is active, phosphorylated. What does it do? It activates protein kinase 3 by phosphorylating it. I've only shown 3, but there can be many more in this chain, and we get this domino-like effect of one kinase activating the next and activating the next. Once we get to adenylyl cyclase, then each step involves multiple activations. Adenylyl cyclase will activate many cyclic AMPs. Each of these cyclic AMPs will start different phosphorylation chains, and the result is signal amplification. We had one epinephrine enter the system, but by the end, and I couldn't of course depict that here, but you'll have the activation of millions of enzymes to bring about a massive cellular response. In the case of liver cells and the way that they're acted upon by epinephrine, the response is activation of the terminal enzyme, which is glycogen phosphorylase. And what glycogen phosphorylase does is it converts glycogen, again a polysaccharide, into glucose, a monosaccharide. That glucose diffuses into the blood, giving you energy for the fight or flight response. We've seen how epinephrine, working through this G protein coupled receptor system, can mobilize this massive cellular response. This is incredibly quick, and it has to be because it's part of the fight or flight response. But after the threat has been escaped from or dealt with, the system also needs to be shut down, and that needs to happen quickly as well. To explain that, I'm going to show you this excerpt from my cell communication wrap. Enjoy.
Back to the ligand at the receptor. It only stays for a moment before it diffuses away. So when the thread is gone, the cascade gets shut down. With no adrenal secretion, the receptor's unbound. G protein drops the phosphate, now bound to GDP. It goes back to sleep, stops its activity. Bound to GDP, it no longer stimulates adenylyl cyclase, which no longer creates cyclic AMP, so the second message stops. Kinase phosphorylation quickly drops as other enzymes, protein phosphatases, clip off phosphates, turn off kinases. Glycogen phosphorylase stops hydrolyzing glycogen, so blood glucose normalizes. Liver cells return to their resting state. I love how G proteins let cells communicate.
I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Topic 4.5, feedback and homeostasis. What is homeostasis? What are feedback mechanisms, and how do they connect to homeostasis? Homeostasis is the tendency of a living system to maintain its internal conditions at a relatively constant, optimal level. Your body temperature, for example, stays around 37 degrees Celsius, 98.6 Fahrenheit, despite fluctuations in the external temperature. The changes in your body to maintain that temperature, that's a great example of homeostasis. Feedback is when the output of a system is also an input. So here we have some system, here's the thing that's coming out, and feedback is when it feeds back into the system. So the output is also an input. And feedback can do two things. One is connected to homeostasis. It can allow organisms to maintain homeostasis as they respond to internal and external changes, just as I told you with temperature, or it can accelerate internal changes and drive a process towards a conclusion. This is generally connected with what's called negative feedback, and this is connected with what's called positive feedback. Let's talk about set points. What is a set point? How are set points used in negative feedback? First, a word about the method here. We're going to talk about this in relationship to your home heating and cooling system. It's much easier to understand this before we look at the biological examples. So a set point is where you set the thermostat. It's the value around which a homeostatic process fluctuates. So above, The set point for the thermostat is 68 degrees Fahrenheit. In negative feedback, the output of the system feeds back to the system in a way that decreases the system's output. So here we have the set point. Here's a measurement of the temperature. The set point is above the temperature. That'll send a signal to turn on the furnace. That'll generate heat. Heat feeds back to the system. There's a thermostat over here with a thermometer. That will increase the temperature, and then when we get to 68 degrees Fahrenheit, the set point, the system will turn off. Negative feedback promotes homeostasis, returning a system to its set point. How can antagonistic negative feedback loops be paired to promote homeostasis? So, same goal. Keep the temperature at 68 degrees. Well, here we have a negative feedback system that responds when conditions are above the set point. So, temperature 70, the set point is 68. What will you do? You'll have a signal that turns on an air conditioner. That will put out cool, it's a real word, and that will feed back to the system, and eventually it'll get the system to turn itself off. There's been cooling, and we've therefore lowered the temperature. But we still have a heating system, and if the temperature goes below the set point, then that system would turn on, it would release heat, that would feed back to the system, and that would wind up turning the system off. So we have paired antagonistic systems, one for cooling, one for heating. Now we're going to look at some biology. Let's talk about how feedback works in blood sugar homeostasis. It's important to your body to maintain your blood sugar levels, your blood glucose levels. The main hormone that controls that is insulin, and it's a negative feedback system. In response to high blood glucose levels after eating a sugary or a starchy meal, your pancreas will release the hormone insulin, just represented here as a triangle. In the liver, insulin will bind at a receptor over here, and that will unleash a signaling cascade, and that talks to a glucose transporter. That's a channel that allows for facilitated diffusion. If glucose is in higher concentration outside than inside the cell, it'll diffuse into liver cells, and the glucose will get converted into glycogen, which is a storage polysaccharide, or it'll get converted into fat. Blood glucose levels down, homeostasis restored. But as with our home heating and cooling system, insulin is paired with another hormone. So let's explain how insulin and glucagon maintain blood glucose homeostasis. Our blood glucose has a set point about 90 milligrams per deciliter. Above the set point, the pancreas will release insulin. Eat a big meal, insulin gets released, and your liver, fat, and muscle cells, which aren't shown, will absorb glucose and store it away as glycogen. Below the set point, that's when you've gone a little while without eating, what will happen is that the pancreas will release a second hormone called glucagon, and that will induce the liver to convert its stores of glycogen into glucose. Glucose goes into the blood, homeostasis is restored. An important AP Bio skill is explaining what happens when systems get disrupted. And here we're going to explain how blood glucose homeostasis breaks down in type 2 diabetes. That's also called adult onset diabetes, but increasingly it's happening in teens and even children due to the obesity epidemic in the United States. We're going to look at normal metabolism first, which we just discussed. Here we see insulin in response to a high blood glucose level is binding with the insulin receptor. There's a signaling cascade that causes the glucose channel to open. There's glucose absorption into the cells. Blood glucose level falls. Homeostasis restored. In people with type 2 diabetes, what happens is that the cells become insulin resistant. There's glucose in the blood. There's insulin released from the pancreas, but the binding of insulin does not lead to the signaling cascade. And because of that, the glucose channel remains closed. Because of that, blood glucose level stays high, and that damages organs and tissues. Breakdown of homeostasis leading to type 2 diabetes. We just discussed adult onset type 2 diabetes. How does that compare with type 1 diabetes? Type 1 is also known as juvenile diabetes. It's an autoimmune disorder, And basically what happens is that cells of your immune system attack the pancreas cells that produce insulin. Therefore, those cells can no longer secrete insulin. And therefore, even in response to a high blood glucose level, insulin is not secreted. That can only be treated by insulin injections. Type 2 diabetes, we've just discussed, also called adult onset, and that involves insulin resistance where the receptor is become insensitive to the insulin signal. All of the biological feedback loops that we've talked about so far relating to blood sugar have involved negative feedback loops. What about positive feedback loops? How does positive feedback work? Explain how positive feedback works during childbirth. So in positive feedback, the output of a system feeds back into the system, increasing the system's activity and output. It doesn't involve quieting that leads to homeostasis. It involves acceleration. It drives a biological process such as childbirth to a conclusion, after which the system shuts down. In childbirth, the growth of a baby activates uterine stretch receptors. The uterus, among other things, is a big muscle. When that muscle is stretched out, it sends messages to the brain. The brain releases oxytocin, a hormone. That oxytocin then circulates in the blood. When it's picked up by receptors in uterine cells, that leads to more contraction. That increased contraction leads to more oxytocin release, and ultimately that continues until the baby is born. Explain how feedback leads to fruit ripening. If you were to pause the video and take a look at this diagram, I'll bet you that you could figure it out. This is a positive feedback system. Ripening in fruit leads to the release of a hormone that is a gas. It's called ethylene. Ethylene receptors in nearby fruit pick up the ethylene, and that induces additional ripening and more ethylene production. That increased concentration of ethylene accelerates the ripening process in all the fruit, leading to more ethylene. So eventually all the fruit ripens. In fruit shipping, you can prevent that by pumping carbon dioxide into the storage container where the fruit are, and that suppresses the ethylene, and that's how you can ship fruit long distances. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or a 5, you need an easier way to study, and that's why we created learn-biology.com. It has quizzes, it has flashcards, it has interactive tutorials about every topic in the AP Bio curriculum. It has a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence that you'll need to ace your biology course and to crush it on the AP Bio exam. So here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. You won't believe how much you'll learn. Topic 4.6, the cell cycle. On a big picture level, what does mitosis do? List 3 of its key functions in living things. Note for this question that mitosis, as it often is synonymous with eukaryotic cell division. Mitosis duplicates the chromosomes of a eukaryotic cell, transmitting that cell's entire genome to its daughter cell. So here you have the parent cell. It's got 2 chromosomes. Here those chromosomes have been duplicated. Now they're being pulled apart. And now you have daughter cells, each with 2 chromosomes. And each one is going to be an exact clone of its parent cell. In a multicellular organism like you and me, mitosis is how an organism grows and repairs itself. Remember that you started life, as all multicellular beings do, as a single cell. In a unicellular eukaryote, like a paramecium or an amoeba, mitosis is how reproduction occurs. Describe what happens during the cell cycle. The cell cycle, as you can see in this diagram, can be divided into 2 main phases. So the outside orange part of the circle, that's interphase. This yellow part over here, that's mitosis or M phase, represented by the letter M. During interphase, you can subdivide 3 basic phases, but the cell doesn't appear to be dividing during interphase. The first is G1, and during G1, the cell increases in size. G1 stands for growth phase 1. During S, which stands for synthesis, you have DNA replication or chromosome duplication. And during G2, growth phase 2, you have the growth of the structures that are required for cell division. During M phase, you have mitosis, separation of the chromosomes followed by cytokinesis. So at the end, you have two daughter cells that are clones of the parent cell. Describe the phases of mitosis. We begin with interphase, during which the cell grows and replicates its DNA. During that time, it doesn't look as if it's dividing, but DNA replication, for example, has occurred. During prophase, chromosomes, which are spread out as what's called chromatin during interphase, they condense into these X-like structures. The nuclear membrane disintegrates, and a spindle apparatus, which is these fibers over here, you can see the entire apparatus in the next phase, start to grow from each centrosome. During metaphase, the spindle fibers grab onto the chromosomes and they pull and push them to the cell equator. If you can sort of imagine a line running down from top to bottom, that would be the cell equator. I talked about this X-like formation that the chromosomes are in. That's because each chromosome is doubled and consists of two clones called sister chromatids. During anaphase, the spindle pulls the sister chromatids apart and starts dragging them to the opposite ends of the cell. At the same time, there are what are called non-kinetochore microtubules. A kinetochore is like a handle on the chromosomes that these spindle fibers use to pull the chromosomes apart. But there are other fibers that basically push on one another, and that causes the cell to elongate. During telophase, a new nuclear membrane starts to grow around each set of chromosomes. The chromosomes spread out into their interphase formation, so you can't really see them anymore. And a nucleolus reappears in each cell. It disappeared during interphase. The nucleolus makes ribosomes, and ribosome production shuts down during most of mitosis. Finally, during cytokinesis, the cell splits apart into two daughter cells. I have a great song about mitosis, and I'll put the link below.
Mitosis, chromosomal ride in a pro-meta and a telophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew.
Explain the importance of the G0 phase of the cell cycle. Basic idea here is that not all cells go through the entire cell cycle. So if you have highly specialized cells like a nerve cell or a muscle cell, they'll leave the cell cycle and they'll enter into G0, after which they won't divide. Certain stimuli, however, can induce cells in G0 to re-enter the cell cycle, but for the most part, the nerve cells that you have are the nerve cells that you're going to have, which is why a traumatic spinal cord injury is something that you can't recover from. Topic 4.7, regulation of the cell cycle. I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. Describe the role that checkpoints play in regulating the cell cycle. Cell cycle checkpoints are moments when the cell checks its internal conditions and decides whether to progress to the next phase of the cell cycle. If certain molecules are in the right concentration, then the cell continues through the cell cycle, and if not, the cell moves into G0 or might initiate what's called apoptosis. We'll explain that in a moment, which is called programmed cell death. The primary checkpoints to know about for AP Bio are the G1 checkpoint over here, the G2 checkpoint, and the M checkpoint. What is apoptosis? I've talked about that several times. Note that the second P is silent. Apoptosis is programmed cell death. It's part of a signaling cascade that involves the mitochondria and the nucleus. It's highly regulated, which is very different from cell death that results from traumatic cell injury. Cells are broken down into cytoplasmic fragments that are called blebs. You can see some of those over here and over here. And blebs are consumed by cells of the immune system, preventing cellular debris and enzymes from damaging nearby cells. What are cyclins and cyclin-dependent kinases? Cyclins and cyclin-dependent kinases are important internal regulators of the cell cycle. The cell cycle is regulated by outside signals, but also by internal conditions. Cyclins are molecules whose concentration rises and falls throughout the cell cycle. So like, for example, you can see that cyclin E rises right before the S phase. Cyclin A rises right before G2. The cell has mechanisms to read the level of these cyclin concentrations. Kinases, which we discussed previously in the context of cell communication, those are molecules that activate other molecules, often by phosphorylating them. They're not shown. And cyclin-dependent kinases, or CDKs, are kinases that respond to rising and falling levels of cyclin levels. Now we're going to put that all together and see what some of the mechanisms are that regulate cell division. Explain how the interactions between cyclins and cyclin-dependent kinases control the cell cycle. So CDKs, here they are over here, are present at a constant level throughout the cell cycle. By contrast, the level of cyclins that we saw in the previous slide rise and fall. When cyclin levels are high, the cyclins bind with CDKs to form a complex called maturation promoting factor, but a good way to remember that is just think of it as mitosis promoting factor. Because once you have MPF, that allows the cell to pass through the G2 checkpoint and actually divide. During M phase, however, the cyclin is broken down, and that allows the process to repeat in each daughter cell when it grows to the appropriate size and gets ready for cell division. We talked about the cell cycle. Now let's talk about dysregulation of the cell cycle. What's the connection between cell division and cancer? What are the two types of genetic mutations that are connected to cancer? Cancer is a disease of unregulated cell division. As opposed to cells staying in place doing what they're supposed to do, they become rogue players pursuing their own destiny at the cost of the organism. Mutations in genes that are called proto-oncogenes increase cell division by creating too many growth factors. Growth factors are things that stimulate cell division within a cell itself or within other cells. And there are other kinds of genes that are called tumor suppressor genes, and what they do is they remove cell division inhibitors, the kind of checkpoints that we've seen in previous slides. So in normal cells you have the brakes, those are tumor suppressors, and you have the accelerator, those are growth factors, but they act at appropriate times. When cells become cancerous, it can be for one of two or actually both reasons. You can have mutated tumor suppressors that can't prevent cell division even when cell division shouldn't be happening. And you have growth factors that promote cell division at unneeded moments. Describe how a mutation in the ROS proto-oncogene can induce a non-cancerous cell to become cancerous. What we're going to do now is really cool because we're going to connect what we've learned about the cell cycle and its control to what we learned about previously relating to cell communication. So ROS is a G protein over here, and as a proto-oncogene, it only becomes active when an outside growth signal binds with ROS's coupled receptors. So here we have binding that activates ROS, it picks up GTP, that sets off a phosphorylation cascade, and that leads to cell division. So this is in a healthy, normal system. ROS only gets activated when there's a ligand. When ROS becomes cancerous, when it mutates from a proto-oncogene to an oncogene, then it becomes constitutively active. And constitutively means it's part of its nature to be active. Normal ROS is only active, it only binds GTP when the receptor binds a ligand. But this ROS, which is an oncogene, is constitutively active. It's binding GTP even in the absence of a growth signal, and because it's always active, the growth factor over here is overproduced, and that results in too much cell division. This is connected with about 30% of human cancers. Describe how a mutation in a tumor suppressor gene such as P53 can contribute to the development of cancer. P53 is a tumor suppressor gene. When cells experience DNA damage, a signaling cascade activates P53. So here we have DNA damage, it's detected, there's a signaling cascade, and now P53 is activated. If the damage can be repaired, then what p53 will do is it'll halt the cell cycle while DNA repair enzymes fix the damage. So we're going to fix up the DNA. That's what's happening over here. If the damage is too great, then p53 will initiate a whole signaling cascade that'll cause the cell to initiate apoptosis. So either we have repaired DNA that occurs while the cell cycle is halted, or the cell will self-destruct. Cancer's been prevented. If mutations lead p53 to become non-functional, then the cell will continue to divide even with damaged DNA. So here we have DNA damage. It's been detected, but p53 can't do anything about it. Therefore, there's no stop signal. The cell will continue to divide, and that'll increase the chance of the cell acquiring further mutations that can lead it to become cancerous. Again, dysregulation of a signaling cascade, dysregulation of a cell cycle repair mechanism leading to cancer. Your success in AP Biology starts here. Are you struggling with AP Bio? With learn-biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to learn-biology.com to find out how you can master your biology course and crush the AP Bio exam. Meiosis, the big picture. What is meiosis? Why is it important? Meiosis is how sexually reproducing eukaryotes, that includes animals, plants, fungi, protists, transmit genes from one generation to the next. It creates variation between parents and their offspring, and it creates variation among the offspring. Describe the life cycle of sexually reproducing eukaryotes. In eukaryotes, adults have specialized tissues, testes and ovaries, for creating gametes. Gametes are sperm and egg cells, and they do that through this process that we'll be exploring called meiosis. The sperm fertilizes the egg, and that produces a zygote, or a fertilized egg. That zygote then divides and develops, the tissues differentiate to produce an adult organism. In relation to meiosis, compare haploid and diploid cells. These are super important terms for understanding meiosis. Parents have 2 sets of chromosomes in all of their body cells, with the exception of their gametes. Those chromosomes are paired. So like, for example, here's chromosome 1. There are 2 of them. Here's chromosome 2. There are 2 of them. One was inherited from one parent, one was inherited from the other. Those pairs are said to be homologous, and that's a term that we'll explain in the next slide. When parents pass on their chromosomes to the next generation, they can't pass all of them on. If they did, then the chromosome number would double in every generation. So what happens in meiosis is a halving of the number of chromosomes. And the half number of chromosomes kind of rhymes with the word half, or it begins with the same prefix. It's called haploid. So notice that there are 4 chromosomes down here. There are only 2 over here. What's the difference? The difference is what happened during meiosis, which is division of cells that involves reduction. Reduction division. Define homologous chromosomes. Homologous chromosomes are the matching chromosomes that you inherited from your parents. So, for example, here's chromosome 3. One of them came from your mom, one of them came from your dad. Here's chromosome 4. It's the same thing. Here's chromosome 5, all the way on down the line. They are not identical. How could they be? Your mom and dad aren't identical, so the chromosomes that they passed on to you wouldn't be identical. They do have the same genes in the same order. But the alleles, the specific code that is in the location where those genes are found, that might be different. Let's use the analogy of a gene as a recipe. Well, the one that you inherited from your mom, if that were a recipe for tomato sauce. Maybe that one has a lot more garlic, and the one that you inherited from your dad, that might have a lot more basil in it. Well, let's now think more biologically. If C refers to a specific protein, then the DNA that's coding for a specific sequence of amino acids might be slightly different in what you inherit from your mom and your dad. And that might even be to the extent where the amino acid sequence of that protein differs. So the genes are the same, but the alleles might be different. That's what homologous means. More essential meiosis genetics vocabulary: compare and contrast germ cells, gametes, and somatic cells. Germ cells are the diploid cells that are found in the testes and the ovaries that undergo meiosis. They produce gametes, and after meiosis we have sperm and egg cells that are haploid. In a human being, the diploid number is 46. That's 23 pairs of chromosomes. In the haploid gametes, the chromosomes aren't paired anymore, so there are just 23 chromosomes in the sperm, 23 in the egg. Now the sperm goes ahead and fertilizes the egg, and that egg or zygote will divide, develop, the cells will differentiate, and what you'll wind up with are somatic cells. Those are the cells of the body, the diploid cells that make up the body tissues. Somatic cells are diploid, germ cells are diploid, gametes are haploid. What happens during meiosis? Meiosis is reduction division. Why reduction division? It's cell division that reduces chromosome number. Cells go from diploid with 2 sets of chromosomes to haploid with 1 set of chromosomes. The first step is DNA replication, which we see over here at number 1. It creates doubled chromosomes consisting of 2 sister chromatids. What's going on? Even though meiosis is reduction division, it starts in exactly the same way that mitosis starts, with a round of DNA or chromosome replication. So if you look at this cell over here, there are 4 chromosomes. If you look over here, there are still 4 chromosomes, but each one is doubled, consisting of 2 sister chromatids. In step 2, we have meiosis 1. And what meiosis 1 does is it separates the homologous pairs. So this chromosome is homologous to this one, this one, this one, and they're going to be separated. So now each of the resulting gametes only has 1 member of each homologous pair. That means it's now a haploid cell. But each chromosome is still doubled, and that's why we have meiosis 2. What meiosis 2 does is it separates the sister chromatids. The result is 4 unique haploid gametes. Compare mitosis and meiosis. Mitosis consists of 1 round of cell division that separates the sister chromatids. The cells begin as diploid and they end as diploid. The daughter cells over here are clones of the parent cell. It's used for growth and repair. Meiosis consists of 2 cell divisions. Meiosis 1 over here separates homologous pairs. So here we have the doubled chromosomes, homologous pairs. They get separated in meiosis 1. Meiosis 2 separates sister chromatids. We go from diploid to haploid. It's used to create gametes for reproduction. It introduces variation. The daughter cells are unique. How meiosis creates variation. I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. What are the 2 ways that meiosis generates diversity? They're shown here. The first is independent assortment. The second is crossing over and genetic recombination. We'll explain both of those now. Explain what independent assortment is and how it generates genetic diversity. Note that the phases of mitosis and meiosis have the same names, the same designations. But because there are 2 cell divisions in meiosis, we have to give them a kind of suffix. So in meiosis, there's interphase 1 followed by prophase 1, metaphase 1, etc. Then there's a cytokinesis interphase 2, followed by a prophase 2, metaphase 2, anaphase 2, etc. The reason why that's important is because the events that we're going to talk about happen in between prophase 1 and metaphase 1. That's where independent assortment really takes place. What happens is that during prophase 1, homologous pairs pair up. And if you think about that, it's quite poignant and extraordinary. In the adult organism who's undergoing meiosis, the chromosomal parents wind up finding each other. And what I mean by that is that in the germ cells of an organism doing meiosis, the mother and the father's chromosome number 1 will find one another, and I'm not kidding, they actually embrace. And chromosome 2 does that, chromosome 3 does that. All those chromosomes find one another and embrace one another. During metaphase 1, they're pulled by spindle fibers, just as happens in mitosis, to the cell equator. Right? So here we have that. But the way that each pair gets dragged to the middle is independent of every other pair. So in this very simplified system, it's possible that the paternal chromosomes might be on the left side and the maternal ones might be on the right side. It's equally possible that you could have this arrangement versus this arrangement, whereas you have paternal chromosome 1 on the left, maternal chromosome 1 on the right, whereas maternal chromosome 2 is on the left and paternal chromosome 2 is on the right. It's as random as flipping a coin, and that randomness is essential. What happens is that with 2 homologous pairs, 4 different chromosome arrangements are possible. That's 2 squared in the gametes. In other words, what we're going to do now during anaphase is we're going to pull these homologous pairs apart. So one possible arrangement is like this, and then in the gamete we have paternal chromosome 1, paternal chromosome 2, and in this gamete we have maternal chromosome 1, maternal chromosome 2. And if the chromosomes are organized like this, then this gamete can have paternal chromosome 1, paternal chromosome 2, and this gamete can have maternal chromosome 1 and paternal chromosome 2. Now, you can play around with this. You can make cut out little pieces, or you can label coins M1, P1, M2, P2, and you can try different combinations, but you won't get more than 4 in a system that has 4 chromosomes as its diploid number. With 3 homologous pairs, then the math takes you to 2 cubed. That's 8 possible arrangements, and with 23 pairs, like we have in Homo sapiens, you have 2 to the 23rd possible arrangements. That's 8,388,608 possible combinations. That's the chance that any 2 sperm cells or any 2 egg cells would have exactly the same array of maternal and paternal chromosomes. So what's the chance that you and a sibling would have the same chromosomal inheritance? In other words, you'd inherit the same array of chromosomes in your dad's sperm and the same array in your mom's egg. Well, those are independent events. So the same egg, it's 1 over 2 to the 23rd. The same sperm, it's 1 over 2 to the 23rd. You multiply those 2 together, and that's 1 in 70 trillion. You ever wonder why you're different from your siblings? This is only one of the reasons why. That's why meiosis is so phenomenal. So this is independent assortment. What every chromosome does is independent of every other chromosome pair, and that creates tremendous diversity in the offspring. Independent assortment is a phenomenal engine for creating diversity, but there's yet another one in meiosis. That's crossing over. What is crossing over, and how does it create variation? When those homologous pairs pair up during prophase 1, they don't only embrace, they embrace in such an intense way that they actually exchange parts. So this embrace is called synapsis, and at a point called a chiasma, segments of DNA will move from one homolog to the other. The result is that you start like this. This array of 4 sister chromatids is called a tetrad. 4, tetrad. And so this is what it's like before crossing over. And then after crossing over, it's like this. Well, you'll notice this isn't really a maternal chromosome anymore. It's a maternal chromosome with a paternal piece. And the same thing for this one. So crossing over creates what's called recombinant chromosomes. And these have unique and novel sequences of DNA. How does sexual reproduction create diversity? One engine for diversity is independent assortment and how it randomly arrays different combinations of chromosomes in the gametes. The other engine is crossing over and genetic recombination, which creates uniquely recombinant chromosomes. And then finally, there's fertilization, where the sperm and egg from different individuals combine in a fertilized egg. And that is the third generator. And that's why sexual reproduction creates diversity. It explains you, it explains me. Biology, is it amazing or what? Meiosis, the whole shebang. Let's walk through the entire process. We begin with interphase, and the thing to remember about interphase of meiosis is that it does exactly what interphase of mitosis does. It replicates the chromosomes, duplicates the DNA. That's why when we get to prophase 1, each chromosome consists of 2 sister chromatids. What else happens during prophase 1? The homologous pairs pair up. The maternal and paternal chromosomes find one another and they embrace. They do this thing called synapsis and crossing over, where they actually exchange pieces of DNA. So that's happening during prophase. During metaphase, the spindle fibers pull these homologous pairs to the center of the cell. And remember that each pair is pulled independently from every other. That's the source of one of the main sources of variation in meiosis, independent assortment. The way that these maternal and paternal chromosomes get arrayed is completely independent. It's 2 to the number of pairs if you want to mathematically calculate the number of chromosomal arrangements in the gametes. During anaphase 1, the homologous pairs are pulled apart. During telophase 1, a new nucleus forms. There's a cytokinesis 1 and an interphase 2 that's not shown in this diagram. That moves us to prophase 2, where the chromosomes once again condense. Note that whereas there were 4 chromosomes in prophase 1, there's only 2 in this cell in prophase 2. We've gone from diploid over here to haploid over here. That's the main transition, or one of the main transitions that occurs in meiosis 1. During meiosis 2, during metaphase, the doubled chromosomes get pulled to the cell equator and then they get pulled apart during anaphase 2. Then there's a telophase where a new nuclear membrane forms, and then there's another cytokinesis. The result, 4 haploid gametes, each consisting of single chromosomes. We've gone from diploid to haploid, from doubled chromosomes to single chromosomes, and each of these haploid gametes is unique. It's genetically unique, and that's what happens during meiosis. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or a 5, you need an easier way to study, and that's why we created learn-biology.com. It has quizzes, it has flashcards, it has interactive tutorials about every topic in the AP Bio curriculum. It has a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence that you'll need to ace your biology course and to crush it on the AP Bio exam. So here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. You won't believe how much you'll learn. Topic 5.6, Part 1, Chromosomal Inheritance, Sex Determination. How is sex determined in mammals? We've seen this image before. It's a karyotype. It shows all of the homologous pairs paired up. Chromosomes 1 through 22 are called autosomes. They're paired homologous pairs. They're the same in chromosomal males and chromosomal females. But the last pair are called the sex chromosomes. Females have 2 X chromosomes. Males have an X chromosome and a Y chromosome. Unlike the autosomes, chromosome pairs 1 through 22 in humans, the X and the Y are quite different from one another. For one thing, unlike those homologous pairs, they don't cross over and swap pieces of DNA. The X is a normal chromosome. It has a variety of alleles related to various non-sex relating functions. That includes immune function, vision, blood clotting, and so on. The Y has a region that's called SRY. It's indicated by this yellow bar over here. And that initiates the development of the testes during early embryonic development, and later on, production of testosterone, which winds up differentiating the body into its male form. During fertilization, it's the sperm that determines the chromosomal sex of the zygote, which becomes the embryo, which becomes the baby, which becomes the person. The males, which have their 22 autosomes and then an X and a Y chromosome, during meiosis will pass on either an X chromosome or a Y chromosome, because even though they're not truly homologous, they will get separated, just like how the other homologous pairs get separated. The egg has 2 X chromosomes, so every single egg cell is going to have the X chromosome. If the egg is fertilized by a X-carrying sperm, then the zygote will have 2 X chromosomes and it'll develop into a female. If the sperm that's carrying the Y chromosome does the fertilization, then the resulting zygote will be XY and it'll develop into a male. That's chromosomal sex determination in all of the mammals. The fact that half of the eggs will be fertilized by an X-carrying sperm and half will be fertilized by a Y-carrying sperm explains the fact that among the births in any mammal population, the initial ratio of males to females will be 50-50. Birds also have a chromosomal system of sex determination, but it's different. It's kind of a flipped version of the one in mammals. In birds, it's the egg that determines the chromosomal sex of the offspring. That's because the females have what we call a Z chromosome and a W chromosome, and during meiosis, half of the eggs will wind up being Z and half will be W. The females will pass on either that Z or W chromosome. The males have 2 Z chromosomes, so when they form their sperm, all of the sperm carry Z. Fertilization of a Z-carrying egg will lead to a male who's ZZ. Fertilization of a W-carrying egg will lead to a female who is ZW. That's chromosomal sex determination in birds. As in mammals, because half of the eggs will be Z eggs and half the eggs will be W eggs, the result is that among the initial births that happen in any bird population, half of those birds will be male and half of those birds will be female. Among certain reptiles, sex is determined by the temperature at which the embryos develop. Some reptiles lay their eggs in a nest that's dug in the sand. And as you might imagine, it's warmer on the top, closer to the sun, and cooler down below. These animals develop based on a pivot point that's represented by TPIV over here. So in sea turtles, the eggs that develop above the pivot point become female. Here's the proportion of males over here. So if you're above the pivot point, if you're in the warmer area, you'll develop into a female. If you're in the cooler area over here, below the pivot point, you'll develop into a male. And then at the pivot point, it's pretty much random, 50-50. In another kind of reptile called a tuatara, it's the opposite. So basically, if you develop above the pivot point, then you have a higher chance of being a male. If you're below the pivot point, then the proportion of males goes down. You have a higher chance of being a female. And in crocodiles, there are two pivot points, a low temperature one and a high temperature one. In the coolest and the warmest temperatures, the eggs develop into females. In other words, the proportion male, way down. But at intermediate temperatures over here, the eggs develop into males. How is sex determined in ants, bees, and wasps? This is completely mind-blowing. It's a system that's called haplodiploid sex determination, or haplodiploidy. The males are haploid. They develop from unfertilized eggs. So here's a male here, haploid. The females, which include the queen and all of the workers, they're all diploid, and they develop from fertilized eggs. So the mother in a bee colony, just to use that example, is the queen. She undergoes normal meiosis when she creates her eggs. But the father is a haploid male, also called a drone, and he can't really do meiosis because he's haploid, he's not diploid. So essentially he passes on 100% of his chromosomes in the sperm that he creates. The consequence is that all the bees in a hive, with the exception of the drones, all of the worker bees are sisters, but they're more closely related to one another than any two mammal sisters are. Just think about it. Mammal sisters inherit half their mom's genes, half of their dad's genes, so they're 50% related to one another, whereas these sisters inherit half of their mom's genes and 100% of their dad's genes, so they're 75% related to one another. They're more closely related to one another than they would be to their own offspring. So that is thought to be an explanation of why the workers cooperate with one another to help the queen create more offspring, to keep the hive going, to find food, all of these incredible foraging behaviors that you find in ants, wasps, and other social insects. But it's not a complete explanation because, like, for example, the termites are also social, but they don't have this haplodiploid system. So biology, amazing. This is as much as you need to know about haplodiploidy for the AP Bio exam. Topic 5.6, Part 2, Chromosomal Inheritance, Nondisjunction and Chromosomal Variation. What is nondisjunction? What are its consequences? Nondisjunction is kind of a cool word. So a junction is where things come together. A disjunction would be things coming apart, and nondisjunction means things failing to separate. It's when the homologous pairs or the sister chromatids don't separate during meiosis. There's a couple of variations. In meiosis 1, the homologs don't separate, and as a result, so you see that over here, this blue chromosome over here, these homologs didn't separate, they stayed together. So the result is that in meiosis 2, we're going to have 3 chromatids on this side, 3 over here, 1 over here, 1 over here. So 50% of the gametes are N haploid plus 1 extra, and 50% are N minus 1. So N, the haploid number, missing a chromosome. If nondisjunction occurs during meiosis 2, it involves the sister chromatids not separating. In meiosis 2, the sister chromatids don't pull apart. So the result is that 25% of the gametes are N plus 1, 25% are N minus 1, in other words, the haploid number but missing 1, and then 50% of the gametes will be normal. So that's what nondisjunction is and how it can happen in meiosis 1 or meiosis 2. We saw in the last slide how nondisjunction during meiosis 1 or meiosis 2 can result in gametes that have an abnormal number of chromosomes. If the eggs are N+1, they have the haploid number plus one more. Then the zygote will have an extra chromosome. Then what we have is a trisomy. And what that means, tri is 3. Instead of a homologous pair with 2, we have 3. The most famous example of that is Down syndrome, which is a trisomy of the 21st chromosome, which has various developmental consequences and developmental delays. If the eggs are N-1, like over here or over here, and the sperm, again, are carrying a normal number of chromosomes, so that's not necessarily the case. You can have nondisjunction that occurs during the formation of sperm as well, but then the zygotes will have a missing chromosome. So they'll have all of the homologous pairs, but one will be short. And the result of that is a monosomy, and most of those aren't really survivable except in the case of the sex chromosomes. So one to know is called Turner syndrome, where instead of females having 2 X chromosomes, they have 1. And there's a significant amount of variation that could happen in the sex chromosomes. There can be men who are born with an extra X chromosome. There can be men who are born with an X and 2 Y chromosomes. So those are all chromosomal variations that come about through nondisjunction followed by fertilization. I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Topic 5.3, Mendelian genetics. Genes are the basic unit of heredity. They are what gets passed from parents to offspring. They determine traits or characteristics. You can also think of genes from a molecular genetics perspective, and you can think of them as a sequence of DNA nucleotides that code for RNA and ultimately code for protein, and those proteins ultimately determine the characteristics of the organism. Describe Mendel's principle of segregation. In your answer, cover the difference between the terms homozygous and heterozygous. Just a historical note, Gregor Mendel is considered the father or grandfather of genetics. In the 1800s, he figured out many of these basic principles that we're talking about right now. Every individual has 2 copies of each gene. Those copies are located on the chromosomes that are organized into homologous pairs. The alleles are alternative versions of those genes that might have different DNA sequences that will produce proteins that have different amino acid sequences. Homozygous means that the 2 alleles are identical. For example, in this parent over here, both of the alleles are designated with a capital letter A. They're the same. This person is a homozygote for this particular gene. In a heterozygote, the alleles can be different. In this individual over here, one of the alleles is capital A, and one of the alleles is small a. The principle of segregation shows what's happening here. When individuals create gametes, their sex cells, they pass on only one of their 2 alleles. So the alleles are together in the parent, but they become segregated or separated during gamete formation. Here that's happening in the formation of sperm. Here that's happening in the formation of eggs. If you feel that that corresponds to the events of meiosis, you're onto something, and we'll talk about that later. Define the terms dominant and recessive. Dominant alleles are always observed in the phenotype of the offspring. They're represented by a capital letter. So, for example, the capital letter A. So here's an individual who's homozygous dominant, and they express the dominant characteristic, dark black fur. In these heterozygotes, there's also a recessive allele, but because the dominant allele is present, then the characteristics of the organism is determined by the dominant allele. A recessive allele only shows up in a homozygote. So this mouse over here is homozygous recessive, and therefore it has the recessive appearance. That homozygous allele recedes into the background when it's paired with a dominant allele, and these are represented in the Mendelian system by a lowercase letter, in this case, lowercase a. What's the difference between genotype and phenotype? Phenotype is your appearance. It's the observable characteristics in an organism. An example is brown eyes. Genotype is the genes that you've inherited, the type of genes that you have. It's your underlying DNA. In my case, my eye color is brown. That's a dominant phenotype, though eye color is actually complex. There are many alleles involved in determining eye color. Actually, I think it's about 3, but I'm a heterozygote. So my genotype would be like capital B, little b. How do I know that? Because my wife has blue eyes and both of my kids have blue eyes, and that would only be possible if I were a heterozygote, because if I was a homozygote, then I could only pass on the big B allele, and then both of my children would have been heterozygotes. Explain what a monohybrid cross is and what the results of such a cross will be. In your response, create a Punnett square to demonstrate your understanding. This, which we've seen before, is a Punnett square. It uses the rules of probability to quickly predict the offspring of genetic crosses. A monohybrid cross is a cross between 2 heterozygotes. So these are peas, which was one of Mendel's study organisms, and this pea has purple flowers. The genotype is capital P, little p, capital P, little p. Why? Because it's a heterozygote, and this is a monohybrid cross, so they're hybrid for one characteristic. Now, the result will be offspring in a 3-to-1 ratio. So how do you do these Punnett squares? Well, you start by identifying the genotype of the parents. Well, in the problem, it's given to you. It's a monohybrid cross, big P, little p, crossed with big P, little p. So you can think of this as a germ cell. It's going to go through meiosis. The homologous pairs are going to separate, principle of segregation. So some of the gametes will have big P, some will have little p. And that's true for each parent here, big P, little p. Flowers are hermaphroditic, so it doesn't really make sense to talk about father and mother in this case. What you do is then the gametes will fertilize one another. You know, if this were a pollen, the equivalent of sperm, it would fertilize the egg, assuming this is the female and this is the male over here. But in other words, this combines with this, and we have this organism is capital P, capital P. It's got the dominant phenotype. It's homozygous dominant. This organism gets a big P and a little p. It's a heterozygote, and it still has the dominant phenotype. Same with this one over here. This one gets 2 little p's, one from each parent, and as a result, it shows the recessive phenotype, and that recessive phenotype is white flowers. We have 3 individuals with the dominant phenotype, 1 with the recessive phenotype. 1/4 of the offspring are homozygous dominant. Half of the offspring, 2/4, are heterozygous. They still have the dominant phenotype, and 1/4 will be homozygous recessive, and they'll show the recessive phenotype. That's how you do a Punnett square for a monohybrid cross. In genetics, what are the P, F1, and F2 generations? This is a common notation that's used in genetics, and you, as an AP biology student, need to understand it. The P generation is the parental generation, and in general, they're true breeding homozygotes. Now, notice that we've jumped in complexity. In this example, we're not dealing with just 1 gene, we're dealing with 2. So there's a gene for tallness, and there's the gene for flower color. And the alternatives are tall versus short, and purple flowers versus white flowers. When you breed the P generation together, those offspring are called F1s. It's the first filial generation. And in this case, they're heterozygotes. They're double heterozygotes because they're big T, little t, that's a heterozygote, and big P, little p. So double heterozygotes. Now, let's go from the F1s to the F2s. You breed together the F1 generation, these double heterozygotes, and you get the F2s. So the F1, that's the first filial generation, and the F2, it's the second generation. It's the grandchildren of the P generation. And crossing the dihybrid F1s. Why are they dihybrid? Because they're double heterozygotes. They're hybrid. They're mixed for 2 characteristics. That's called a dihybrid cross. We're going to take that apart in just a moment. Define Mendel's principle of independent assortment. Independent assortment, it's what we talked about in the context of meiosis. This is essentially the same process. Mendel was able to intuit this just looking at the transmission of traits from one generation to the next. Genes carrying different traits are segregated and passed on independently from one another. So in an organism that's dihybrid, big T, little t, big P, little p, the way that the T gene pair gets passed on is independent from the way that the P gene pair gets passed on. And therefore, in a double heterozygote, a dihybrid, big T, little t, big P, little p, 4 unique gametes can be created. Here they are. Big T, big P, big T, little p, little t, big P, little t, little p. How can you figure this out for yourself? It's very, very simple if you use the FOIL algorithm. That's for factoring binomials. You should know it from algebra. If you don't, just remember it. First, outside, inside, last. So you think about this and you think about the gene pair. So you take the first one, big T, big P. You take the outside one, big T, little p, that's over here. Then you take the inside one, little t, big P, that's over here. And then the last one, little t, little p. So 4 gametes can come from a dihybrid organism. This is limited to genes that are found on different chromosomes. The game completely changes when those genes are on the same chromosome, and we'll deal with that later. Explain what a dihybrid cross is and what the results of such a cross will be. It's a cross between 2 double heterozygotes. A couple of slides ago, we were looking at the production of the F2s when there was a dihybrid cross. We're crossing big T, big P, little p with the same. You're going to use the FOIL algorithm to figure out the gametes, and we just described that in the last slide. You just bring these alleles down and over and combine them. You do that for all 16 squares of this dihybrid cross, and what do you wind up with? You wind up with a 9:3 to 3:1 ratio in the offspring, and that is actually something that's worth memorizing. What's the connection between Mendel's laws of inheritance and what happens during meiosis? We've just articulated Mendel's 2 laws, the principle of segregation and the principle of independent assortment. The principle of segregation, according to Mendel, says that parents have 2 alleles for each trait, but pass on only one to their offspring, which inherit their 2 alleles from 2 separate parents. In meiosis, there's a diploid parent that produces haploid gametes. The diploid phase corresponds to the 2 alleles in each parent. The haploid phase corresponds to the 1 allele that the parent passes on to their offspring. In zygotes, that diploid condition is restored, and that's not shown in this diagram over here. Independent assortment, according to Mendel, is what happens to one gene pair is independent of every other gene pair, at least in the ones that Mendel studied. In meiosis, chromosomes assort independently during metaphase 1 of meiosis. What is the rule of multiplication? Demonstrate your understanding by explaining how to predict the probability of genotype little a, little b, little c, resulting from a trihybrid cross between big A, big B, little b, big C, little c times the same. Don't make a huge Punnett square to try and solve problems like this. What you want to do instead is use one of the laws of probability, the rule of multiplication. Here's what it is. The probability of independent events occurring together is the product of their individual probabilities. Here's how to think about this. It's like 3 independent Punnett squares, because what each gene does is independent of every other gene. What A is going to do is independent of B, is independent of C. So 3 independent Punnett squares, 1 for A, 1 for B, and 1 for C. But you don't have to create 3 Punnett squares. You just have to use the rule of multiplication. In a cross between big A, and big A, the probability of little a is 1 out of 4. It's the same for B, 1 out of 4, and it's the same for C, 1 out of 4. These are independent events, so you use the rule of multiplication. It's 1/4 times 1/4 times 1/4, or 1 out of 64. Topics 5.4 and 5.5, non-Mendelian genetics and environment-phenotype interaction. Non-Mendelian genetics is about genetic principles that were discovered after Mendel's original contributions. One of the most important of these involves linked genes. What are linked genes? Describe their inheritance pattern and explain which Mendelian rule linked genes don't follow. Linked genes are genes that are on the same chromosome. So fruit flies, as opposed to peas, were the widely used experimental organism to discover the principles of non-Mendelian genetics. And here you can see one chromosome from a fruit fly, and there's a whole variety of genes, sequences of DNA that code for specific traits that are located on the same chromosome. One has to do with this kind of bristled appendages on the head. One controls body color. One controls one type of eye color. One controls wing length. These genes are mostly inherited together, which is different from the independent assortment that we saw with Mendelian genetics. Because they're on the same chromosome, these genes don't independently assort. So like, for example, above, genes T and A in this cell over here, they're linked. In this cell over here, they're not linked. These would independently assort, and these wouldn't. So what happens in crosses involving linked genes? Note, first of all, that we have a different symbol system over here. You can see that right over here. We have B plus, VG plus, VG plus. In this system, in non-Mendelian genetics, a plus sign indicates the wild type or the dominant allele. If you have a symbol that can be more than one letter without a plus sign, that indicates the recessive. In this P cross, what we're doing is we're crossing normal body, normal winged fly, B plus, VG plus, VG plus, with a black-bodied vestigial winged fly. Those are both recessive traits. And note that all the F1 offspring are dihybrid. They're B plus B, VG plus VG, and they have both dominant phenotypes. This is what you'd expect in a Mendelian trait for the F1s. We have a gray body and normal winged fly, which has all of the dominant characteristics. Now over here, what we're doing is we're representing this chromosomally. B plus, VG plus, and BVG. Notice that B plus and VG plus, they're on the same chromosome, and BVG are on the same chromosome too. These genes are linked. What if they were perfectly linked and they never separated? What would you expect to happen? The method here is a little bit different than the method in a Mendelian cross. We're not doing a dihybrid cross, we're doing what's called a test cross. And a test cross, a dihybrid, so B plus B, VG plus VG, is being crossed with a double recessive. And the way that it's done is the double recessive is the male, the female is the double hybrid, the double recessive. I've represented this over here. Here's a representation of the female. On one of her chromosomes, she'll have B plus and VG plus. On the other chromosome, she'll have B and VG. The male is a double mutant, and he has B and VG on both of his chromosomes. When the female produces gametes, then half of her gametes will have B plus and VG plus, and half of her gametes will have B and VG. Now that's assuming perfect linkage. In the male, all of the sperm have to have B and VG. Here's a Punnett square. These are the eggs, that's going here. The other eggs are going here, the other half of the eggs, and all of the sperm are going over here. You put them together, and what you'd expect is that half of the offspring would be B plus B, VG plus VG. The other half of the offspring would be B, B, VG, VG. This organism, these offspring would have normal body and normal wings. That's 50% of the offspring. And the other 50% of the offspring would have black body and vestigial wings. But that is not what happens. And what I'm doing here is I'm letting you know what you would expect if there were perfect linkage. What actually happens in a cross involving linkage? Note that the numbers won't always be the same, but the general concepts apply. What we see is that the majority of the offspring have parental phenotypes. What does that mean? Well, the mom's phenotype was gray body with normal wings, and many of these flies have that phenotype, gray body, normal wings. The father's phenotype is black body with vestigial wings. Many of the offspring have that phenotype, but a significant number of offspring have recombinant phenotypes. What are recombinant phenotypes? They take one of the phenotypes of the mom and they combine it with one of the phenotypes of the dad. That's what's happening over here in these flies that have a gray body like the mother and vestigial wings like the father. Or you can have a black body like the father with normal wings like the mother. Those are recombinant phenotypes. Why do we have most of the offspring having parental phenotypes, but a significant minority having recombinant phenotypes? It's because linkage is not perfect. Genes that are linked don't always stay together. Why not? Because during meiosis, there's recombination and crossing over. So linked genes, because of that process, can separate. We'll see the details of this in the next slide. What happens in crosses involving linked genes? Why are there recombinants? In diagrams A and B, we have a dihybrid female and her germ cells. In C, we have meiosis 1 with crossing over. We have homologous pairs. They're going to get together. They're going to swap genes and the production of recombinant chromosomes. But notice that some of the sister chromatids are recombinant and some aren't. So now what we're going to do is we're going to complete meiosis. We're going to go through gamete formation. And when we do that, note that of the eggs that the female produces, 2 of them at letter H, they're recombinant. What do I mean by that? Well, they have B plus and they have VG. In other words, this chromosome is B plus VG plus, this chromosome is B VG. Well, the recombinant ones are the ones where those alleles swapped places. So this is a recombinant B plus VG, this is a recombinant B VG plus, but these 2 at letter I, those are parental. Right? They're just like the parents, B plus VG plus BVG. Now over here we have the male parent who's a homozygote, and his gamete, he can only produce one kind of gamete. He's homozygous. He can only produce a gamete that has B and VG. Here's what happens with the offspring. Offspring N and P have recombinant phenotypes. In other words, they have, in this case, gray body vestigial wings. That's recombinant. Neither of the parent looked like that. And this is black body normal wings. Again, recombinant. Neither of the parent looked like that. But O and Q are parental phenotypes. O is gray body normal wings and Q is black body vestigial wings. Why are there so many more parentals than there are recombinant phenotypes? It's because crossing over happens, but it happens at a rate that's dependent on the distance of these alleles on the chromosome. And so the closer 2 alleles are, the less they'll tend to cross over. And this frequency, which is, I believe, 17%, that represents the distance between the B allele and the VG allele on the fruit fly's chromosomes. What's the relationship between the percentage of recombination and the distance between any 2 linked genes? The further apart any genes are on the chromosome, the higher the percentage of recombinant gametes. In the chromosome map above, genes A and E will recombine the most because they're the furthest apart. In other words, there can be crossing over here, over here, over here, and all of those will get E to jump over here and big E to jump over here. That's going to look like recombination. That's going to actually almost look like independent assortment because they're so far away. But genes B and C, they'll recombine the least because the only way that they can cross over is if there's a chiasma right over here at this specific spot that would enable little c to jump over here, big C to jump over here, or B could do the same. The percentage of recombination can be used to calculate the map distance between 2 alleles. So over here, this is a chromosome map, and it's saying here that the distance between this long bristled appendages called long aristea and gray body, that's 48.5. Well, those are units that reflect the frequency of recombination. Over here between gray body and red eyes, that's a little bit under, that's 9 recombination units. And really what this amounts to is the frequency of recombinants. So doing all of these mapping experiments or doing all of these breeding experiments with fruit flies, the researchers at Columbia University in the 1900s, Thomas Hunt Morgan and his crew, were able to create maps of chromosomes. And this was establishing that genes are on chromosomes and was part of the pathway that ultimately led to the double helix and all that other great stuff. Note that these are not physical maps so that the numbers don't add up, but this should get you far enough to have a basic understanding. Again, on learn-biology.com, I have a whole tutorial about linkage and recombination with practice problems that'll get you all the way there. Your success in AP Biology starts here. Are you struggling with AP Bio? With learn-biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to learn-biology.com to find out how you can master your biology course and crush the AP Bio exam. What are sex-linked genes? Sex-linked genes are located on the X chromosome. Males, because the genes are located on the X chromosome, can't be heterozygous. They either have the allele or they don't. So the whole heterozygous, homozygous thing doesn't apply to males. But females can be heterozygous, as shown. In this case, it's X big H, X little h, a heterozygote. Or they could be homozygous dominant. That would be X big H, X big H. Or they could be homozygous recessive, X little h, X little h. Using hemophilia as an example, how can males inherit a recessive sex-linked trait? Hemophilia is a blood clotting disorder, and essentially hemophiliacs can't clot their blood. It's much more common in males than it is in females because the allele for hemophilia, the mutated allele that leads to ineffective blood clotting, is on the X chromosome. Sons inherit their X-linked alleles from their mothers. So we'll look at the Punnett square first. This Punnett square is from learn-biology.com. These boxes are things that you would drag the alleles into. Here we have a hemophiliac, X little hy. It's a recessive X-linked condition. How did this young man inherit that X-linked allele? From his mom. Mom is a heterozygote, X big H, X little h. She's not a hemophiliac, but she carries the allele. It's commonly known as a carrier. The dad, in this case, is unaffected, X big HY, but it doesn't matter because dads don't pass on their X chromosomes to their sons. They pass on their Y chromosomes. That's why they're sons to begin with. So the mom has to be a heterozygote, as shown here, or she could be homozygous recessive, though that's quite rare. We'll look at that in the next slide. Here's a pedigree that shows the same thing, basically a cross between a heterozygous female and a normal male. And note that in this case, the mom passed on her normal chromosome, and the dad passed on his normal chromosome. It's the only X chromosome that he has to pass on. In the case of these two sons, the mom passed on her defective X chromosome with the hemophilia allele. So we have two sons who are X little hy. They're both hemophiliacs. This daughter over here is a carrier. She received the normal X chromosome from her dad and an X chromosome with the mutant hemophilia allele from her mom. And here we have a son who inherited the normal X chromosome from the mom. She had one of two to give. He got lucky, and the Y chromosome from the dad. So he's a normal young male. Examples of X-linked recessive conditions in humans, hemophilia, which we just talked about. Also, a very common one is red-green color blindness in fruit flies, which have a similar sex determination system to humans. It's just the X chromosome and the Y chromosome, just like mammals. The allele for white eyes is a mutation that's on the X chromosome in fruit flies. And in fact, this was the first allele that was ever located on a specific chromosome. Can a female inherit a recessive sex-linked trait? Absolutely, but it's uncommon. Here's what has to happen. The male parent must have the sex-linked recessive trait. So here we have a wide-eyed fruit fly, X little w, Y. The female must be a heterozygote in this case, or have the trait. So it could be a wide-eyed female. Here the female is X big W, X little w, and when we create the Punnett square, we can see that among the female offspring, 50% of the offspring are carriers. They have to be carriers. They get the normal allele, for red eyes in this case, from their mom. They get the recessive allele for white eyes from their dad. And in this case, they get the recessive allele from both parents. So 50% are carriers and 50% of the females have the trait. Among the male offspring, 50% of the males are normal. Why? Because the mom's a heterozygote. She passes on her normal X chromosome with the red-eyed allele to half of her sons, but there's also the 50% chance that she'll pass on the recessive mutated allele, and that's why this offspring over here will be white-eyed. But the key thing that we were looking for was, how can a female wind up inheriting a recessive sex-linked condition? She can if the dad has the condition and the mom is a heterozygote. What is non-nuclear inheritance? I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. It's inheritance of genes that are not on a nuclear chromosome, but on the chromosome of a mitochondrion or a chloroplast. A nuclear chromosome is one of the autosomes or a sex chromosome that are found inside the nucleus of a eukaryotic cell. These genes that are on mitochondria or chloroplasts are only passed on to the offspring through the female gamete. Why? Let's look at the example of sperm. Sperm have mitochondria. The mitochondria power the flagellum that enables the sperm to swim towards the ovum. But when there's fertilization, only the head of the sperm penetrates through the egg membrane, or is allowed in through the egg membrane, and all of the mitochondria are left outside. So all of the mitochondria that get passed on to the offspring are mitochondria that were passed on through the female germ cell. The inheritance pattern of these mitochondrial or chloroplast genes doesn't follow Mendelian patterns. You can see that in this pedigree over here, where the mom passes on the mitochondrial gene to all of her offspring, male and female. But the male who inherits the mitochondrial gene doesn't pass it on to any of his offspring. The male is essentially a mitochondrial dead end, if we're talking about mitochondria, where the female passes on her mitochondria to all of her offspring. In the case of mitochondrial inheritance in eukaryotes, all of one's mitochondria are inherited from one's mother. And in plants, they also have mitochondria, so the same rule applies, but they also have chloroplasts, and only the ovule, not the pollen, passes on chloroplasts and mitochondria to the offspring. So it's the same thing. It's female line inheritance of alleles when it relates to genes that are on an organelle, such as the mitochondria or a chloroplast. What is incomplete dominance? This is when the phenotype of the heterozygote is different from, and usually intermediate between, either of the homozygous phenotypes. Neither allele is dominant. So in this case, we have to use a different notation system. This is carnations, and C big R with a superscript represents the red flower color allele. C big W with a superscript represents the white flower color allele. So here's our P generation, C big R, crossed with C big W, C big W. The F1 generation, they're all hybrids, C big R, C big W, but they're pink. It's because 2 doses of gene expression, in other words, this is DNA that's coding for protein, and you need that much protein in order to produce that vibrant red color. If you just have one, then it's not enough to push the phenotype all the way to red, and you wind up with this intermediate phenotype. One dose can produce sufficient pigment to create the red color. Let's see what happens when you do your F1 cross to produce the F2 generation. So we have a pink carnation, C big R, C big W, crossed with another pink carnation, C big R, C big W. What do you get in the offspring? You get one offspring that's red because it's C big R, C big R, one offspring that is white because it's C big W, C big W, and two that have the intermediate phenotype pink because they're going to be C big R, C big W, C big R, C big W. Explain how the same genotype can result in different phenotypes under different environmental conditions. This is also known as an environment-genotype interaction. And what happens is that factors in the environment influence gene expression, and that leads to variation in the subsequent phenotype. And basically what it's saying is something that's fairly well known, that genes don't determine everything. Genes interact with the environment. And note that this is the norm, not the exception. Here are two examples to know about. If you have hydrangeas in your yard, you can determine the color of the hydrangeas because the flower color is determined not by genes but by the acidity of the soil. In a more acidic soil, shown over here, you'll get this beautiful purple color. In more alkaline soil, you'll get reddish color, and there's actually mixes that you can add to the water that you give to your hydrangeas that will adjust the alkalinity or acidity of the soil. Example number 2 is dark fur regions in mammals, and they develop in cooler body regions away from the core. This is a Himalayan rabbit, but if you can picture a Siamese cat, it's pretty much the same thing. So this is a cool region, this is a cool region, this is a cool region, and all the cells have the same genes, but the cells that are being exposed to cooler temperatures express different pigment alleles than those that are in the warmer regions of the body. There are many other examples. Height and weight in humans is caused by an interaction between the genes that someone inherits and the environment that they're in. The way that your skin color can change in relationship to the amount of sun that you're exposed to. That is genes being expressed in relationship to environmental cues. We talked earlier about how sex is determined by temperature in certain reptiles. Another example of environment genotype interaction. Topic 6.1, DNA and RNA structure. Describe the structure of DNA. DNA is a double-stranded helical molecule composed of nucleotide monomers. In this flattened out representation of DNA, here's one strand, here's the other strand. In this helical representation, you can see one strand, another strand. Because there's two strands, it's a double helix. The monomers are nucleotides. They consist of a 5-carbon sugar called deoxyribose, hence deoxyribonucleic acid, a phosphate group, and one of 4 nitrogenous bases. So it doesn't have to have this exact structure. It can vary over here in terms of the nitrogenous base. Each strand consists of covalently bonded deoxyribose sugars and phosphate groups, which comprise DNA's sugar-phosphate backbone. Within the helix, bases with complementary shapes bind through hydrogen bonds. Thymine is complementary to adenine. Guanine is complementary to cytosine. In the case of adenine and thymine, you can see the hydrogen bonds that form between the oxygen over here, the hydrogen over here, hydrogen over here, nitrogen over here. The bonding follows base pairing rules that you have to commit to memory. Adenine binds only with thymine, A binds with T. Guanine binds only with cytosine, G binds with C. For the nucleotides to bind, they have to be oriented upside down relative to one another, making the two strands anti-parallel. This strand has its 5' end over here, its 3' end down here, and this strand is the opposite, anti-parallel, like this. Explain how DNA's structure allows it to serve as the molecule of heredity. We'll start with information storage. The 4 bases can occur in any order. I've represented them over here as A, C, T, G over and over again to show the structure, but there could be 3 A's in a row, followed by 2 C's, followed by a T, followed by whatever. The sequence isn't determined by DNA's chemistry. That allows the sequence to be an informational code that specifies sequences of RNA and protein. Replicability. The specific base pairing, ATGC, that we talked about previously, allows each strand to serve as a template for the synthesis of a complementary strand during DNA replication, which we'll talk about in a moment. That also ensures high fidelity transmission of genetic information from parent cells to daughter cells. DNA is highly stable. Its double helical structure protects the sequence of bases that are inside. But while it's stable, it's also capable of mutation. Mutability is the fourth characteristic. There's a low level of mutation where bases can change from one to another, either spontaneously or caused by mutation-causing factors in the environment, and that allows for change in this code, which allows for evolution. Compare and contrast the functions of DNA and RNA. DNA is the molecule of heredity in all organisms. Anything that is cell-based life, which is all life, has DNA as its molecule of heredity, as the stuff that genes are made of. RNA is the hereditary molecule in some, but not all, viruses. Viruses that you might know about that are RNA-based include HIV and SARS, a form of which just caused the COVID-19 pandemic. In all organisms, RNA is involved in information transfer related to protein synthesis, how DNA becomes RNA and how RNA becomes proteins. And that includes forms of RNA such as mRNA, tRNA, and rRNA. In eukaryotes, RNA is also involved in the regulation of gene expression, and this previews some topics that we'll talk about later in this unit. That includes splicing out introns, non-coding DNA from pre-mRNA to create mRNA, and regulating protein synthesis. Compare and contrast how genetic information is stored in prokaryotes and eukaryotes. Prokaryotes store their DNA in looped circular chromosomes. In other words, the beginning and the end is connected. It's sometimes described as circular, but looped is really more accurate. The genomes of bacteria and archaea range from about 100,000 base pairs to 10 million base pairs. And their DNA is naked. It's not wrapped around a protein scaffold. In eukaryotes, the DNA is organized into multiple linear chromosomes, so there's one end and there's another end, and the DNA is wrapped around these proteins that are called histones. Eukaryotic genomes are much larger than prokaryotic genomes. The human genome, as one example, consists of 3.2 billion base pairs, but there are some plant genomes that consist of 150 billion base pairs. What are plasmids? What's their function? How are they used in genetic engineering? Plasmids are small extrachromosomal loops of DNA, commonly found in bacteria, less commonly in archaea, rarely in eukaryotes. Here's the main bacterial chromosome. These loops, also made of DNA, are the plasmids. They're involved in horizontal gene transfer between bacterial cells through a process called conjugation. These transfer genes, because they're transferring DNA that codes for protein from one cell to another, they're often for antibiotic resistance. Plasmids are commonly used in genetic engineering for replicating DNA and for expressing engineered genes within bacterial cells. Both horizontal gene transfer and genetic engineering are going to be covered later in Unit 6. Topic 6.2, DNA replication. Your success in AP Biology starts here. Are you struggling with AP Bio? With Learn-Biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to Learn-Biology.com to find out how you can master your biology course and crush the AP Bio exam. On a big picture level, describe how DNA replication occurs and what the term semi-conservative means. During DNA replication, a team of enzymes using each strand of the double helix as a template synthesizes new daughter strand. Here's the original strand. Enzymes pull that strand apart, and that results in two daughter strands that are each single strands. These single strands serve as a template, and what that means is that nucleotides that are available in the nucleus following the base pairing rules will bind with the exposed strand. A will bind with T, C will bind with G, etc. The result is that each daughter DNA double helix consists of one conserved strand from the parent molecule and another strand that was synthesized anew. In these daughter molecules, one of the strands is this strand. Here we go. The other strand is this strand, whereas these strands over here are new, semi-conservative. One strand is conserved, the other strand is new. You can see that represented here with a kind of color coding where the parent strand has both strands colored red, and you can see in the daughter strands, one strand is red, conserved, one strand is orange, new. That method of replication is known as semi-conservative replication. Describe how DNA replication starts. In the model of DNA replication that we're about to talk about, there's a lot of simplification compared to how the process actually works in nature, but don't worry, this is exactly what you need to know for AP Biology. The process begins when an enzyme called helicase over here at B finds a sequence called the origin of replication that basically says start replicating here and separates the double-stranded DNA. As you know, that means breaking the hydrogen bonds that are holding the two strands together. This exposes two single strands, and it creates a structure that's called a replication fork. Describe the roles of DNA polymerase, primase, and primers in replication. Let's get oriented here before we start. This is a replication fork. This is DNA helicase that's opening up the parent strand, exposing the nucleotides in the two daughter strands. DNA polymerase is this enzyme shown over here and over here. It's the key enzyme involved in creating new DNA. The parent DNA is shown in dark blue. The new DNA that's coming in is represented in light blue. The nucleotides bind based on base pairing rules. DNA polymerase doesn't know which nucleotide should fit. The knowledge is basically in the template strands. So if there's a C over here, then a G will bind. If there's an A over here, then a T will bind. What DNA polymerase does is it binds new nucleotides to the 3' end of a growing strand, and that's a sugar-phosphate bond. DNA polymerase has a limitation. It can only add nucleotides to an existing strand. So think of it as an enzyme, and it has an active site. Its substrate is the pre-existing strand and the new nucleotide that came in. So to start the process, DNA polymerase needs an RNA primer, a couple of bases of RNA that DNA polymerase can start connecting DNA nucleotides to. Here's the primer shown over here. Here it's shown, and it's represented by number 4. There's another enzyme that can come to an open replication fork and start laying down that primer, and that's represented here at 5. It's called primase. Primase lays down the primer. What role do single-strand binding proteins play during replication? The single-strand binding proteins are shown at 8, and what they do is they keep the double helix from rewinding so that all of these other enzymes can get into place and carry out replication. How is DNA replication at the leading strand different from replication at the lagging strand? In each replication fork, there's going to be a leading strand and a lagging strand. In the leading strand, which is shown over here at J, DNA replication is relatively continuous because DNA polymerase at G is following the opening replication fork that's being created by helicase over here. In the lagging strand, which is shown at L, DNA polymerase synthesizes in the opposite direction from from the opening replication fork. So what you have to imagine is that DNA helicase opens up the helix a little bit, DNA polymerase gets in and starts synthesizing, and then it opens up a little bit more. Well, DNA polymerase can't go in this direction. It can only go in the 5-3 direction, so that means another DNA polymerase comes in and synthesizes this over here, and yet another, and each time there's a primer. What that means is that on the lagging strand, DNA replication is discontinuous, and it's built from short sequences that are called Okazaki fragments, named after the researchers who discovered that this is how the process works. Describe the roles of DNA polymerase I and ligase during DNA replication. DNA polymerase I, shown over here at K, removes the RNA primers. Here's one over here, here's a whole bunch over here, and it replaces the RNA with DNA. Another enzyme called DNA ligase is required to finish the process and create the complete daughter strands. What it does is it seals the gaps between fragments with sugar phosphate bonds. Topic 6.3, transcription. Explain the overall flow of genetic information within cells. This is the central dogma of molecular genetics, which is DNA makes RNA makes protein. Information flows from a sequence of DNA triplets to a sequence of mRNA codons to a sequence of amino acids. What is a gene? If you've been following this series, we looked at this slide in Unit 5, but now let's look at it again in the context of molecular genetics. A gene is the basic unit of heredity, passed from parent to offspring. It determines a trait. In terms of molecular genetics, it's a sequence of DNA nucleotides that codes for RNA, which codes for protein. List the principal forms of RNA and describe the function of each one. mRNA, or messenger RNA, is a linear molecule, and it brings instructions from DNA to ribosomes. rRNA, ribosomal RNA, makes up the catalytic part of ribosomes and binds amino acids together during protein synthesis. Ribosomes are these particles that are composed of rRNA and protein. We'll look at them in depth later, but they're essentially enzymes, and they're the enzymes that bind amino acids together during protein synthesis. tRNAs, transfer RNA, bring specific amino acid to the ribosomes, again for protein synthesis. Small RNAs are a large group of RNAs of different shapes and sizes, and they're involved in eukaryotic gene regulation. What happens during transcription? Transcription is the creation of RNA, which we see over here in blue, from DNA. Every gene begins with a promoter region that indicates that that's where the gene starts. And during transcription, an enzyme called RNA polymerase binds with the promoter on DNA. Then it transcribes the sequence of DNA bases on DNA's template strand into a sequence of RNA. RNA polymerase, like all of the enzymes involved in working with DNA, reads the DNA in the 3' to 5' direction and synthesizes new RNA in the 5' to 3' direction. And when the RNA polymerase reaches a terminator region, which is at the end of the gene, it dissociates from the DNA, ending transcription. Define and describe template strand, minus strand, non-coding strand, or antisense strand in relationship to RNA transcription. The template strand, it's this one over here in blue, it's also called the non-coding strand, the antisense strand, and the minus strand. That's what gets transcribed from DNA into RNA. The complementary strand to the template strand is called the coding strand. Why? Because you can see that it has the same sequence of nucleotides as the mRNA will. Here's the coding strand, GGTTAA. Here's the RNA that's being produced, GGUU. U substitutes for T in RNA, AA. So GGUUAA, GGTTAA. It's the same. Why is it the same? Because it was created in response to this template strand over here. That's why the coding strand is called the sense strand, or the positive strand. What are some unique features of prokaryotic transcription? Prokaryotes don't have a nucleus. There's no separation between the genetic material and the cytoplasm. As a result, in prokaryotes, transcribed RNA, which is shown here at D, can immediately be translated by ribosomes into protein. And that's what you see as these strands over here. Often, multiple ribosomes read the same RNA strand. These multiple ribosomes are sometimes called polysomes, and you can see them in a more zoomed in version over here. The genetic code and translation / protein synthesis. What is the genetic code? How do you read a genetic code chart? Use the code to translate AUG, GUC, AAG, GUU into protein. The genetic code is the code used by living things to translate nucleotide sequences into amino acid sequences. We've talked about how DNA makes RNA, makes protein. DNA to RNA is transcription. Now we're going to get from RNA to protein. In the genetic code, groups of 3 RNA nucleotides, so like for example AUG, GUC, they're called a codon, and they code for one amino acid. Codon, code 1. The code is nearly universal. Nearly every living thing uses it in exactly this way. There are some minor exceptions. It's specific. Every codon can determine one amino acid, but it's redundant. There are synonymous codons. AUG, GUC, AAG, GUU codes for methionine, valine, lysine, valine. How did I do that? Here's how you use the code. AUG, this is the first nucleotide in the RNA codon. This is the second nucleotide in the RNA codon, and this is the third. So you work from the inside out. AUG codes for methionine. GUC codes for valine. AAG codes for lysine, and GUU codes for valine. So we can see how GUC and GUU are synonymous, and that's an important relationship. Often the first 2 nucleotides are more important than the third one, and ones that have the same 2 first nucleotides are often synonymous. Use the genetic code dictionary below to code out CUC, GAU, GCA, GUC, CGU. The code that we looked at on the previous slide is a circular code. I actually think it's easier to use, though you'll frequently see codes like this that are tabular. In this code, the first base is represented on this column, the second nucleotide base is represented over here, and the third is going to be one of these 4. Let's demonstrate how that's used. CUC is the first codon. C, U, and here's C, that codes for leucine. GAU, G is the first base, A is the second base, U codes for asparagine. GCA, G, C, A codes for alanine. GUC, G, U, C codes for valine. And CGU, C, G, U codes for arginine. Here you see all of the amino acids and their corresponding codons represented here. What's the big picture of translation? Who are the key players? mRNA is shown over here at G2. It contains the codons, this group over here, AUG, or this AUG, or this CCG, that specify the order of amino acids. Here are amino acids shown over here. The ribosome connects amino acids to create a polypeptide. Ribosome is represented as K, and what it does is it'll connect, for example, this amino acid, proline, to this growing chain of amino acids. A chain of amino acids is also referred to as a polypeptide. Remember, that's the first level of protein structure, the linear sequence of amino acids. tRNAs are shown over here at letter O. Here's a tRNA. And tRNAs bring amino acids, such as this one over here, to this ribosome mRNA complex. tRNAs have an anticodon, such as this over here, GGG, letter H, and an amino acid binding site over here. In the next slides, we'll put the entire process together step by step. What is the role of the ribosome in translation? What are the key parts of ribosomes to know? Ribosomes are general purpose protein factories. They can take any mRNA, which is what brings it information, and convert it into any sequence of amino acids. They're the enzymes that string together amino acids to form polypeptides, and they do that following the instructions in mRNA. They have a large and a small subunit, and they have 3 tRNA binding sites. The E is the exit site. That's where the tRNA that's given up its amino acid will leave from. The P site holds the polypeptide, and the new amino acid comes into the A site. Describe how translation protein synthesis begins. Processed mRNA, that means mRNA that's ready to be translated, that doesn't have any introns in it, leaves the nucleus. It'll leave through a nuclear pore. The small ribosomal subunit will bind with the mRNA and make its way over to the start codon, which is AUG. That's where translation begins. That small subunit then waits, quote unquote, for a tRNA with a matching anticodon to bind with the start codon. So this tRNA has the anticodon UAC, which complements AUG. This first tRNA is carrying the first amino acid, methionine. Then the large subunit binds with the small subunit. The ribosome is now complete, and that first tRNA with methionine is located in the ribosome's P site. That's the middle binding site. Describe the elongation phase of translation. The next tRNA comes to the A site, and it bears a new amino acid. Here it is. The ribosome then catalyzes a peptide bond between the P and A site amino acids. So here's a peptide bond that's forming between methionine and valine. Then the ribosome translocates. It moves over one more codon. So that means that a dipeptide is now hanging off the P site amino acid. The A site is empty, and there's a tRNA in the E site, but it's not connected to the polypeptide. It's not connected to any amino acid. Now what happens is that the tRNA that's in the E site exits. The new tRNA enters at the A site. The A site's empty over here. A new tRNA comes in. The ribosome is going to catalyze a bond between that new amino acid and the existing dipeptide. So there's going to be a tripeptide that's temporarily at the A site, but the process is going to continue. It will be another round of translocation, followed by exit, followed by another tRNA that's charged with an amino acid coming in, followed by another peptide bond happening. And that continues along the entire length of the mRNA. Describe the termination phase of translation. The ribosome gets to a stop codon. The stop codons in the genetic code have no corresponding tRNA. Instead, what they do is they code for a release factor. That's a protein that can bind with the stop codon and induce certain changes in the mRNA, tRNA, ribosome complex. And those changes cause the ribosome to dissociate and the polypeptide to be released. The only thing that needs to happen now is for this polypeptide to fold up into a functional protein. Translation is done. I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Topic 6.5 to 6.6, Regulation of Gene Expression, Part 1, Operons. Let's start with a little context. E. coli is a bacterial cell that lives in our colons. That coli is related to colon, and it also lives in the colons of many other animals. The colon is the large intestine. E. coli has about 4,000 genes. This is a chromosome map of E. coli's chromosome, and it shows a small portion of these 4,000 genes, which code for a variety of proteins. The overall genome of E. coli consists of about 4 million base pairs, A, T's, C's, and G's. And this leads to a question of regulation, which is, what's the control system for turning its genes on and off? Let's start by responding to this very general question, what is an operon? One definition is that an operon is a cluster of genes transcribed as a single RNA. Here we have a portion of DNA that's labeled structural genes, and it's all transcribed as one RNA transcript, but then that RNA is processed so that it's producing a variety of enzymes. A cluster of genes transcribed as a single RNA. But our focus in AP Biology is that an operon is a mostly prokaryotic system of gene regulation that has control elements that allow for gene regulation. Describe the structure of an operon. Here we see a string of DNA, and it's an operon. It consists of structural genes, and those are genes that code for protein. There's an operator, which is where a repressor protein binds, and that enables this system to be regulated. There's a promoter where RNA polymerase binds, and there's a regulatory gene that produces the regulatory protein. That regulatory protein is generally a repressor protein that binds at the operator. How does the trip operon work? The trip operon is a system that codes for a series of enzymes that make tryptophan. That's what the structural genes do. These enzymes work as part of a metabolic pathway that codes for tryptophan, which is one of the 20 amino acids. But it's also a regulatory system that only turns on production of these enzymes at certain moments. If there's no tryptophan in the environment, then the regulatory protein over here, which is produced by the regulatory gene, and that's pretty much always on, that can't bind with the operator. Look at the shape of this regulatory protein over here, and notice that this part won't bind with this. And really what we're talking about is a protein with a complex shape that can actually bind with a sequence of DNA, because that's what the operator is, it's DNA. That means that RNA polymerase can bind at the promoter, and it can roll down the length of the gene and transcribe the structural genes, creating these enzymes. When tryptophan is in E. coli's environment, that tryptophan, the amino acid, will diffuse into the cell. What will happen? When tryptophan is in the cell's environment, then what will happen is it'll bind with the repressor protein. And when it does, that will cause the repressor protein to change shape. Think of this like an enzyme that's doing an allosteric shift. Binding over here causes a chain over here. How and why? This is a protein that has alpha helices and pleated sheets, and it's very dynamic. So the binding over here causes a change over here. That enables this regulatory protein, a repressor, to bind with the operator. When it does, it blocks RNA polymerase. That means that RNA polymerase can no longer transcribe these structural genes to make enzymes that synthesize tryptophan. That makes a lot of sense. The basic rule is if tryptophan is present, don't make it. It's an adaptation for saving energy. Trp is therefore called a repressible operon, and tryptophan is the co-repressor. This protein, when it binds with tryptophan, blocks the operator, repressing the system. Transcription becomes impossible. That's the trip operon. How does the lac operon work? We just looked at the trip operon, which controls the synthesis of enzymes for synthesizing tryptophan. What about this lac operon? The lac operon is an inducible operon, as opposed to trip, which was a repressible one, and it codes for enzymes that digest lactose, a disaccharide. So here's lactose. You can see it's composed of 2 sugar monomers, and the enzymes that digest lactose will break it down into glucose and galactose. What happens when lactose is in the environment? Remember, these bacteria live inside our guts. So if you had E. coli in your gut and you drank a glass of milk, the sugar in the milk, which is lactose, would then be in the environment of E. coli. That lactose will diffuse into E. coli. Once lactose is inside E. coli, it binds with the repressor protein. Here's lactose, it's binding with the repressor protein, and notice the effect. In this case, the lactose causes the repressor protein to change shape so it can't bind with the operator. That keeps the operator free, and when RNA polymerase binds at the promoter, it can roll down the length of the operon, it can transcribe the structural genes, and the structural genes produce enzymes that break down lactose into monosaccharides and also increase the permeability of E. coli cell membrane so that more lactose can enter. When lactose is absent, however, there's no lactose available to bind with the repressor. The repressor's default shape lets it bind with the operator. RNA polymerase, therefore, after binding with the promoter, can't transcribe the structural genes. The rule is if lactose is absent, don't make genes to digest it. Again, think of this as a metabolic adaptation. This saves energy. Don't make enzymes to digest something when the thing that you're digesting isn't around. Lac is therefore an inducible operon. It can be induced to be turned on. What turns it on? Lactose. Lactose is the inducer. The lac operon is a negative feedback system. Explain. Think about how the lac operon works. Lactose turns the system on. Turning the system on removes lactose from the system. Why? Because turning the system on, allowing RNA polymerase to transcribe these genes, allows for the production of enzymes and proteins that enhance lactose digestion. That enhanced lactose digestion will make all of this lactose go away. When all of this lactose goes away, there'll be no more lactose to bind with the repressor, which will then bind with the operator. The result is that the system turns off, and that's negative feedback, where the output of the system has the effect of quieting or repressing the system. You can say the same thing about the trip operon. It's also a negative feedback system. Why? The absence of tryptophan starts transcription. When tryptophan is not in the environment, then the regulatory protein can't bind with the operator. That enables RNA polymerase to transcribe these structural genes, producing these enzymes that are part of the metabolic pathway to produce tryptophan. That produces tryptophan, and the production of tryptophan puts tryptophan at high enough concentration in the cell so that it binds with the repressor protein, changing its shape, allowing the repressor protein to bind at the operator, shutting down transcription. That turns the system off. That's also negative feedback. Both trip and lac, negative feedback systems, even though lac is an inducible system and trip is a repressible system. The graph below shows the growth of an E. coli culture that's fed with both glucose and lactose. X and Y show the glucose and lactose concentrations. So note how the glucose concentration is going down over here, and notice that the lactose concentration maintains itself and then goes down over here. The red line shows the growth of the bacteria over 9 hours. There are 2 lags in growth. One is at B over here between A and C, and the other one is here at D. What's happening? E. coli prefers to metabolize glucose. Why? Because glucose is a monosaccharide, lactose is a disaccharide, and as you know, glucose is the fuel that goes right into the glycolysis process that begins cellular respiration. Now, up to point A, E. coli eats glucose and grows rapidly. But then the glucose starts to run out. As the glucose starts to run out, there's a lag in growth during activation of the lac operon and lactose digesting enzymes. From C to D, the lac operon is churning out those enzymes that break down lactose into glucose and galactose, but at a certain point, lactose runs out, and then there's another lag, which might be a permanent lag, until another food source is introduced into the culture. The key idea is that glucose is easier to digest than lactose. Glucose will be metabolized, it'll be digested first, followed by lactose. There was a graph like this that was an FRQ on one of the previous tests, and this is why you have to understand operons in order to succeed in AP biology. Regulation of gene expression part 2, eukaryotic gene regulation. I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. Gene regulation in multicellular eukaryotes, key issues. Organisms like you and me and lizards and redwood trees and jellyfish, any multicellular eukaryote is composed of trillions of cells organized into specialized tissues. We have 46 chromosomes, 3 billion base pairs in each haploid genome, and 20,000 genes. Gene regulation is a big and complex issue. Here are some more parts of that issue. Every single cell has the same DNA, but cells need to know which genes to express as they develop. And gene regulation, as we just saw with prokaryotes and operons, is also influenced by factors in the environment. How do genes get turned on and off? Note that most eukaryotic DNA is non-coding. So what's the difference between the coding DNA and the non-coding DNA? And genes contain introns. We've mentioned these before. Now we'll really look at them in depth. In eukaryotic cells, what determines which genes are expressed? Let's start with this fact. In any cell in a multicellular organism, most of the DNA is not expressed. You have cells that make up the lens of your eye. Those cells express a single protein. That means that 19,900 something other proteins are not being expressed. All those genes are turned off. Those genes that are turned off are tightly packaged around proteins that are called histones. That's what these discs over here represent. There's an additional chemical modification which is called methylation. It's the addition of a methyl group, that carbon attached to 3 hydrogens, and that prevents transcription. In the few genes within any cell that are turned on, there's a process called acetylation that loosens up the DNA, and that makes it possible for RNA polymerase to come in, find the promoter, and transcribe the genes. What is epigenetics? We just talked about how in most cells, most of the DNA is not transcribed. It's silenced, it's turned off. Only a small number of genes are turned on. What's the difference? That is all defined by this newly emerging topic that's called epigenetics. Epigenetics are changes in DNA expression that involve reversible chemical modifications of DNA or modifications in DNA packaging. Chemical modifications of DNA, methylation. Modification in DNA packaging, wrapping around these proteins that are called histones. But the genes themselves, the sequence of nucleotides, is not changed. It's a level above the genetic level. That's why it's called epigenetics. It's responsible for the differentiation of tissues during development. Why are skin cells expressing skin proteins, where fingernail cells are expressing fingernail proteins and muscle cells expressing muscle proteins? Those are all about epigenetics because all of those cells contain the same genes. Somewhat astonishingly, sometimes these changes can be transmitted from one generation to the next. That's a newly emerging field of study, and that's intergenerational transmission of epigenetic modifications of the genome. What's the connection between epigenetics, cell differentiation, and gene expression? The key idea, one that needs to be memorized, is that all cells in the same organism are genomically equivalent. Every cell in your body, except for your gametes, has the same DNA. All cells are descended from the zygote. That's shown at number 1 in this diagram. All cells have the same DNA. That's shown at 3 and 4. Cells differentiate because they express different genes, and that relates to the epigenetic modifications that we just talked about in the previous slide. Describe on a big picture level how transcription is regulated in eukaryotes and eukaryotic cells. Previously, we talked about operons, which is how genes can be turned on and off in response to environmental changes. Eukaryotes have to be able to do that too. Some of that relates to acetylation, methylation, histones, the things we've talked about, but some of this is on a more immediate regulatory level. So let's look at the regulatory processes that occur in eukaryotes. Eukaryotes possess regulatory DNA sequences that interact with regulatory proteins to control transcription. I know that this diagram looks horrifyingly complex, but you really only need to know it on a basic level so you can understand questions that might come your way on unit tests or on the AP Bio exam. Some of these regulatory sequences include promoters. We've talked about those in the context of transcription. So there are promoters shown at letter E. There are also enhancers that are shown at letter A, and what they do is they increase the probability that a gene will be transcribed. They enhance that possibility. Interactions between activator proteins, they're shown at B, DNA bending proteins, that's at F, mediator proteins at G, and general transcription factors, H, enable RNA polymerase, shown at letter I, to bind, making transcription possible. All you really need to know is that this kind of system is one that's used for eukaryotic gene regulation. You'd never be asked to differentiate between these mediator proteins at G and these general transcription factors. You just need to know the big picture. This is what eukaryotic gene regulation can look like. How can gene expression be coordinated in different body tissues? During development, as we've discussed, different tissues express different genes. But those different tissues can also share common regulatory sequences that enable the transcription of genes within those various tissues to be coordinated. An example of that is that the tissue in a male lion's neck skin, I'm talking about this over here, and their muscle tissue express different genes. One's expressing the hair that makes up the mane, and the other is expressing the tissue in the muscle. But both of those tissues share a common testosterone receptor gene. That testosterone receptor gene gets expressed as a cytoplasmic receptor, and therefore when testosterone gets released into the body, it binds with the testosterone receptor. This becomes a transcription factor that goes into the nucleus and activates genes. The genes that are activated are going to be different depending on whether those cells are in the lion's neck or in the lion's muscle tissue. But that leads a single hormone, in this case, to be able to induce changes in different tissues. It's coordination of gene expression in different body tissues. What are introns, exons? What's required to make translatable mRNA in eukaryotes? Introns are intervening sequences of DNA within genes. They are transcribed into pre-mRNA. Here's an intron in DNA. Here's an intron in pre-mRNA. Exons are DNA that becomes RNA that ultimately becomes mRNA that gets expressed into protein. It gets translated into protein. And that is just a bit of the processing of pre-mRNA that has to happen in eukaryotes. Here's the process of transcription, relatively straightforward. But then what we need to have happen is all of these introns need to be cut out, and then the mRNA needs some modification so that it can survive in the cytoplasm and be translated by a ribosome into protein. We'll see that in a couple of slides. Describe some of the post-transcriptional modification that has to happen to pre-mRNA in eukaryotes before it can be translated into protein. In eukaryotic cells, pre-mRNA is what's transcribed from a protein coding gene. So this is DNA over here. You can tell because it's a double helix. And this is pre-mRNA, right over here at number 2. Before it can be translated into protein, that pre-mRNA has to be processed in several ways. It has to get an addition at its 5' end of a GTP cap and a 3' poly A tail. Poly A tail, it just means it's adenine, 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 adenine all over again. As we've discussed previously, introns, those intervening sequences that don't code for protein, need to be excised. They need to be cut out. And then the fragments that consist of the exons need to be spliced together. Then you wind up with mRNA that can be translated into protein. What is the function of the 5' GTP cap and the 3' poly A tail that's added to mRNA during eukaryotic RNA processing? That 5' GTP cap, which is shown over here at G, protects the mRNA from breakdown by enzymes in the cytoplasm. And it also assists the mRNA in leaving the nucleus and binding with a ribosome. The 3' poly A tail, shown over here, that makes the mRNA more stable, and it delays its enzymatic breakdown by enzymes that are in the cytoplasm. Explain how the organization of eukaryotic genetic material into introns and exons can increase phenotypic variation. As we've discussed before, exons are expressed sequences. They're translated into amino acid sequences. Introns are intervening sequences that are spliced out of mRNA before translation. Here we have DNA, and here we have pre-mRNA. And what we've got to do is we've got to cut out these introns. But in eukaryotes, there's a process called alternative splicing. Through alternative splicing, exons can be spliced together in alternative ways, allowing for the production of multiple protein versions from the same pre-mRNA transcript. So for example, in this mRNA and in this protein, what we've done is we've dropped out a couple of the exons. There's exon 1, 3, 4, and 6. Here's another version of the same protein, exon 1, 2, 5, and 6. And here's yet another one, exon 1, 2, 4, 5, and 6. The basic idea is that each of these exons codes for what's called a functional domain, a piece of the protein that can actually do something. You put those functional domains together and you get proteins with slightly different functions. They're all within the same close family. They're all from the same gene, but they're different manifestations of those genes, and they provide for additional phenotypic variation that's found in eukaryotes, but it doesn't happen in prokaryotes who don't have the intron-exon organization of their coding genes. Explain the role of small RNAs in eukaryotic gene regulation. Small RNAs are exactly what they sound like. They're segments of RNA that don't consist of a huge number of nucleotides, yet they play important regulatory roles in the cell. One of these is microRNAs. MicroRNAs are particularly small, and they play a role in what's called post-transcriptional control of gene expression. That's exactly what it sounds like. It's after transcription. A key process that microRNAs are involved in is called RNA silencing. Here's how it works. Here's DNA. That DNA will contain a gene that codes for a microRNA. Not all genes code for proteins. Some of them just code for RNAs. In the same way as pre-mRNA needs to be processed before it matures, there's processing of the pre-microRNA to make it into mature microRNA. In the same way as ribosomal RNA will connect with protein, this microRNA will connect with a protein that's called an RNA silencing complex protein. Together, that RNA plus the RNA silencing complex protein will do one of two things. If the microRNA completely matches 100% part of a sequence within an mRNA, then that complex will cause the mRNA to be degraded and destroyed. If, on the other hand, there's a partial match, then this complex will cause a pause in translation. In either case, what have we done? We've changed expression of a gene through microRNA. Topic 6.7, Part 1, mutation. What is a mutation? What's a point mutation? A mutation is a random change in DNA or an entire chromosome. A point mutation is a change in a single nucleotide. You see that here with the nucleotide C mutating to the nucleotide T. Distinguish between silent mutations, nonsense mutations, and missense mutations. Silent mutations are mutations that result in the same amino acid being coded for. The DNA changes, but the amino acid in the protein doesn't. Why? Because the genetic code is redundant, with many codons coding for the same amino acid. A nonsense mutation is a mutation that inserts a stop codon instead of an amino acid. And a missense mutation changes the amino acid from one to another. We see a silent mutation over here. The original DNA is lysine. The DNA that's now being coded for, despite this mutation, is lysine. Here's a nonsense mutation where instead of lysine, we have a stop codon. And here we have missense mutations. One is coding for arginine instead of lysine, and one is coding for threonine. The effect of a missense mutation depends on the chemistry of the substitution. This isn't a term that you need to know, but a conservative missense mutation is one where the chemistry is of less significance. So lysine is a basic amino acid. It has this amino group over here at the end, and so does arginine. So that might not change the structure of the protein very much. It might not change it in a functional way that is really observable in a phenotype. It's not clear, but it might not be a big deal. On the other hand, substituting threonine, which is a non-polar amino acid, for lysine would be a big deal. This is non-polar, this is basic. That's a very significant change in chemistry, and that will impact the function of the protein. What are frameshift mutations? To review this concept, I put together a sentence composed of 3-letter words. There are no spaces, but we have these little dividers here. If we were to do a mutation where we did a substitution of E for A over here, then note that most of the words still make sense. We have the ket. What's a ket? Well, you could probably surmise that that was intended to be cat and that it's just a typo. But if we deleted one of the letters, like hitting the delete key on your keyboard, then what we get is if we drop this A over here, then essentially we've significantly changed the meaning. We have one word that makes sense insofar as it's a word, but it doesn't make sense in the context over here. That is called a frameshift mutation because codons are read in groups of 3. If you delete or insert, you change the reading frame. Now let's look at some nucleotides. Here is a series of codons that code for 4 amino acids and then a stop codon. And note that this example shows RNA to show the consequences. The mutation would have been in the DNA. If we have a frameshift mutation, then what we've done is we've done a deletion or an insertion that changes the reading frame just like we did over here. And that will cause extensive missense or nonsense. Deleting this U over here causes these 2 amino acids to be wrong. That's missense. Or what can happen is you can have the insertion of a stop codon, and then the entire protein doesn't get coded for after this first amino acid. That is the impact of a frameshift mutation. Sickle cell disease is caused by a single substitution mutation. Explain how one such substitution can cause sickle cell disease. Sickle cell disease is one of the first genetic diseases that was understood well on a molecular level. It's important to know about. The disease involves changes in the protein hemoglobin. Hemoglobin, shown over here, is a quaternary protein that carries oxygen in red blood cells. So here's hemoglobin in red blood cells, and here the red blood cells are carrying oxygen, delivering it to the tissues of the body. The mutation that causes sickle cell disease is a missense mutation. That causes valine, which is non-polar, to substitute for glutamic acid. That causes a significant change in the chemistry of hemoglobin. It causes hemoglobin molecules to stick together. So notice how over here they're separate within the red blood cell, but the mutated form, they'll form very weak bonds, and that happens under low oxygen conditions. That just means when you exercise or walk up a flight of stairs, anything like that, that causes the cells to become sickled or spiked, and they get stuck in red blood cells, and that causes extensive tissue damage. This is a recessive mutation. You need to be homozygous in order to express the phenotype. Though there is a phenotype that is caused by being a heterozygote, that's called sickle cell trait. In any case, this is how a single substitution mutation can be responsible for a significant genetic disease. Mutations can be positive, negative, or neutral. Explain. The big idea is that a mutation's effect always depends on the environment. It's contextual. A positive mutation improves a phenotype in a way that increases evolutionary fitness. Fitness is about survival and reproduction. If it increases both of those things, then it's a positive mutation. Here's an example. This is a kind of fish that's called a 3-spined stickleback. There are populations that live in oceans. There are populations that live in freshwater. Note this pelvic spine over here. It's an adaptation that promotes survival in marine environments because it protects against certain kinds of predators. There are populations of sticklebacks that became stranded in freshwater lakes. In those populations, the predators were absent. There was a mutation that emerged that resulted in the loss of that pelvic spine. Not only because it doesn't make sense to produce a structure that has no survival benefit, but there are predators that actually can prey on sticklebacks with the pelvic spine. So losing the pelvic spine, positive mutation. We just talked about sickle cell anemia. That's, in most cases, a mutation that reduces fitness. Why? Because it causes diseased red blood cells and it causes tissue damage, as we just explained. However, in the context in which the mutation for sickle cell anemia evolved, in some ways it's a positive mutation. Why? Because having one dose of the sickle cell allele, in other words, being a heterozygote, gives you resistance to malaria. So that makes it a positive mutation in a malaria-ridden area. In that context, it's positive. A neutral mutation has no effect on the phenotype, and that's because it might happen in non-coding or non-regulatory DNA, or it might result in a silent mutation where the amino acid doesn't change. How are mutations important to evolution? Mutations provide the raw material upon which natural selection acts. Note that I'm using the same illustration as in the previous slide, and I'm doing that on purpose. Mutation makes evolution a creative process that results in adaptation. Without mutation, natural selection could only cull harmful variants from a population. But with mutation, new variants can arise that increase a population's fitness. What's the difference between germline and somatic mutations? Germline mutations are mutations in the cells that make gametes and all other cells. Here's sperm, here's an egg. If there were a mutation in either one, then that mutation would be present in every cell in the embryo. That means it would be present in every cell in the organism, and it would be present in the gametes that that organism produced. Germline mutations can be inherited, they're subject to natural selection, and what's an example? Any inherited genetic disease, such as sickle cell anemia, which we have discussed, or every adaptation. A somatic mutation, it emerges in some tissue during the course of development or during the course of adult life. It only affects the organism, it's not passed on to the future. And an example are the somatic mutations that can cause cancer. There are some inherited cancer mutations, but you can also have a mutation that's caused by some environmental exposure, ultraviolet radiation, for example, that can cause skin cancer, and that becomes a somatic mutation that can cause a cancer. Are you asking yourself, how am I going to get a 4 or a 5 on the AP Bio exam? It's a good question because it's a hard test, but we have a plan for your success. Go to learn-biology.com, sign up for a free trial, and complete our interactive tutorials and interactive AP Bio exam reviews. We guarantee you a 4 or a 5 on the AP Bio exam. See you on learn-biology.com. Topic 6.7, Part 2, Horizontal Gene Transfer. Contrast horizontal gene transfer with vertical gene transfer. In vertical gene transfer, parents transmit all or half of their genome to their offspring. That's what's happening here. Here's a bacterium, it's reproducing, and in this generation, all of the genes have been transmitted, and in the next generation, all of the genes have been transmitted. You inherited your genes from your parents through vertical gene transfer. Horizontal gene transfer is quite different. In horizontal gene transfer, one organism transfers genes to another organism that is not its offspring. That's what we see happening down here. This bacterium over here is transferring genes on this loop of DNA to this second bacterium. In unicellular recipients, as is shown here, the newly acquired genes become part of the recipient's genome, and when this bacterium reproduces, it'll pass on the newly acquired genes to its offspring. In a multicellular recipient, there's only long-lasting results, intergenerational results, if the genes are transferred into the germline. Describe bacterial conjugation. If you want to build your vocabulary or use your vocabulary to enhance your understanding of biology, conjugation is another word for sex. Here's how it works. It's unlike any kind of sex that human beings or other animals have. Bacteria have, in addition to their main chromosome over here, they have a loop of DNA that's called a plasmid. Plasmids can express genes for a membrane extension that's called a pilus, that's shown over here at C. When the pilus contacts a second cell, the plasmid can be copied and transmitted to that recipient. And the recipient now has all of the genes that are on the plasmid. We have horizontal gene transfer. Conjugation plays a key role in the spread of antibiotic-resistant genes through bacterial populations. Describe bacterial transformation. In bacterial transformation, bacteria pick up DNA fragments, shown over here at 1, from the environment. And those DNA fragments enter into the cell and then become incorporated into the genome. That's what's shown over here. This DNA can include plasmids. So here's a circularized piece of DNA, a plasmid that's being incorporated into the cell. In genetic engineering, transformation using plasmids is used to introduce foreign genes, including human genes, into bacterial cells. Describe how horizontal gene transfer can occur through viral transduction. Transduction is a kind of horizontal gene transfer that occurs through viruses. It occurs through mistakes in the viral replication cycle. During viral infections, the virus breaks apart the host's genome. Here's a virus, it's injecting its DNA into its victim, this cell over here, and one of the first things that happens is that the cell that was infected has its DNA broken apart. What the virus then does is it uses the cell's molecular machinery to create new viral genes and to produce new viral proteins, and those become assembled into new viral particles. But during a mistake in the process, sometimes DNA fragments from the host over here are mistakenly incorporated into a virus. So this virus over here is leaving the cell carrying genes from the host. It should just be carrying its own genes. When that virus infects a cell in another organism, another bacterial cell in this case, it can bring in the other organism's DNA. So here's the DNA from the original host, and notice that DNA is being injected into this new host. That DNA will recombine with the DNA that's in the new host, and this can happen to animals as well. And if the virus infects a germline cell, then new genes can be incorporated into the gene pool of the recipient. What is viral recombination? This is a kind of horizontal gene transfer that occurs within viruses. In this case, two different viruses, different strains of different viruses, infect the same host. So this virus and this virus, they're variants of one another. And here you see them infecting a new host. And here you see both of these viruses inside a cell from the host. As they carry out their replication cycle, there's DNA from the host, there's DNA from the viruses, and the DNA from the viruses can get mixed up. So the genes of the viruses can recombine, and the result is instant emergence of new viral strains. Sometimes, if animal immune systems can't recognize the new strain, this can lead to pandemic viral outbreaks. This is what happens when every couple of years there's a strain of the flu that's new and that's novel and which infects many people, sometimes with disastrous results. This kind of viral recombination is what causes it. Topic 6.8, biotechnology. Explain what recombinant DNA is and how it can be artificially created. Recombinant DNA is DNA that's been combined from more than one source. During meiosis, you would create recombinant DNA as you combined the DNA that you inherited from your parents in the form of new gametes. But this is artificial recombinant DNA. It's DNA from more than one source, shown over here, that might have bacterial DNA with a snippet of human DNA with more bacterial DNA. So DNA artificially combined, more than one source, it's been recombined. The main tool in creating recombinant DNA is something called a restriction enzyme, shown over here at letter C. What these restriction enzymes do is they find sequences of DNA that are called restriction sites. So here's one at B, and they cut the DNA, here's the DNA, with sticky ends. And you can see these sticky ends over here. Those sticky ends are exposed single strands of nucleotides. You can see that over here in this diagram that shows the DNA double helix. And those single strands are able to form hydrogen bonds with complementary bases. The result of using a restriction enzyme, shown over here, are restriction fragments. So here's a fragment over here, here's another fragment over here. If you cut a second piece of DNA, for example, a piece of human DNA with the same restriction enzymes, what you'll wind up having are complementary sticky ends. Because of complementarity, the ends of the two pieces will form hydrogen bonds. That's what you see happening over here. And then you need to use another enzyme, it's called DNA ligase, and that creates sugar phosphate bonds connecting the strands, creating recombinant DNA. Using restriction enzymes and DNA ligase, you can create a recombinant plasmid with a human gene. Explain how. Note, for this question, assume that introns have already been removed from the human DNA. The first step is to extract a plasmid from a bacterial cell and then cut open that plasmid with restriction enzyme, leaving sticky ends the way that we just described in the previous slide. Use the same restriction enzyme to cut out a target human gene, and therefore the ends will be complementary. Because they've been cut with the same restriction enzymes, the human gene will combine with the plasmid, forming hydrogen bonds between their complementary sticky ends. Then you have to use DNA ligase, we referred to that in the previous slide, it's not shown here, to bind the human DNA and plasmid DNA together, creating a recombinant plasmid that contains a human gene. Then you'd insert the plasmid into a bacterial cell. That's using the technique of transformation, which we previously referred to when we talked about horizontal gene transfer. This genetically engineered recombinant bacteria, this over here and its descendants over here, will produce the human protein and produce the plasmid in every reproduction cycle. This is how genetically engineered insulin has been created. That means bacterial cells that produce a human protein that's widely used. In order for the genes for human proteins, such as insulin, to be expressed in bacteria, introns need to be removed. Explain why and how. To review, introns are non-coding sequences of DNA within eukaryotic genes that have to be spliced out before the gene's RNA can be translated into protein. Here's human DNA. There are exons that are expressed sequences, and they're separated from one another by introns, these intervening sequences. The consequence of the presence of introns is that to transfer a human gene to a bacterium to create a gene product, you have to use DNA from which the introns had been removed. The bacteria would just translate everything, including the introns, and that would lead to a non-functional protein. How do you remove the introns? You have to do it before transforming the bacterial cells, and you can do it in two ways. The first method involves determining the amino acid sequence for the protein. Biochemists can look at a protein like insulin and figure out what the linear sequence of amino acids is. Once you do that, you use your genetic code chart to reverse engineer DNA that codes for that amino acid sequence. Another method is shown here. You find cells that produce the desired protein, you extract mRNA from those cells that codes for this protein. That mRNA already has had its introns removed, and then you use the enzyme reverse transcriptase. That's an enzyme that's in retroviruses, which are viruses that are RNA-based but can create a DNA copy of themselves that gets incorporated into the human cell that they've infected. HIV is an example of one such virus. You use reverse transcriptase, which is shown here at B, to create cDNA, complementary DNA from the RNA, and then you insert that complementary DNA into the plasmid, and that's how you do your successful genetic engineering. What is gel electrophoresis? How is it used to analyze DNA? Gel electrophoresis is a technique that's widely used. It's used to sort molecules by size and or electrical charge. It's the basis of a technique that's called restriction fragment analysis, also called DNA fingerprinting, widely used in forensics. It involves placing molecules in a porous gel. Here's the gel over here at number 5. That is in a device, an apparatus, a box, that can produce an electrical current. So you'd run an electrical current generated over here through the gel. Because DNA's phosphate groups, shown over here, are negatively charged, DNA fragments will move away from the negatively charged side of the electrophoresis chamber. So you put the DNA over here, like repels like, negative charge, and that's going to push the DNA in this direction. The small fragments will be impeded by the gel less than the large fragments. So over time, the smaller fragments will move more than the larger fragments, enabling the fragments to be sorted by size. By the end of the process, you'd have here DNA with one large fragment, here DNA with 2 fragments, and here DNA that's been cut into 3 fragments. How would it be cut? By restriction enzymes. So you use a combination of these techniques to get results like this as you're analyzing DNA. Material related to biotechnology often shows up on the AP Bio exam in this form. Here's a simple restriction mapping problem. A 20 kilobase plasmid, KB kilobase, has several restriction sites. The image on the right shows the results of electrophoresis following various combinations of restriction enzymes. Which lane shows the gel that would result if the plasmid were digested with the restriction enzyme BAMH1? Your success in AP Biology starts here. Are you struggling with AP Bio? With Learn-Biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to Learn-Biology.com to find out how you can master your biology course and crush the AP Bio exam. This line over here indicates a restriction site that's been labeled with BAMH1. So there's a restriction site over here for BAMH1, a second one over here, and a third. The entire plasmid is 20 kilobases, and the map is telling you that this is a 3 kilobase difference. So if you cut the plasmid with BAMH1, you'd wind up with 3 fragments. Here's one. Here's a second one, it would start here, go all the way down to here, and here's a third one that would start here, go all the way up to here. The first one would be 3 kilobases in size. The second one would be 11 kilobases in size. How did I do that? 3 kilobases plus 8 equals 11, and the last one is 6 kilobases in size. And what that means is that you'd have to look over here at the gel, and you'd see, oh, this fragment is 11 kilobases, this fragment is 6 kilobases, and this one is 3. Perfect. That means B would be your answer if this were on a multiple choice test. What is PCR? What is it used for? PCR stands for polymerase chain reaction. The polymerase is DNA polymerase. It's a cell-free technique for cloning DNA. In other words, you can clone DNA in a test tube. You don't need a cell in order to do it. It requires the DNA sample that you want to clone. It's shown over here at A. It requires primers. Those are short strands of single-stranded DNA that bind to sequences at the start of the DNA that you want to amplify. So here is a primer, and here you see the primer binding to the target DNA. It requires heat-resistant DNA polymerase, shown over here at G. That would be a large protein. Why does it need to be heat resistant? Because the process involves repeated cycles of heating and cooling, and you need a DNA polymerase that won't be denatured by the heating process. Where do you find it? From bacteria and archaea that live in hot springs. And you also need free nucleotides that are going to be used for DNA synthesis, because what we're doing is we're making lots of DNA from a sample that we want to amplify. How does it work? It involves repeated cycles of heating the DNA to separate it into different strands. So here's the DNA, you heat it, you break those hydrogen bonds, and now you've separated it into single strands. That's step 1 over here. Then you cool the DNA enough so that primers can bind to it, and so that DNA polymerase can synthesize new DNA. That's shown at 2 and it's shown at 3. The DNA polymerase will read the template strand, and it'll seal sugar phosphate bonds between the nucleotides that bind with the template strands. So here you are, you're seeing DNA polymerase creating new DNA. And every heating and cooling cycle will double the amount of DNA. So we started with one piece of DNA. Now we have two pieces of DNA. That's an exact copy of the original DNA. Do it again. You have 4 pieces of DNA. Do it again and you'll have 8 pieces of DNA. After 10 cycles, you have 1,000 times more DNA than you started with. And after 30 cycles, you've amplified your DNA 1 billion-fold. This is widely used in any kind of science that needs to work with DNA. It's widely used in forensics, where little DNA samples from a crime scene, for example, are amplified so that they can be analyzed for electrophoresis, DNA fingerprinting, etc. What is DNA sequencing? What are some of its uses? DNA sequencing, you just need to know what it is. You don't really need to know how it's done. Though if you're interested in seeing how it's done, you can do that at learn-biology.com. DNA sequencing involves taking a sample of DNA, anything from a small fragment to the entire genome of an organism, and figuring out the specific sequence of A, T, C, and G nucleotides that make it up. It allows biologists to determine what proteins an organism can produce. It's used to infer evolutionary relationships, and it's used by cancer biologists to sequence tumors to see what genetic mutations are causing the cells to become cancerous. During the COVID-19 pandemic, sequencing was used to analyze the emergence of new SARS-CoV-2 variants, and of course it was used in order to create the vaccine. In forensics, sequencing is being used along with DNA fingerprinting to identify and exonerate suspects and resolve paternity disputes. Welcome to AP Bio Unit 7, Evolution. To help you study, I've put together a checklist that you can download at apbiosuccess.com/checklist. Topics 7.1 to 7.3, Natural, Artificial, and Sexual Selection. In terms of understanding the process of evolution, the most important idea to understand is natural selection, developed by Charles Darwin in the 1800s. Darwin himself built the idea of natural selection upon the foundation of artificial selection, so that's where we're going to start. Artificial selection is also known as selective breeding, and in this process, breeders envision a desired trait in an animal or a plant. For many generations, the breeders select organisms with that trait, and the process over time, over many generations, creates a carefully guarded gene pool with individuals that only have genes for the desired trait. Here are 2 examples. The plants that are in the Brassica oleracea family include cauliflower, broccoli, Brussels sprouts, etc. They're all the same species. The ancestral plant, Brassica oleracea, has been bred for specific traits, for flower clusters in terms of cauliflower, for flower buds in terms of broccoli, for lateral buds in terms of Brussels sprouts, for kale in terms of leaves. But all of these are varieties of the same species that have been bred for specific traits. The same is also true of dogs. Dogs and wolves are all members of the same species, Canis lupus, and all the breeds, varieties of dogs, the German Shepherds and the Schnauzers and the Dachshunds, those are all varieties that have been bred for specific human purposes, for protection, for hunting, or for just looking cute and being really cuddly. So how does natural selection work? In any population, there's variation. The kind that matters is inherited variation that's in the genes, and that comes about through recombination and mutation. In any population, the reproduction rate, the number of individuals who are born, exceeds the survival rate, and that's even true of slow breeding populations. Sometimes that's summarized by the phrase, many are born but few survive. The survivors have a beneficial trait that gave them some type of advantage. As the generations proceed, mutation continues to create new variants. And if you repeat steps 1 through 4, you wind up with adaptation. Adaptations can include things like the incredible structure of the wing of a bat, or the incredible behavioral adaptation, which is sonar, which is of course combined with structures like the ears of a bat, which can hear high-frequency sounds. They can be camouflage mutations, like this satanic leaf gecko, or they can be on a molecular level, like the way that enzymes fit with substrates. You've learned a lot of biology, and whenever you see things and you find yourself thinking, how could that work? How could that be so perfectly put together? It was put together by natural selection. In addition to artificial selection and natural selection, a third type of selection to know about is sexual selection. Sexual selection is selection for traits that directly increase reproductive success. They create an evolutionary dynamic that results in sexual dimorphism, where there are different phenotypes in males and females. One type to know about is called intersexual selection, and that's where members of one sex, typically the females, choose mates of the other sex that are typically males. So here's a photograph that I took relatively near to my home. This is a female turkey. There's a bunch of male turkeys who are displaying to her, and they're essentially saying through their movements and through their extension of their tail feathers, I'm really attractive, you should mate with me. And that means that there's been selection for these tail feathers and for features like these wattles and for the male's behavior. Peacocks are a bird species where that's been carried to extraordinary lengths, and that typically leads to colorful and highly adorned males and choosy females. The other kind of selection is called intrasexual selection, and that's where competition between males for control of a harem of females or control of a breeding territory leads to a dynamic where the males are aggressive and often really big and strong. And that's what we see over here with these elephant seals that live off the coast of Northern California. And they're about twice the size of females. They're huge, aggressive, testosterone-filled animals. Selection can change the distribution of phenotypes in a population in a variety of ways. In any population, most characteristics can be represented by a bell curve of continuous variation. If you took all the students in your school and you organized them by height, most of the people are average height. There are a few people who are short and a few people who are tall. That's a bell curve of distribution. Directional selection, which is shown over here, selects against one of these extremes. In other words, here's the original population, you're selecting against this extreme over here, and so you're pushing the population in this direction. In stabilizing selection, you are selecting against the two extremes. An example of stabilizing selection is birth weight in babies. Babies that are too small have lower survival. Babies that are too big have lower survival. The babies who survive most are the ones who are of average size. The third type is called disruptive selection. If you can imagine a scenario where the average phenotype is maladaptive, then there'd be selection for the two extremes. It would kind of split the population into two. An important type of directional selection to know about is adaptive melanism. Adaptive melanism is the darkening of the body within a population in response to the darkening of the environment. It occurs within a population as its gene pool evolves in response to natural selection. So it's not like getting a tan, which happens in an individual. This is something that happens over evolutionary time as genes for light coloration are selected against and genes for darker coloration are selected for. The selective pressure that results in adaptive melanism is typically predation. What's shown here in this example is the rock pocket mouse, which lives in the southwest of the United States. And what's been documented on a genetic level is that populations of these mice that live on a dark colored substrate have evolved a mutation that results in more melanin production, and that mutation spreads throughout the population and results in subpopulations that have the dark coloration. We've been talking about various types of selection. Natural selection is often summarized as survival of the fittest. What does it mean to be fit? How is evolutionary fitness measured? Evolutionary fitness is not about strength or speed. It's the number of offspring and offspring of offspring that survive to reproduce. And the reason why I have this montage of penguin life is just to indicate that it is something that emerges at every level of the life cycle. When the penguins are feeding, when they're doing their march inland, when they're breeding, when they're juveniles huddling together. There's fitness at every single stage, and fitness throughout the life cycle is what enables genes to be passed on to the next generation. To end this discussion of all types of selection, we're going to look at this question: explain how the peppered moth demonstrates an evolutionary change in response to environmental change. The key point about the peppered moth is that it provides science with a directly observed example of directional selection and adaptive melanism in response to an observed change in the environment. The peppered moth comes in two forms. There's a peppered form, mostly light colored, and a dark form. And before the Industrial Revolution began with the invention of the steam engine, the burning of coal. The predominant phenotype was peppered, was this one over here. So in any population of moths, almost all of them were peppered. There'd be a few dark moths. Why is this? Because that was a form of adaptive coloration. The moths were camouflaged as they rested on light colored tree trunks, which were often covered by these whitish colored lichens. That's the situation before the Industrial Revolution. With the Industrial Revolution, soot from factories was put into the air, it landed on the trees, it killed the lichens and darkened the tree trunks. And what that did is that it created a selective advantage for the dark-colored moths. If you were to do a census of the moths at the beginning of Industrial Revolution, most of them were white, only a few were black. But as the Industrial Revolution proceeded, the mean phenotype shifted from light to dark until in the depths of the Industrial Revolution, the majority of the moths were dark colored. The interesting thing is that this process was also historically reversed. About 1960, soot from pollution declined with the development of laws like the Clean Air Act. That happened around the world. The lichens returned, the trunks became lighter, the mean phenotype of the peppered moth shifted from dark colored to light colored. I have a song about the peppered moth, and there's also fascinating stuff related to the history of this because it was embroiled in some controversy, but the work of Michael Majerus, which was done in the early 2000s, which replicated some work done by B.D. Kettlewell, which was done in the late 1800s or early 1900s, established this as a clearly observed case of adaptive melanism, phenotypic shift in response to an environmental change. Your success in AP Biology starts here. Are you struggling with AP Bio? With Learn-Biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to Learn-Biology.com to find out how you can master your biology course and crush the AP Bio exam. Topic 7.4 to 7.5, Population Genetics and Hardy-Weinberg. What is population genetics? Population genetics is a study of how genes are distributed in populations and how they change over time. The key thing that we measure in population genetics is allele frequency. In this population over here, we have 20 individuals and we're tracking the frequency of the allele big A and little a, and we can see that the frequency of little a is 0.5. It makes up 50% of the alleles in this gene pool. What is a gene pool? It is simply all of the alleles of all of the genes in a population. And in that context, evolution means a change in the genetic makeup of a population over time. It's the change of allele frequencies within a gene pool. Before we get more into the details of population genetics, I want to address the biggest misconception I've seen among my students related to population genetics. That's the wrong idea, that's why it's crossed out, that the dominant allele has to be more common than the recessive allele. These are different dimensions of biological analysis. And allele frequency, whether an allele is common or rare, has nothing to do on dominance or recessiveness. It's based on the advantage or harm an allele confers to its associated phenotype or random historical factors. And here's an example. The allele for the condition called achondroplasia is exceedingly rare, but it's a dominant allele. Population genetics is one of the more mathematical parts of the AP curriculum, and it's built around two equations. P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. In this system, P represents the frequency of the dominant allele, and Q represents the frequency of the recessive allele. If you load P plus Q into a Punnett square, the whole thing makes a lot more sense. P plus Q equals 1 means that for a gene with two alleles, the frequency of the dominant allele and the frequency of the recessive allele equals all the alleles. P plus Q equals 1. P squared plus 2PQ plus Q squared equals 1. What does that mean? Well, first of all, here's P and here's P. If you do the Punnett square kind of thing, this would be P squared. The frequency of homozygous dominance, P squared, plus the frequency of heterozygotes, PQ plus PQ equals 2PQ, plus the frequency of recessive individuals, Q squared, is all the individuals in a population, and you can see that right here. Here's how you might see this in a sample problem. You've identified, you've been told that 49% of the mice in a population have the recessive trait of white fur. Figure out the frequency of PQ, P squared, 2PQ, and Q squared. So here's our original setup. What we're going to do is Q squared is this value over here. So we're just going to put 0.49 over here. From there, what we're going to do is we're going to work to figure out that the square root of 0.49 is 0.7. Then we know that this plus this equals 1. So therefore, this, the value P, has to be 0.3. Then we know that 2PQ has to be 0.42. Why? 0.3 times 0.7 is 0.21 times 2 is 0.42, and P squared is 0.09. 0.3 times 0.3 is 0.09. If you set up Punnett square cross multiplication tables, you'll get this right every time. The Hardy-Weinberg principle is that the frequencies of alleles in gene pools will stay constant unless one of the more following conditions is not met. This Hardy-Weinberg population is a fictional population that's used to understand what causes evolutionary change in real populations. The first is that the population is infinitely large. There are no harmful or beneficial alleles. There's random mating, there's no emigration or immigration, and there's no net mutation of one allele to another. If any of these 5 conditions is violated, then allele frequencies can change. And these are the factors that can cause evolution. What are these factors? Small populations leading to a phenomenon called genetic drift. Natural selection, where certain alleles are beneficial and others are harmful. Sexual selection, which is the opposite of random mating. Gene flow, where genes move into a population or out of a population. And directional mutation, where one allele has a tendency to mutate into another allele. So what's genetic drift? And we'll also look at one variant of genetic drift called a population bottleneck. Genetic drift is random change in allele frequencies in a population's gene pool, usually associated with small population size. A population bottleneck can cause genetic drift. A biotic, living, or abiotic, non-living factor wipes out a large percentage of the individuals in a population, leaving only a few survivors. The survivors' alleles might not be representative of the allele frequencies in the former larger population. And some alleles might disappear completely. The reason why I have a picture of a cheetah here is that cheetahs are extraordinarily genetically uniform. It's as if all cheetahs are practically identical twins, or at least very close siblings. Based on genetic analysis, it looks like the cheetahs underwent some enormous population bottleneck, maybe a viral infection or something like that, that wiped out almost all of the cheetahs down to only a few individuals. They repopulated from those few individuals, but as a result, many alleles were lost, and they're highly genetically uniform. Note that in a population bottleneck, the survivors didn't have any advantage. It's not natural selection. They were just lucky. Another form of genetic drift is the founder effect. And this is where A small number of individuals from a large population founds a new population, but because of insufficient sampling, because it's only a few individuals who leave the larger population, the allele frequencies of the gene pool of the founders who found the new population might be different from those in their parent population. In this graphic over here, here's the original population, and the squares and circles represent different alleles. The founders go off to colonize 3 islands. In this situation over here, the blue allele is completely lost, and now the red allele is at 100%. Here there's a representation of the diversity of the former population, and in this case, the red allele is lost, and the frequency of the blue allele is now at 100%. What is gene flow, and what's its effect on evolving gene pools? Gene flow is the movement of alleles from one population to another, and that can involve the movement of individuals, like we see here. This beetle is moving from population 2 to population 1, but it can also involve the movement of gametes, and that's easier to imagine in the case of plants, where pollen is flying from one field to another. What's the effect of gene flow? It can change allele frequencies, especially in the recipient population, and it can diminish differences between adjacent populations. Mutation is one of the conditions that violates the Hardy-Weinberg equilibrium. So what's its importance to evolution? Mutation is the ultimate source of genetic variation within and between populations. And if it's directional, if allele big A tends to mutate to little a, then it can change allele frequencies within a population. Here's a population genetics illustrative example. Sickle cell disease is caused by a recessive allele in the gene for hemoglobin. It can significantly decrease the quality of life and lifespan, yet the allele is in high frequency in certain populations. Explain. Sickle cell disease is caused by a point mutation in the gene for hemoglobin. It results in the substitution of valine, a non-polar amino acid, for glutamic acid, an acidic amino acid. It occurs only in children who are recessive for the allele, but heterozygotes can experience a small amount of sickling, not enough to generate pain crises and tissue damage under most circumstances. The red blood cells in heterozygotes, despite the fact that they don't normally sickle, are hostile environments for the Plasmodium parasite that causes malaria. This is a parasite that's carried by mosquitoes and then lives inside red blood cells. So what we have is a phenomenon that's called heterozygote advantage, where big S, homozygous dominant, is selected against. Why? Because of malaria. Big S, little s, heterozygosity, is selected for. Why? Because it doesn't have the symptoms of sickle cell anemia, but it provides protection against the malaria parasite. And small s is selected against because you have sickle cell disease. What this map shows is the high correlation between the frequency of the little s allele and the intensity of malaria in various parts of Africa. And this same model, heterozygote advantage, might apply to genetic diseases like cystic fibrosis and Tay-Sachs. I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. Topic 7.6 to 7.8, Evidence of Evolution. There is abundant evidence to support the theory of evolution. For AP Bio, the most important type of evidence to know about is the idea of homologous traits, homologous features. Homologous traits are traits that share a common underlying structure and a common embryological origin. They show descent with modification from a common ancestor. Here's the forelimb of a human, dog, bird, and whale. The bones that make up their forelimbs have been given the same name by anatomists who recognize that they were the same bones. You can also see that they develop from the same piece of tissue embryologically. It's because these structures evolved from a common ancestor who had these structures, and then natural selection, evolution, led these structures to evolve differently in different lineages. Homologous features result from an evolutionary pattern that's called adaptive radiation. Adaptive radiation occurs when one parent species produces several descendants, each of which has unique adaptations and fills a different ecological niche. These are some of the finches that live on the Galapagos Islands. They are all the descendants of an ancestral finch that was blown over there from Ecuador, a small population of finches, and that evolved in different directions. What's the connection? These are descendants of the same species, and their beaks are actually homologous features. They descended from a common ancestor. They became modified in each of the descendant species in different ways. A vestigial structure is a special kind of homology that provides further evidence for evolution. A vestigial structure has no apparent function, but it was inherited from an ancestor from whom that structure had a function. For example, whales have no hind limbs, but they have a pelvis over here onto which to attach those limbs. Why do they have that? Their ancestors possessed hind limbs, which were lost as whales adapted to their aquatic lifestyle. We have a coccyx, a tailbone. This is homologous to the tails of cats and dogs and other vertebrates, other mammals. We lost it in the course of our primate evolution into one of the great apes. What can be confusing when you're looking at common features to draw evolutionary connections are the difference between homology and analogy. Analogous features have a similar function, but when you look deeply, there's a different underlying structure. They arise through a different kind of evolution, not adaptive radiation, but convergent evolution. And an example is shown here. We have sharks, we have ichthyosaurs, an extinct kind of reptile, and we have dolphins. They all have a hydrodynamic form, and that's a convergent solution to the challenge of swimming. Here's a tricky question. Are the wings of birds and bats analogous or homologous? Think about the answer, then see mine. The wings of birds and bats are a convergent solution to the challenge of flying. They did not evolve from a common ancestor. Bats evolved from some ancestral mammal that was probably like a rodent, and birds are dinosaurs, and they evolved from small land-living dinosaurs. The function of the bird wing, the function of the bat wing is similar, but the underlying structure, the organization of the bones is different. The wings are an analogous structure. But if you look at this question a little bit differently, you get a different answer. The forelimbs of birds and bats are homologous. They're the same bones inherited from a common ancestor. So another example of why on the AP Bio exam or in your AP Bio course, you need to carefully read the prompt and think before you answer. What are molecular homologies? How do they serve as evidence for evolution? Molecular homologies are homologous features, but at the molecular level. They're molecules that by their structure and monomer sequence indicate common ancestry. All vertebrates, for example, have hemoglobin that has the same structure, 2 alpha chains, 2 beta chains. Many of the amino acids are the same. But as you might expect, the closer an animal is to us morphologically, the closer its hemoglobin is to us in terms of amino acid sequence. So there's only one difference in amino acid sequence between us and gorillas. Between rhesus monkeys and humans, there are 8 differences. Mice, which are also mammals but not primates, there are 27 differences. Chickens, which are yet more distantly related to us, have 45 differences, and frogs, which are the most distantly related, have the greatest amount of difference, 62 differences. Pseudogenes are yet another category of evidence for evolution, and they're really extraordinary. A pseudogene is a non-functional gene that's a variant of a functional gene in a related species. An example is the glow pseudogene that humans have, as well as most of our primate cousins. Here's the glow pseudogene. It has a series of mutations that keep it from working. Perhaps the mutation's in a broken promoter, or perhaps the mutation is in the structural genes. But rodents have a functional version of this gene. It enables them to produce the enzymes to synthesize vitamin C, but primates can't do it. Why do we have the remnants of this gene? It's a molecular vestigial feature. These pseudogenes only exist because of descent with modification. We and rodents share a common ancestor over here. That common ancestor had the glow gene, and it was able to produce the enzymes to synthesize vitamin C. We primates lost the ability to do that. A mutation emerged, broke the gene, but because we lived in a fruit-rich environment, that mutation wasn't selected against. What's really fascinating is that we're not the only mammals that have that pseudogene. It's also found in guinea pigs, and it's also found in bats. But if you look at the sequence of those pseudogenes, they have different mutations from our pseudogenes. So this pseudogene, and our pseudogene, those are all convergent features. They're analogous, not homologous. Biology, so amazing. Homologies can take us all the way back to the origin of life. There are a couple of features that show that all living things are related. What are they? DNA, used as a genetic material. ATP, used for energy coupling. The same genetic code that's universally used. Ribosomes, used for protein synthesis. Shared metabolic pathway like glycolysis, the Krebs cycle, the electron transport chain, and ATP synthesis through chemiosmosis. This shows that all living things are cousins. We all share a common ancestor going back to the origin of life. The homologies that we just discussed go back 3.8 billion years or so to the origin of life. There are also more recent ones, still ancient, that go back 1.8 billion years that indicate that all eukaryotes are cousins. These include the presence of a nucleus, mitochondria or organelles that are derived from mitochondria, the endomembrane system, genes that possess introns, linear chromosomes as opposed to the circular chromosomes of prokaryotes, and sexual reproduction involving gamete production and fusion of gametes to form a diploid zygote. These are features shared by all eukaryotes. They indicate common ancestry. We're cousins to all the eukaryotes. We're cousins to all life, but we're closer cousins to those in our domain, eukarya. Describe how embryological development provides evidence for evolution. Early embryos of vertebrates look similar. Here's a fish, a reptile, a bird, and a human. During development, the embryo differentiates, and that leads to the adoption of the body form of the adults of the lineage. So bird and human embryos, quite similar, but we all know what adult birds look like, and we all know what adult humans look like. The similarity of the embryonic form indicates common ancestry. There was a common vertebrate ancestor that gave rise to the lineage of fish, reptiles, birds, and humans. Descent with modification leads to the elaboration of form in different lineage, the different body forms of fish, reptiles, birds, and humans. But the embryonic similarity, along with fossil evidence, shows that all of these organisms are descendants of a common ancestor. An additional line of evidence for evolution related to embryos is as follows. Embryos often show vestigial features, such as the tail in humans, the pharyngeal gill slit in humans, that indicate descent with modification from a common ancestor. So again, embryonic development, an entirely separate line of evidence for evolution. Diverse species share common genes for animal development. Describe how this provides evidence for evolution. This is a mind-blowing example of homology. In animals separated by hundreds of million years of evolution, shared genes control certain developmental processes. Here's an example. There's a gene that's called eyeless. That's a master switch that turns on eye development in arthropods and vertebrates. The genes can be transplanted, leading to successful development. What do I mean by that? Here's a mutated version of Drosophila that doesn't develop an eye. It has a mutant version of the eyeless gene. You can find mice that have similar mutations that also don't develop eyes. But if you can take the gene from eye-developing Drosophila and implanted it into the eyeless form, it'll develop eyes. But the amazing thing is that if you take the gene from an eye-developing Drosophila and put it into an eyeless version of a mouse, that mouse will develop an eye. In other words, this doesn't control the eye. The eyes are quite different, but it basically says, build an eye. In a fruit fly, it'll build a compound eye that's common to insects, and in a mammal, it'll build the kind of eye that we have. But there's a master switch that's common to all animals that have eyes that control eye development. Another example of that is homeotic genes. These are genes that basically say that you should have a head up here, and you should have a tail down there, and you should have these appendages over here. And these are common across all animals as diverse as arthropods and vertebrates. And these shared genes, again, are homologies, and they indicate that all animals shared a common ancestor that goes back to the origin of the animal clade about 600 million years ago. What is biogeography? How does biogeography provide evidence for evolution? Biogeography is the study of the geographic distribution of species and varieties. If you enjoy looking at birds as I do, you know that there are certain species that are found on the west coast of North America, and there are others that are found on the east coast. That in and of itself is biogeography. The pattern of distribution on the surface of the earth fits the idea that populations first evolve in one area, then spread to adjacent areas where subsequent evolution occurs. In other words, the pattern of evolution is not only seen through time, it's also seen through space. And a great example of biogeography is the fact that the marsupial mammals are mostly, but not completely, limited to Australia. Why? Because Australia is a kind of isolated island continent. The only placental mammals that were ever able to get to Australia were the bats, which could fly there, and a few rodents that were able to reach Australia on floating vegetation mats. But other larger, land-living, non-flying placental mammals couldn't get to Australia, and hence it's the marsupials that are the predominant mammals in Australia. A fascinating thing connected with the evolution of marsupials is parallel evolution, where because placental mammals never made it to Australia, all of the niches that are filled by placental mammals in North America and Eurasia and Africa are filled by marsupials, and those similarities are all convergent. So the marsupial mole and the eastern mole, convergent similarities based on a common niche. They're both digging in the soil. The sugar glider and the flying squirrel, and even top predators, the now extinct Tasmanian wolf and the wolf that we find in North America and Eurasia. Biogeography, another entire line of evidence for evolution. As we've seen, you can find evidence for evolution in the structure of animals, in their genes, in their distribution over the face of the earth. But you can also find evidence for evolution in time. That's what fossils are all about. What are fossils? How are they evidence for evolution? Fossils are petrified remains of living things. Evolution is change over time, and fossils demonstrate this change. As you go back in time, you find different arrays of living things. Through transitional forms, fossils show descent of modification in a variety of lineages. Now, this example over here is an artistic rendition, but it provides the point, which is that as you go back in time, you don't find modern whales. You find the ancestors of whales who have features that indicate that whales evolved from animals that once were mammals that lived on land, that over time adapted to an aquatic lifestyle. What is relative dating of fossils? How does it work? It's based on the idea of superposition. And the idea is that in rocks that are sedimentary, that are laid down layer by layer, the younger material is on top of the older layer. So this would be younger over here, and this would be older down there. The deeper layers are older than the fossils in layers closer to the surface. That's superposition. But because the Earth is complex, and because there are earthquakes and volcanic eruptions, there's faulting, uplift, and inversions that can complicate this analysis. But based on the position of fossils within sedimentary strata, you can determine which fossils are older than others. In addition to relative dating, determining which fossil is older than others based on their position in sedimentary strata, there's also absolute dating. Describe how absolute dating of fossils works. It's based on the decay of radioactive isotopes that are in fossilized remains or nearby volcanic strata that are interspersed with sedimentary strata. The key idea is the idea of half-life. That's the time it takes for half of a sample of radioactive isotopes to decay from one element to another. So an example, carbon-14 decays to nitrogen-14. Carbon-14 is radioactive, unstable, it decays to nitrogen-14. The half-life is 5,730 years. If a sample has 50% carbon-14 and 50% nitrogen-14, then, for example, if this was a piece of bone, then that bone would be 5,730 years old. It's one half-life. If 1/4 of the carbon-14 is left, then it would be two half-lives, 11,460 years. Other isotopes have longer half-lives. You can find carbon-14 directly in organic remains, but you can also find, as I said before, radioactive elements that are in volcanic strata, and you can use that to date older fossils. So that's how we know how long ago the dinosaurs became extinct, when the first vertebrates emerged, when the first fish emerged, so on and so forth. That's absolute dating of fossils. Evolution can be observed in structures that we see in modern organisms, but there's plenty of evidence that evolution continues to this day. A great example of that is shown in this slide. How is evolution of resistance to DDT in mosquitoes evidence of continuing evolution? Let's talk about what's happening in this slide. Here's a mosquito. This is showing the percent of mortality of mosquitoes to a 4% solution of DDT exposed to them for 1 hour. DDT is a pesticide, kills mosquitoes. Why do we want to kill mosquitoes? Mosquitoes can spread a parasite that causes the blood-borne disease malaria. And malaria is a devastating disease that causes illness in hundreds of millions of people around the world. What happens is that when you spray the pesticide, at first it's very effective and it kills most of the mosquitoes, but never all of them. There's always some mosquitoes that have some inborn resistance that survive the initial use of DDT. Those mosquitoes pass on their genes for resistance. The other ones have been wiped out. So the mosquitoes that survive have resistance and they pass them on. Over time, within a matter of months, you have only about 50% of the mosquitoes are killed. Why? Because mosquitoes have a very short generation time. So this process is occurring quite rapidly. And then after about 16, 18 months, very few of the mosquitoes are being killed. This is observed genetic change. There's some gene that's producing some protein, and that gene is increasing in frequency throughout the population, caused by human selection for DDT-resistant mosquitoes. This is not the only example of the rise of resistance. There are parallel instances of evolution of resistance that have been observed in the evolution of antibiotic resistance in bacteria, resistance to herbicides in weeds, resistance to chemotherapy drugs by cancer cells. It's a phenomenally important example of evolution that we can observe in this day. I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Speciation and extinction. What is the biological species concept and what are some of its limitations? The biological species concept defines a species as a group of organisms that can naturally interbreed to produce viable, meaning healthy, fertile, meaning able to reproduce, offspring, and which is reproductively isolated from other such groups. Here is an example of one species. There are different breeds of dogs, but the dogs don't care. Left to their own devices, they'll interbreed and they'll produce viable, fertile offspring. These two species of ducks, by contrast, don't interbreed, or at least they interbreed to such a small degree that their gene pools are separate. The concept is not perfect, and it falters in a couple of cases. The first is in the case of closely related species, which can often hybridize. They can often interbreed, but not so much. So the gene pools maintain relative separateness. Extinct or asexual species, how could you tell if they could produce fertile or viable offspring? And prokaryotic species. What are reproductive isolating mechanisms? Compare and contrast pre- and post-zygotic forms of isolation. Reproductive isolating mechanisms are processes, behaviors, or other traits that keep the gene pools of closely related species separate. Mallards, pintails, they're both ducks. How do they maintain separate gene pools? There are two kinds of barriers. The first are pre-zygotic isolating mechanisms, and they prevent breeding altogether. They prevent the formation of a zygote. Post-zygotic barriers exist between species that are close enough to mate and form a zygote, but formation of a zygote doesn't ultimately lead to the production of successful individuals, viable individuals, who can survive and produce offspring themselves. Let's look at pre-zygotic isolating mechanisms. The first kind is behavioral. Different mating rituals or courtship behaviors would keep a female and a male from accepting one another as members of the same species and mating. Temporal means that they would breed during different times of the day or different seasons. One species mates in the winter, another mates in the summer. They're not going to interbreed. Mechanical, structural barriers that prevent sperm or pollen from reaching an egg. If the flower structure is very different, then a pollinator wouldn't be able to bring pollen from one flower to another flower. Habitat. One species lives in the forest, another lives in a meadow. One species lives in the uplands, another lives in the lowlands. They won't meet each other in order to breed. And finally, gametic. The egg won't allow fertilization because there's some molecular mismatch that would keep the sperm from being able to bind with the egg and induce the kind of changes that lead to fertilization. List and describe 3 post-zygotic isolating mechanisms. Post-zygotic barriers can exist between species that are close enough to mate and form a zygote, but nevertheless their gene pools remain separate. These can include hybrid inviability. The hybrid organisms don't develop. There might be hybrid sterility. The hybrid offspring are healthy, but they can't reproduce. That's what's going on between horses and donkeys, which can interbreed to produce mules, but the mules themselves are sterile. Finally, there's hybrid breakdown. The hybrids are healthy and can reproduce, but the next generation, the F2s, are inviable or infertile. Contrast allopatric and sympatric modes of speciation. Allopatric speciation involves a geographic barrier. It's what's shown over here in number 1. Sympatric speciation occurs without a geographical barrier, as shown over here in number 2. In allopatric speciation, geographical isolation leads to genetic differentiation, which leads to reproductive isolation. In stage 1, a species is spread out over a geographical range. There's gene flow between any subpopulations. In stage 2, some kind of geographic barrier, like this shown at B, arises and it splits the species apart, so there's no longer gene flow between these two isolated populations. In stages 2 and 3, environmental differences result in different selective pressures, which lead to genetic differentiation. There's mutation happening, and the environment is selecting which variants are going to be more favorable in each different environment. And finally, when that barrier disappears, the two formerly subspecies have become so different that they can no longer interbreed. They were geographically separated, now they're reproductively isolated by any of the mechanisms that we've just discussed. Sympatric speciation can occur in animals and plants. In plants, there can be sympatric speciation through polyploidy. This can lead to changes in chromosome numbers, causing instant one-generation reproductive isolation between the newly emerged species and its parent species. One example of sympatric speciation in animals involves the hundreds of species of these fish that are called cichlids that live in Africa's Lake Victoria. What is thought is that sexual selection has led to reproductive isolation between the subspecies. That's been experimentally verified. It's a very complex story, and what I want to encourage you to do is go look at the case study on learn-biology.com. The link is below. Here's another mechanism of sympatric speciation in animals. Adaptation to specific microhabitats can also lead to reproductive isolation and speciation, such as the evolution of a variety of lice that inhabit different parts of birds. This is kind of astonishing, but there are lice that, for example, live only on the head of an albatross, and there are different species that live on the wings. There are other species of lice that only live on the heads of parrots, while others live on the wings of parrots. These are different species, and they are in different parts of the bird, and you might say, well, for a little louse, well, maybe that's like a mountain, but that doesn't count as allopatric speciation. There's no geographic barrier. All of these lice are just becoming extraordinarily specialized as they adapt to different parts of these birds. And these specializations are getting passed on, and that's leading to speciation over time. So sympatric speciation in animals because of microhabitat adaptation. What is adaptive radiation? This is a concept that we dealt with earlier when we talked about the evolution of homologous features. Adaptive radiation occurs when one parent species produces several descendant species, each of which has unique adaptations and fills a different ecological niche. The 14 species of Galapagos finches shown here, all of which are descendants of a single South American species, are an example. This one species arrived at the Galapagos Islands and then radiated into a whole variety of descendants, each of which has different features, each of which has a different ecological niche. Phylogeny is a reflection of adaptive radiation. Each of the different branches show splitting from a common ancestor. That's adaptive radiation on a very large scale. Finally, homologous and vestigial traits, which we talked about in the context of evidence for evolution, are also a result of adaptive radiation. It's about splitting from a common ancestor and subsequent descent with modification. We're now going to pivot away from speciation and just look at the evolution of variation and its importance. So, why is phenotypic variation important for evolution? I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. Phenotypic variation is essential for evolution. It's the raw material upon which natural selection acts. Natural selection selects for organisms with phenotypes that confer a selective advantage. Remember that the genotype is invisible to natural selection. What natural selection sees, what natural selection acts upon, is the phenotype. And the result is that individuals with advantageous phenotypes survive and reproduce at higher rates than organisms with less advantageous phenotypes. Now, as those organisms reproduce, they're passing along their genotype, but that's somewhat of a secondary effect. Natural selection acts upon phenotypes. And note that with no phenotypic variation, there can be no natural selection and no adaptation. And that's why loss of variation is so dangerous to a species and can often lead to extinction. So here are a couple of examples of the importance of phenotypic variation. Let's start with this one. Explain how variations in phospholipid structure can serve an adaptive function in browsing mammals that forage in snowy environments. Phospholipids, we remember those from Unit 1. They've got a glycerol with a hydrophilic head and a hydrophobic tail. These are fatty acids over here. Note that these fatty acids can be saturated or unsaturated, and there can be a lot of variation in that. In mammals that browse in the snow, there is a tendency for the phospholipid tails that are in the body core to be more saturated, and those in the extremities to be more unsaturated. Why? Because as the temperature decreases, these unsaturated fatty acid tails keep the membrane fluid so that diffusion can continue to occur, so that oxygen can continue to flow from the blood into the cells, whereas the phospholipid tails in the body core are more saturated, and that provides a correct level of fluidity in that part of the body. So there tends to be a gradient of more saturated fatty acid tails in the core, more unsaturated fatty acid tails in the extremities. Explain how variation in hemoglobin maximizes oxygen absorption in humans and other placental mammals at various life stages. Let's remember that hemoglobin is the protein that transports oxygen in red blood cells in almost all vertebrates. Before birth, humans and other mammals produce fetal hemoglobin. Fetal hemoglobin is a variant, and it has a higher affinity for oxygen than adult hemoglobin. Here's the difference. All hemoglobins are the same in that they have these two alpha chains. They're shown in red over here, but adult hemoglobin has beta chains, where fetal hemoglobin has gamma chains. They're shown in yellow over here. Because of the higher affinity for oxygen, the fetal hemoglobin will absorb, it'll create a gradient that will cause oxygen to flow from maternal blood in the placenta over here into fetal blood. So here's the gradient, and that of course is highly adaptive because that's how the fetus gets oxygen while it's in the uterus. But after birth, what happens is that developmental genes kick in, and slowly the beta form starts to be produced more and more until it's completely predominant in the adult, whereas the gamma form starts to fall and fall and fall. And you can see that that happens even before birth, but the transition is pretty complete 42 weeks after birth has occurred. Explain how variation in chlorophyll types increases the efficiency of photosynthesis. Green plants have two main types of chlorophyll. There's chlorophyll A over here and chlorophyll B over here, and the difference comes down to just a functional group. Chlorophyll A has a methyl group over here, whereas chlorophyll B has a carbonyl group. Chlorophyll B absorbs more blue light, and that's best in indirect light or in shady environments or shady situations. And chlorophyll A absorbs more red light, and it's best in direct light. And therefore, plants can do a couple of things. One is that there are plants that are shade adapted and other plants that are direct light adapted, and those will have different proportions of chlorophyll A and chlorophyll B. And then most plants have both. And having both types of chlorophyll increases the amount of light energy that plants can use during photosynthesis over the course of a day or throughout the seasons. Now that we've looked at processes that increase biological variety, speciation, the development of variation, now let's look at the flip side, an equally important side of the evolution of life, which is the idea of extinction. And the big idea is that extinction is a normal part of the process of life. Greater than 99%, it might be 99.9% of the species that have ever lived have become extinct. Let's describe the process that a species goes through as it heads towards extinction. This is called an extinction vortex, and it starts with a population decline. So small isolated populations are the ones that are most vulnerable to extinction. That population decline can be caused by an adverse change in the physical environment, climate change, which has happened throughout the history of Earth, or the arrival of a competitive species that reduces the species' fitness and reproductive rate. That leads to genetic drift, which we discussed in population genetics. That results in a loss of genetic diversity. That leads to less variability, and that loss of variability leads to reduced fitness. There's more genetic uniformity, and that reduces the ability to adapt to environmental change, and the result is a smaller population that then goes through this process again and again. It's a positive feedback loop. It's not a positive thing, but it's a positive feedback loop that feeds on itself, it accelerates itself leading to extinction. What we just described is the typical normal process of extinction. That's quite different from mass extinction. What is a mass extinction? A mass extinction is a widespread, rapid decrease in Earth's biodiversity. Often, mass extinctions are caused by geological or even astronomical causes, though biological causes are possible as well. And there have been at least 5 major extinction events during the last 600 million years, and those are the numbered peaks in this diagram. The most recent peak, which occurred 60 million years ago, is the Cretaceous extinction. That's the one that wiped out the dinosaurs. Now, you might think that extinction is all bad, mass extinction in particular, but there's a connection between mass extinction and adaptive radiation. Mass extinction leaves vacant a variety of ecological niches, and that is an ecology term that we'll deal with in Unit 8. Those are ways for a species to make a living. And the result is a mass extinction, such as that shown at 2, which wiped out all of this previous existing biological diversity, is followed by extensive adaptive radiation in the species that survive. And here's one, the species at 4, and it made its way through, for whatever reason, this mass extinction over here, and then it underwent an extensive adaptive radiation. And an example of that is the diversification of placental mammals, mammals like us, that followed the Cretaceous extinction that wiped out the dinosaurs. And again, just to connect this to our own lives, if that mass extinction 65 million years ago hadn't happened, we wouldn't be here talking about mass extinctions. It's because our ancestors made it through that mass extinction and then radiated to produce the whales, the hippopotamuses, the primates, the rodents, and our group is the primates, and here we are. How is human activity affecting extinction rates? Humans are the cause of what's been called the 6th extinction. Here are the 5 great extinctions that have happened in the past. Humans are causing a decline in global biodiversity that could rival these previous 5 mass extinctions. How are we doing that? Human activities that are causing extinction include destruction and fragmentation of habitat, overhunting, overharvesting of animals and plants, intentionally or unintentionally introducing invasive species into new habitats. This is material that we'll look at more closely at the end of AP Bio Unit 8. Are you asking yourself, how am I going to get a 4 or a 5 on the AP Bio exam? It's a good question because it's a hard test. But we have a plan for your success. Go to learn-biology.com, sign up for a free trial, and complete our interactive tutorials and interactive AP Bio exam reviews. We guarantee you a 4 or a 5 on the AP Bio exam. See you on learn-biology.com. Topic 7.9, phylogeny. What is phylogeny? What is a phylogenetic tree? How are such trees built? Phylogeny means evolutionary history. A phylogenetic tree, also known as an evolutionary tree, is a branching diagram that shows evolutionary relationships. And phylogenetic trees are built using morphological, that means structural, molecular, or genetic evidence. This is a phylogenetic tree. And one thing that I want to emphasize is that it is a claim that's based on evidence. So the claim here is that hippos and whales are more closely related to one another than whales are to deer. Why is that? Because based on the evidence, which would be morphological, molecular, genetic, hippos and whales have a more recent common ancestor than either does with deer. We'll see the details in the subsequent slides. Define the term clade. Clade is an incredibly important biology term that's not known to the general public, but now you'll know it and you'll be able to explain it to people. A clade is a group of organisms that consists of a common ancestor and all of that ancestor's descendants. In this diagram, all of the species and all of the numbered groups are clades. So down here, the large ground finch, that's a seed-eating finch in the Galapagos Islands, that's a clade. All members of a species have a common ancestor. All humans have a common ancestor. All human beings are a clade. The large ground finch and the common cactus finch have a common ancestor. That's this over here at E. So those two species together constitute a clade. If you go back in time, because there's implied time in any phylogenetic tree, then all of these four species, the woodpecker finch, the small tree finch, the common cactus finch, and the large ground finch, they comprise a clade. That's group number 5 over here. Their common ancestor is this species that's over here at C. Define shared derived character. A shared derived character is a trait that identifies, that distinguishes a clade, and it evolved in the common ancestor of that clade, and it sets it apart from other such clades. So, for example, lungs and forelimbs separate this clade, all of these organisms that are classified as frogs, lizards, alligators, robins, rats, and gorillas. That constitutes a clade, and they all have this shared derived character. Going back in time, the shared derived feature vertebral column separates this clade that includes all the salmon through gorilla. Fur and mammary glands separates the clade that includes rats and gorillas from all the other organisms. These are mammals, and those are shared derived features of mammals. What are nodes and sister groups? A node is where two branches in a phylogenetic tree diverge, and nodes represent the common ancestor of the two diverging lineages. So at left, letters A through E, those are all nodes. Nodes, common ancestor. So over here, there was a common ancestor that led to the divergence between this clade over here and this species over here, the green warbler finch, which is not part of the clade that includes all of these organisms that have a common ancestor at B. Sister groups are the descendants that split apart from the same node, such as the common cactus finch over here and the large ground finch. Those can also be called sister species because that's the taxonomic level at which they're at. So the woodpecker finch, the small tree finch, that clade, that's a sister clade to this clade that's defined by letter E, the common cactus finch and the large ground finch. What is an outgroup? An outgroup is a more distantly related group of organisms that's used to determine the evolutionary relationships among the other organisms in the tree, which is the in-group. In this phylogenetic tree, A is the outgroup for this clade that's over here at number 2. It's a point of comparison for the in-group, and it's a species or it's some other taxonomic category that's not part of the clade to which all the other organisms in the phylogenetic tree belong. What's the biggest mistake to avoid in phylogenetic analysis? The misconception that many students naively have is that vertical closeness on a horizontal tree indicates evolutionary closeness, but it doesn't. For example, the frog and the lizard, they're next to one another, but they're not particularly closely related. The only thing that matters on a phylogenetic tree in terms of evolutionary relatedness is the recency of common ancestry. So alligators and robins, they have their common ancestor over here. Rats and gorillas, they have their common ancestor over here. Frogs are right next to lizards, but their common ancestor, the common ancestor of the frog and the lizard, is all the way back here. And the basic idea is that nodes can rotate. So this phylogenetic tree over here is correctly drawn. It's exactly equivalent to this one over here, but you can see that in this one, the lizard is no longer next to the frog. Right? So here's the lizard down here, here's the frog over here, and here's the lizard over here, and here's the frog over here. It's just about rotation, and if you think about the art form called a mobile, mobiles all rotate. It's a kind of kinetic sculpture. And if you can think of phylogenetic trees as one instantiation of a kinetic sculpture, you'll have the right idea about this. Always look for recency of common ancestry when you're trying to figure out the evolutionary relationships described by a phylogenetic tree. Define ancestral feature. An ancestral feature is a trait that members of a clade share, but which is also shared by larger, more inclusive clades. It doesn't define the clade. So for clade G, which includes rats and gorillas, that's the mammal clade, an ancestral feature would be claws or nails. And why is that? Because mammals, they do have that trait, but it's also found in this larger, more inclusive clade that runs from lizards down to gorillas over here. Lungs and four limbs is another ancestral trait of rats and gorillas, this mammal clade. It doesn't define the clade, but everyone in the clade has to have that because there's a larger, more inclusive clade that has that feature. What type of evidence is typically used to construct phylogenetic trees of existing species? Before the 1960s, morphological similarities was what biologists used to determine who was more closely related to whom and thereby construct phylogenetic trees. But more recently, post-1960, remember Watson and Crick figured out the structure of DNA in 1953. Nucleotide sequences in DNA and RNA and amino acid sequences in proteins have become the gold standard in determining phylogenetic relationships. So a DNA analysis would indicate that the common cactus finch is more closely related to the large ground finch than to any of the other Galapagos species. That's a hypothesis, and you could confirm that through DNA sequencing. What are molecular clocks? Here's how this works. For any specific protein or nucleic acid, the rate of change that's caused by accumulated mutations is constant over time. And if you can calibrate the amount of change in a gene or protein to when species split apart, which you can sometimes determine from the fossil record, then you can do two things. One is you can determine a rate of change in that gene or protein, and you can use that rate to determine when other species split apart. What this graph says is it's looking at hemoglobin as a molecular clock. Based on some fossil evidence, it's basically saying that 20 mutations accumulate every 100 million years. So therefore, if you're looking at the hemoglobin between two species and you notice 40 amino acid substitutions, then you could infer that that occurred more like 230 million years ago. This is widely used in evolutionary biology. It's the basis for claims that, for example, humans and chimpanzees had a common ancestor about 6 million or so years ago, based on the similarity of DNA between us and chimps. Topic 7.13, the origin of life. This is one of the coolest topics in biology. For AP biology, you need to master a very specific body of knowledge. The key question to address when trying to understand the origin of life is, how did life emerge naturally? And this connects to some biology that you've already learned in your AP biology course. For example, you've learned about the cell theory and the idea that all cells come from pre-existing cells. Well, at some point there had to be a first cell, and in fact, that moment really marks the emergence of life. How did a cell emerge with the absence of another cell to create it? That's one of the mysteries that we have to contend with in the origin of life. The other one has to do with chemistry. The chemistry of life is largely controlled by complex proteins called enzymes. Enzymes can take simpler substances and make them into more complex substances through processes like dehydration and synthesis. Well, how do we get the complex substances that life is based on in the absence of enzymes? What are the key steps required to explain the origin of life? The Earth is extremely ancient. It's been around for 4.5 billion years. And life is thought to have emerged about 3.8 billion years. What steps allowed life to emerge? The first thing is that Earth needed to become a habitable and more stable planet. The Earth formed from a nebula about 4.5 billion years ago. That's beyond the scope of this course. But what's important to know is that for the first several hundred million years of Earth's existence, it was pummeled by asteroids and comets, and all of that needed to come to an end before life could begin to emerge. Step number 2 is about chemistry leading to biology. Biology is based on cells, and those cells in terms are based on complex molecules, polymers. The polymers are built from monomers. So how did those monomers emerge? That had to happen abiotically. In the absence of enzymes, monomers had to start to emerge. We need to start to have amino acids, nucleotides, fatty acids, etc. In today's living world, monomers get combined into polymers. How could that happen in the absence of enzymes? There needs to be some kind of abiotic process by which that could happen, where we have the abiotic synthesis of polymers from monomers and the formation of vesicles. Those are going to become the little capsules into which cells will emerge. In step 4, we need to combine those monomers and polymers into those little vesicles to create protocells. These aren't quite living cells the way that we have them today, but they have many of the characteristics of cells. And finally, we have the emergence of self-replicating cells. That would be the last universal common ancestor over here, and that gives rise to the 3 great domains, archaea, bacteria. Those two combine, that's another story altogether, to form the eukaryotes. The Miller-Urey experiment, which was carried out in the 1950s, showed that in an abiotic setup without life, you could successfully synthesize amino acids in a simulated early Earth environment. Now, as we'll see, some of the details that were involved in the setup of the Miller-Urey experiment were wrong. They don't correspond to our current understanding of the geology of the early Earth, but it's an important proof of concept. So, what did Miller-Urey do? They created this sterile apparatus with various chambers and tubings, and the whole thing was sealed off from the outside world and sterile. This chamber over here at number 2 represented the early oceans. It was heated by a Bunsen burner or some other heating device. And then the steam circulated through these tubes over here. This large area over here is a chamber that represented the early atmosphere. And it was loaded with gases that were supposed to be present in the early atmosphere. And that includes gases like methane, ammonia, hydrogen, and water vapor. Those gases were chosen because in the 1950s, that was the current understanding of what the early Earth's atmosphere would be. There are 2 important points. One is that that was chosen because that was the known atmosphere of some of the gas giant planets like Jupiter, and it was thought that that might also be present on the Earth. But the most important thing was that no oxygen was present, because oxygen was produced by photosynthesis. It wasn't present in the atmosphere of the early Earth. There were electrodes, shown over here at number 4, by this plus and minus sign. And what they did is they produced sparks, which simulated lightning. The gases continued to circulate. They circulated through this condenser over here, and that caused whatever was formed in this chamber here to be caught within this trap. This trap could be sampled without contaminating the apparatus. And after a certain period of time, Miller, who was the actual experimenter, it was Stanley Miller, Harold Urey was his graduate thesis advisor, sampled the liquid, and they found the presence of amino acids. Well, like I said, that didn't necessarily establish that life could be produced in the absence of life, but it did establish that some of the monomers of life could be produced in the absence of life. As I said, some of the details were wrong, but this has proven to be the model for many subsequent experiments where there are inorganic catalysts and different mixes of gases that are changed, and in subsequent experiments, the nucleotide bases have been produced, fatty acids have been produced, many of the monomers of life have been produced. So, what's the basic idea? In the absence of life, it's been experimentally established that some of the monomers that make up the chemistry of life could be produced. To understand the origin of life, you have to understand the origin of heredity. It's widely thought that RNA, not DNA, was the first hereditary molecule. Why? RNA does store genetic information in viruses, so it has the capability of being a genetic molecule. But unlike DNA, RNA can also act essentially as an enzyme. RNA can have catalytic properties. It does so in ribosomes, where the catalytic part of a ribosome is what actually stitches together amino acids to create polypeptides and then proteins. We have spliceosomes, we have microRNAs, all of which are catalytic, acting upon the world. DNA with its double helical form is fantastic for storing genetic information. RNA, because it has both catalytic abilities and information storage capabilities, is widely thought to have emerged as the first genetic molecule. Before there were cells, it's thought that there might have been a phase in which there were self-replicating systems of RNA molecules that were subject to natural selection that would grow in complexity. And that's called the RNA world that led up to the last universal common ancestor of life. Here's how that might have happened. In the beginning, what we had were inorganic precursor molecules. It's what we talked about in the preceding slide with the discussion of the Miller-Urey experiment. So at some point, the sugars, the phosphate, and the nitrogenous bases that make up RNA would have to emerge. Those would combine through abiotic, inorganic processes to form RNA monomers. Other abiotic, non-enzymatic processes would lead to the next step. And in that next step, we have RNA polymers that start to emerge. They're shown at C. RNAs can fold up into complex shapes, and that's what we start seeing over here at D. And those complex shapes can start to have enzymatic properties. One of those enzymatic properties could lead systems of RNAs. It doesn't have to be just one molecule, but there could be a system of interacting molecules that could lead the system to replicate itself. At some point, those self-replicating RNA systems would become encapsulated within some kind of lipid bilayer. And at that point, at letter F, we have a protocell. Further natural selection, further evolution would lead to the emergence of the last universal common ancestor. That's the population of organisms that gives rise to both archaea and bacteria, and ultimately through a fusion of those two domains to eukaryotes. And if you can look at this diagram, you should pause and test yourself and identify all of the numbered items. Then you've learned a lot of cellular biology and you're well on the way to being set up for success on the AP Bio exam. Just to run through, at number 1, we have a lipid bilayer. Remember, it doesn't have to be a phospholipid bilayer because archaea have a different kind of bilayer structure. At 2, we have DNA being the genetic material that's found in all living things. At 3, we have RNA, which is used for information transfer and other things. At 4, we have ribosomes, which are translating messenger RNA into protein. At 5, we have a membrane channel that's allowing matter and energy to flow in and waste to flow out. At 6, we have complex proteins that are acting as enzymes, which are combining monomers into polymers through dehydration synthesis and then pulling apart those polymers through hydrolysis. And at 7, we have ATP synthase, which is using a flow of protons across the ATP synthase channel to generate ATP, that universal energy molecule from ADP and phosphate. That's the last universal common ancestor, and that's what you need to know about the origin of life for AP Biology. Topic 8.1, Responses to the Environment. Unlike the rest of Unit 8, Topic 8.1 is somewhat difficult in terms of providing you with exact guidance about what to study. That's because the objectives are very general, and the College Board has provided these exclusion statements which tell you that you don't really need to know any specific body of knowledge in order to be able to handle this topic. So what we're going to do is we're going to look at a couple of fascinating case studies and illustrative examples that'll give you practice at handling the kinds of data sets and images that the College Board or your teacher might be throwing at you on the AP Bio exam or your upcoming test. We're going to start by looking at predator warnings. Predator warnings, also called alarm signals, are calls or cries emitted by social animals in response to predator danger. These have been extensively studied in the Belding's ground squirrel, which lives in the Sierra Nevada mountains of California. Belding's ground squirrel has distinct warning calls for aerial predators like hawks and eagles, and for terrestrial threats from bobcats, coyotes, and weasels. Here's warning call type 1 for an aerial predator. And here's warning call type 2 for terrestrial predator. Note that these predator warnings are not unique to the Belding's ground squirrel. African vervet monkeys, which are primates like us, have distinct calls for leopards, snakes, and eagles. Predator warnings are altruistic. They're self-sacrificing. They involve individual risk to the self to protect others. When a squirrel emits a call about an aerial predator, it increases the chance that that predator will attack the animal that calls out. That's of course true for a terrestrial predator as well. And that leads us to the question, why would an animal risk its own life to protect others? Altruism can be explained by kin selection and inclusive fitness. Let's define both of these terms. Kin selection means that the value of a gene is not based on whether it promotes survival in a single individual, but also in how it affects survival in the individual's relatives. Inclusive fitness means that if an allele promotes sacrificing oneself for the benefit of one's close relatives who also share that allele, then that allele might increase in frequency. The idea of kin selection and inclusive fitness was cleverly captured by the British biologist J.B.S. Haldane, a very important evolutionary thinker of the 1900s, who famously said, "I would lay down my life for 2 brothers or 8 cousins." What did Haldane mean? Well, here's me. I obviously have 100% of my genes, but my siblings have 50% of my genes. So if I sacrifice myself and thereby allow my brother and sister to continue living and passing on my genes, it's an even trade. So it makes sense to sacrifice myself for 2 siblings. I have a 12.5% relationship to each of my cousins. And just think about the fact that you have a 25% relationship to your aunt or uncle, so you'd have a 12.5% relationship to their children. Again, it makes sense for me to lay down my life so that 8 of my cousins would survive and continue to pass on my genes within our shared gene pool. With those concepts in mind, let's take a look at a study by Paul Sherman about predator warnings in Belding's ground squirrels. He carried out the study in the 1970s. To begin with, notice this graph. It shows the mean distance that males and females move from their natal burrow. That means the burrow where they were born. So everybody obviously starts out in the burrow where they were born, but then the behavior of males and females is quite different. Females wander very little over the course of their lifespan. These squirrels don't live very long, so even when these squirrels are 26 months old, which is quite old for a squirrel, they're pretty close to the burrow where they were born, less than 50 meters. The males, by contrast, move quite a bit. So by the time they're 2 years old, they're up to 280 or so meters away from the burrow where they were born. Sherman's key move was to observe these ground squirrels and then to classify who was calling based on 2 characteristics. The age class, whether they were juveniles, 1-year-olds, or adults, and then their sex. I've labeled the age class, but I haven't labeled the sex, male or female. Here's how to read this. The left side of this graph shows the distribution of each of these age classes and sexes in the population. And again, you know the age, you know that these are adults, but you don't know which one's male and you don't know which one's female. And that's true for adults, 1-year-olds, and juveniles. The top graph shows the first squirrel to give the alarm call, and the second shows all callers regardless of precedence. In other words, it might have been the first call, the second call, the third call, and these were calls to a predatory mammal, so to a ground predator. On the right side, it shows what Sherman actually observed in terms of who was giving the call. And what I'd like you to do is to predict which rows are for males and which rows are for females. And I'd like you to inform your prediction by this graph over here, and also what you've learned so far about kin selection and inclusive fitness. Make a prediction, and then I'll show you what the answer is. Here's what Sherman found. Females were overwhelmingly more likely to emit alarm calls than males were. For example, among adult females, they're about 30% of the population, but adult females emitted over 60% of the first call for a predatory mammal. Males represent about 20% of the population, but they don't even call 5% of the time. So males are way underrepresented in terms of their taking the risk to call, whereas the females are way overrepresented, and that's true of every age class, and that's also true of callers in general, not just the first call, but callers regardless of precedence. So my challenge to you is, how would you interpret this? Think about it, and then you can see my answer. Sherman's interpretation is that females are more altruistic than males. But why would that be? Remember that a warning call attracts attention. When females emit a call, they're drawing attention to themselves and warning their close female relatives, their sisters, their daughters, their female cousins. The males don't call because the risk to themselves is not offset by a benefit to their close relatives. So the conclusion is that predator warnings in Belding's ground squirrels are an example of kin selection and inclusive fitness. And what you should take away in addition to this concept is the way that we used claim, evidence, and reasoning to pull this together, especially how you connect this piece of data to this piece of data to thereby conclude what we did about kin selection and inclusive fitness. The altruism that we just discussed in the Belding's ground squirrel reaches its peak in a type of animal behavior and social organization that's called eusociality. What is eusociality? It's a social structure in which some individuals within a colony breed while others are non-reproductive. And just to state the obvious, humans are highly social, but we're not eusocial. Who is eusocial? Bees. In bees, there's a single queen who lays the eggs. She's reproductive. There are thousands of workers that take care of the larvae, they build and clean the nest, they forage, but they don't reproduce themselves. In other words, what they do is they work so that the queen can successfully reproduce. And there are a couple of dozen of males, and the males leave the nest to mate, and then they die. That's eusociality. Eusociality is not just found in the bees, as we just discussed, it's also found in ants and wasps, in termites, in one species of shrimp, and 2 species of mole rats. That's a type of mammal. And again, what are we saying? We're saying that in a termite mound, there are a couple of reproductive individuals, and everybody else is non-reproductive, working for their benefit. And that's true in a colony of naked mole rats as well. There are individuals who get to reproduce, and everybody else serves them and serves their reproductive interests. Eusociality can be partly explained in animals like bees and ants through the phenomenon of haplodiploidy. And that's a kind of sex determination that we discussed back in Unit 5. I'll review it now and I'll connect it to eusociality. The idea of haplodiploidy is that females are diploid in the same way as you are diploid with 2 sets of chromosomes. Here we see the chromosomal situation of the diploid queen, and it's very simplified. Bees have 32 chromosomes as their diploid number. I'm showing only 6 here. The diploid queen creates gametes by meiosis in the same way that humans and mammals do. And as she does, she passes on 50% of her genes to her daughters. The daughters are the non-reproducing females. In males, things are quite different. Males are haploid. They have only half the chromosomes in every cell of their body that the females do. A haploid male drone, who's reproductive, creates gametes by mitosis, not by meiosis. And what that means is that he's passing on 100% of his genes. How do you get to be a male? Males develop from unfertilized eggs that are laid by the queen, and the drones, as I just said, pass on 100% of their genes to their daughters. So how does haplodiploidy connect to eusociality? The sisters are 75% related to each other. What do I mean by that? Well, they inherit 50% of their mother's chromosomes and 100% of their father's chromosomes. So on average, they're 75% related to one another. That means that they're more related to one another, to their fellow sisters, than they would be to their own offspring. That means that genetically it makes more sense in terms of the inclusion of their genes in the gene pool for workers to help their queen create more sisters than it would be for the females to reproduce themselves, because their own daughters would be 50% related to them. So that is a genetic explanation for the altruism of the female workers within a beehive. And just to emphasize something, eusociality can exist without haplodiploidy. Termites are not haplodiploidy, and they are eusocial, and that's true of the naked mole rat as well. And again, this is an important concept. It's important for you to know about as an AP biology student, but the most important thing for you to be able to do is to look at a diagram like this and use that as the basis for formulating a claim with some evidence and some reasoning about the connection between eusociality and haplodiploidy. Topic 8.1 is full of fascinating case studies. We're going to stop presenting them here right now, but what I want to encourage you to do is to go up to learn-biology.com, where you can learn about amazing things like why some voles, a small type of rodent, are monogamous while others are promiscuous. You can learn about how ants learn how to make their way back to the nest after finding food in a straight line, not an easy thing for them to do, how turtles can go out to sea, forage there for years, and then how they make their way back to their nest, how and why animals school, and how honeybees with their relatively small brains can communicate to one another through their dances about the location of food sources. It's unbelievable stuff in and of its own right, but the most important thing is that by looking at these case studies, you'll get to be very good at looking at data sets, illustrative examples, visual representations. That will be the kinds of things that you need to do on the AP Bio exam or your upcoming test on Unit 8. Your success in AP Biology starts here. Are you struggling with AP Bio? With learn-biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to learn-biology.com to find out how you can master your biology course and crush the AP Bio exam. Topic 8.2, Part 1, Metabolism and Individual Energy Use. Let's start by answering the question, what is metabolism? Metabolism is the sum total of the chemical processes that take place within an organism. Here we're showing a cell, but it could be the inputs, the processes, and the outputs for an entire organism. What is metabolic rate? It's the amount of energy that an organism expends during a given amount of time. Basal metabolic rate is the amount of energy consumed by an organism at rest at a comfortable temperature. They don't have to be sleeping as these lions are, but it's resting metabolism. How is metabolic rate measured? To answer this, we have to harken back to Unit 3 and think about the formula for cellular respiration shown over here. You can measure the input oxygen. That could be happening here in this woman at rest who is hooked up to a kind of metabolism measuring machine. You can measure carbon dioxide production. This machine also might be measuring the amount of carbon dioxide she's emitting. In a respirometer, which is typically used in a lab back in Unit 3, you're measuring consumption of oxygen, and you can also measure the production of heat. That leads us to a discussion of two major metabolic strategies that involve being an ectotherm and that involve being an endotherm. Endotherms are limited to mammals, birds, and a few other species. Endotherms generate their body heat internally, metabolically, as a result of heat that's generated through cellular respiration. Their body temperature is regulated around a set point. So, for example, here we see an endotherm, a wolf, and despite the environmental temperature, its internal temperature is going to stay the same. Ectotherms are different. Their body temperature conforms to the environmental temperature. So when it's 20 degrees out, the snake's body temperature will be about 20 degrees. That's true as it gets colder, and that's true as it gets warmer. What are the advantages of being an endotherm? You can be active regardless of the environmental temperature, and that gives you flexibility throughout the seasons and whether it's day or night. And it explains why the predominant animals in the coldest environments are endotherms. Mammals like polar bears and birds like penguins. Those are the animals that can survive on snow and ice. So what are the advantages of being an ectotherm? The energy requirements of ectotherms are much less. Ectotherms need about 1/10 the food per gram of tissue than endotherms do. As an example, at 400 pounds, this American alligator needs the caloric equivalent of only a few slices of bread to meet its energy needs. You, as a mammal, need the entire loaf to meet your energy needs. That's how endotherms and ectotherms compare. What's the relationship between temperature and metabolic rate? In ectotherms, metabolic rate increases with temperature. As the temperature goes up, the metabolic rate goes up in an ectothermic organism like a snake. In an endothermic organism, a mammal or a bird, the relationship is more complex. To begin with, at cold temperatures, the metabolic rate needs to be high. Why? Because a lot of energy needs to be consumed to create heat so that the animal can maintain its metabolic rate at that high, constant set point. As temperatures become more moderate, the metabolic rate falls, and it can fall to the basal metabolic rate. And as temperatures get high, then the metabolism rises as cooling mechanisms are deployed, and those require some energy as well. Metabolism and size in endotherms. As endothermic animals get bigger, their rate of energy use, their basal metabolic rate, increases. No surprise, but an elephant consumes a lot more energy than a mouse does. And if you scale your axes, your x-axis and your y-axis, as is shown here, that increase in energy use scales in a linear way. If you look at energy use from a slightly different angle, there's a fascinating relationship between size and metabolic rate. That relates to the relative metabolic rate. The relative metabolic rate is the metabolic rate per unit of body mass, and it decreases as size increases. So the smaller the mammal, the more energy each gram of tissue requires. In other words, each gram of tissue in a shrew is burning much, much more energy per the same unit of time than a gram of tissue in an elephant. Let's look at some specific examples. Here's an African elephant, weighs in at 6 million grams. It consumes 70,000 calories a day. Its energy usage per gram is 0.011. Now let's look at the other end of the scale. A tiny shrew, which weighs only 1.8 grams. It needs 144 calories a day, and that's 80 calories per day per gram of tissue. That difference in relative metabolic rate has vast consequences in the physiology of an organism. We'll look again at the shrew, 1.8 grams, weighing less than a dime. Its heart rate is 1,200 beats per minute. It has 800 respirations, 800 breaths per minute. It starves to death if deprived of food in just 4 hours, and it has to eat 200% of its body weight every day. An elephant, by contrast, its heartbeat is only 30 beats per minute. It takes about 4 or 5 breaths per minute. It can go many days without food, and it eats about 4% of its body weight every day. Why are relative metabolic rates so different for large animals and small animals? Why is the metabolic engine of a mouse or a shrew revving at such a higher speed than that of a huge animal like an elephant? It has to do a lot with surface area to volume ratios. That's a lot of the explanation. Smaller animals, they'd be over here, they have a much larger surface area to volume ratio than do larger animals. Remember that volume is a cubic function, whereas surface area is a square function. So as an animal increases in size, its volume increases much faster than its surface area. And you can look back to my video about that in Unit 2 if you need more instruction about that. As a result, smaller animals lose heat through their surface much more easily than large animals do. To replace that heat, smaller animals need to perform more cellular respiration, increasing their relative metabolic rate. Topic 8.2, Part 2, Energy Flow Through Ecosystems. What is an ecosystem? It's a community, the living populations in an area, plus the abiotic, non-living parts of the environment. Those abiotic parts include the air, the soil, the water, and the energy that's flowing through that ecosystem. Two essential concepts related to ecosystems are food chains and food webs. A food chain is the passage of energy and matter from one organism to the next within an ecosystem. On the left, we see a terrestrial food chain where a plant is getting eaten by insects, which is getting eaten by other insects, which passes to a frog, etc. On the right side, we see something equivalent for an aquatic food chain. On the right side, we have a food web, and a food web shows all the interconnected food chains in an ecosystem, and the arrows flow from the organism that gets eaten to the organism that eats it. And if these arrows look like they're flowing in the wrong direction, they're not. The small birds eat the fish, and that passes energy and matter from the fish to the small birds. And the same thing happens with mollusks and small birds, and so on and so forth. What are trophic levels? Describe the trophic levels found in most ecosystems. A trophic level is an organism's position in a food chain or a food web. You've seen this word root troph before in terms like heterotroph and autotroph, which we worked with in Unit 3. Producers, autotrophs, create energetic compounds almost always through photosynthesis. The one notable exception are those ecosystems that are based on geothermal energy way under the sea near hydrothermal vents. Primary consumers or herbivores eat the producers. Secondary consumers or carnivores eat the primary consumers. Tertiary consumers eat secondary consumers, and the level goes up to fourth level and fifth level consumers. When organisms at any level die, their remains are broken down by decomposers, really important but not shown in this diagram of a food chain. Describe the pyramid of energy and explain the 10% rule. The pyramid of energy shows the amount of harvestable chemical energy at each trophic level. So this is saying that at the producers, you could harvest 10,000 kilocalories worth of chemical energy. At the primary consumer level, there's only 1,000 kilocalories, and that relationship stands true for the other trophic levels as well. Each trophic level has 10% of the chemical potential energy of the level beneath it. We'll see why in the next slide. What are the reasons for the 10% rule? The first reason has to do with physics. Energy is lost to heat during any energy transformation. That's the second law of thermodynamics. So when primary consumers are eating producers. That's an energy transformation. Some of that energy is simply lost as heat. The other reasons that we're going to discuss are more biological. So any organism is using the fuel that it takes in as part of its metabolism. That doesn't go to growth, and therefore it's not available for the next trophic level. There are limits to harvest efficiency. In other words, these caterpillars over here, they're not going to eat every single bit of the leaves that they're eating, and they're certainly not going to eat the stems or the roots, so they're not harvesting everything. And then, in terms of what they do harvest, they don't assimilate everything. A lot of the food that any organism eats goes through it and passes out as feces. Just note that the 10% rule is an average. It's a rule of thumb that explains how energy is transferred from one trophic level to the next within ecosystems. The pyramid of energy is only one way to represent what's happening in ecosystems. There are other ecological pyramids as well. One is the pyramid of biomass, and what that shows is how much living mass is passed from one trophic level to the next. And that's usually mass with water removed. It's the dry weight of all of the producers compared to the dry weight of all of the consumers. Some pyramids of biomass are indeed like pyramids, but there are others, like this one that's been famously studied in the English Channel, that are not. And if that seems counterintuitive, the basic idea is that the producers grow at an enormous rate and they're consumed by the consumers. So if you measure at any one moment, the standing biomass of the producers can be less than that of the consumers. There's also a pyramid of numbers, and some of those are pyramidal, but if you think about a pyramid of numbers for a tree, you'll have one tree that's being the source of food for thousands or millions of herbivores that are in turn being consumed by carnivores. So the pyramid of numbers, not always pyramidal either. A biogeochemical cycle is yet another way to represent what's going on in an ecosystem. A biogeochemical cycle shows the movement of elements or compounds between the biotic, living, and the abiotic, non-living parts of an ecosystem. And the components include reservoirs, that's locations where elements or compounds accumulate, often in chemically different forms. So for example, one reservoir for the carbon cycle is carbon dioxide in the atmosphere. Another reservoir would be all of the carbon that's found in plants. Fluxes or flows are ways in which these compounds or elements move from one reservoir to another. So photosynthesis is a flux by which carbon dioxide in the atmosphere comes into plants. Consumption is a kind of flux by which the carbon that's trapped in plants moves to animals. If there's one biogeochemical cycle to be familiar with in your AP Bio class, that would be the carbon cycle. And here's how it works. The carbon cycle could start anywhere, but let's start with carbon dioxide in the atmosphere. That carbon dioxide gets pulled into plants through the process of photosynthesis, and think back to the Calvin cycle back in Unit 3. Those plants themselves respire, and that returns carbon dioxide to the atmosphere. Plants can also be consumed by animals. Animals respire, and that returns carbon dioxide to the atmosphere. When plants die, when animals die, their carbon moves to decomposers. The decomposers do cellular respiration on those remains of plants and animals, and respiration returns carbon dioxide to the atmosphere. Sometimes when plants die, and to a much smaller degree when animals die too, they become fossilized into fossil fuels, things like coal and oil. Those get extracted through oil drilling and coal mining, and then those power the machines of our civilization. And so that becomes another major flux called combustion. That, of course, is having impacts on our climate, but that's more of a topic for an environmental science class than it is for AP Biology. Topics 8.3 to 8.4, population growth. What are the 4 factors that directly change population size? If you had driven into my home city, Berkeley, California, a couple of years ago, you would have seen this sign listing the population at 112,580. It's now a little bit bigger. What could cause that to increase or decrease? There's only 4 factors. Those are births, which increase the population, deaths, which decrease it, immigration, which increases population size, and emigration, which decreases it. For AP Biology, one of the most important concepts to understand is exponential growth and its limits. Exponential growth is a pattern of population increase in which the population size doubles at a consistent rate over regular time intervals, resulting in rapid and accelerating increase. Why? Because the growth of the population is proportional to the amount already present. In this growth curve over here, the rate isn't changing. It's continuing to be at 140%. But when there are 1,000 individuals, the number of individuals added over a period of time is going to be much higher than it was when there were 200 individuals. The basic idea is that in exponential growth, the bigger the population, the bigger the increase. Exponential growth is based on a mathematical model, and that's something you need to understand for AP Bio. Then you don't need to memorize it. There's a formula sheet. The formula is delta n over delta t equals r times n. And in this formula, n represents the population size, t equals time, r is the rate of increase. So in plain English, the change in population over time equals the rate of population growth times the population size. In biological systems, when does exponential growth occur? It occurs when a population has ideal resources and nothing is slowing down its rate of growth. This can be the arrival of an invasive species, the early phase of a bacterial infection, the early phase of an epidemic or pandemic. So it's always ultimately limited because nature is finite, but during this phase when the population has ideal growing conditions, exponential growth can occur. That's what occurred early during the coronavirus pandemic. This shows one country, Brazil, 2020, and you can see this exponential growth curve as the virus entered the Brazilian population and spread from individual to individual. Exponential growth. Exponential growth is related to the idea of biotic potential, and that is the maximum rate at which a population can expand, and you can see it represented by the letter R sub max. That represents biotic potential. Exponential growth can't continue forever, and ultimately it's limited by the environment's carrying capacity. That's represented by the letter K. It's this dotted red line over here, and it's the maximum number of individuals that a particular environment can support. It's caused by limiting factors, factors that limit population growth. As a population approaches its carrying capacity, there's increased environmental resistance. The environment starts pushing back against exponential growth as density-dependent limiting factors slow and then stop a population's growth. Density-dependent limiting factors are exactly what they sound like. These are factors that increase in intensity as a population approaches K. As the population grows, its density, the number of individuals in a particular area, is going to go up. That's when these density-dependent limiting factors kick in. They include competition between the individuals in the population for resources, parasitism, parasites that are parasitizing the individuals in the population, and it gets easy for those parasites to spread from one individual to the next as the population increases. It gets easier for predators to pick off members of the population as the population increases, and it becomes more stressful for the individuals in that population, and that tends to lower their rate of increase. To incorporate the effect of limiting factors and carrying capacity in our understanding of population growth, we need a more sophisticated model. That's the logistic growth model, and it's represented by this formula over here. It shows how a population's growth rate decreases as the population's number approaches its carrying capacity, or K, and it's represented by the formula delta n over delta t equals the growth rate times n multiplied by this expression, k minus n over k. N is the population size, t is time, r is the rate of increase, and k is the carrying capacity. Let's plug some numbers into this logistic growth formula and see how it actually works. In this example, K equals 1,000. That's our carrying capacity over here. But let's say that our population is 10. If n is 10, if that's our population size, then K minus n over K is 1,000, that's K, minus n, that's our population, divided by the carrying capacity, 1,000. That's 990 divided by 1,000, that's 0.99. Growth looks exponential because whatever our rate of growth would have been, we're multiplying it times 0.99, will be in our exponential growth phase. But if we increase the population to 900, then look what this does to this expression, K minus n over K. It's now 1,000 minus 900 divided by 1,000, K minus n over K, and that's 0.10. So whatever our rate of growth would have been over here, we're going to multiply it by 0.10, and that's going to cause growth to quickly level off. As we get to K, as population is equal to the carrying capacity, then population growth stops altogether. Those density-dependent limiting factors that are part of the logistic growth model that characterize carrying capacity can be extrinsic or intrinsic. Extrinsic factors come from outside the growing population, and it includes things like predation, parasitism, competition. Intrinsic factors come from within the physiology of the growing population, and it's usually about stress caused by increased crowding and competition between individuals in the growing population, and that lowers the birth rate. Limiting factors can also be density independent, and these are factors that are unrelated to a population size. They're not related to the population's increasing density. So examples are things like hurricanes, floods, earthquakes, human-caused environmental disasters like an oil spill, a toxic chemical release. All of those have nothing to do with the population, but for example, there was a population of birds that was living within these trees. Well, its population is going to be vastly reduced by the damage that's caused by this storm, but that had nothing to do with whether there were a lot of individuals or just a few individuals. It's outside the population's density. It's density independent. So to check your understanding, take a look at this graph, which shows the results of a student experiment where they're growing duckweed in a cup. This graph is from learn-biology.com. It's part of an FRQ or a multiple choice question. This is what duckweed looks like. And the question is, in this graph, I'd like you to identify carrying capacity, I'd like you to identify the exponential growth phase, and I'd like you to identify where you see density-independent regulation. So pause the video, do that, and then see my answer. Carrying capacity is over here at B. You can see that there was an exponential growth phase, and then it levels off. That's got to be carrying capacity. The exponential growth phase is over here at A, when you have the J-curve of growth. And density-independent regulation would be at letter C. How do you know? Because here's the population. It's growing just as it is over here, but then at this point, something abruptly stops population growth. That didn't have to do with the density. The density-dependent regulation kicks in over here, so that had to be independent of density, density-independent regulation. What happens as a population reaches or surpasses its carrying capacity? I'm going to show two scenarios, or probably more. One is oscillation. You have a cyclical overshoot of the carrying capacity and decline, and that's caused by a reduction of resources and then recovery. So here's a population that's grown beyond its environment's carrying capacity. That caused somewhat of a degradation of resources, and that reduces, for example, the food supply, so the population falls a little bit. When it falls, the carrying capacity recovers, and then the population rises, slightly overshoots, and then falls, and that goes on and on. That is different from what's shown on the right, which shows a catastrophic overshoot. The line in blue represents the population of deer. The line in green represents the shrubs that the deer use to forage and to supply themselves with food. Well, in this case, there's a catastrophic overshoot of the carrying capacity, and that causes essentially an environmental collapse, and the population of shrubs crashes and the populations of deer crashes, and there's no subsequent recovery. We have serious environmental degradation. Another population growth pattern to know about for your AP Bio class is about predator-prey population cycles. Here's an incredible data set that shows the population of lynxes, cat, that's a predator in North America, and hares. Both of these animals live in Canada. Both have great data sets because their pelts were collected by trading companies that were active in Canada in the 1800s into the 1900s. So we see this kind of oscillation. What's going on? It's partly explained by density-dependent regulation of the hare population by lynxes. The lynxes are the predators, the hare is the prey. As the population of hares goes up, that provides more food for the lynxes. Their population goes up, they eat more of the hares, the hare population declines, and then the lynx population declines. But when the lynx population declines, there's less predation on the hares, so their population increases. But that's only a partial explanation. What's also going on is that there's cyclical oscillation of the hare population by food availability. So they're herbivores, they're preying on plants that are available in this area, and they're competing with one another. So cyclical oscillation of the hare population by food availability and interspecific competition, which in turn affects the lynx population. Everything is complex. That's what makes biology so fantastic. I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Topic 8.5, Community Ecology Part 1, Symbiosis. What is symbiosis? Symbiosis is an interaction when two species live together in close proximity, and one species might be harmed by that interaction, the other might gain, or both species might benefit, or one of the two species might be unaffected. On Learn-Biology, we have a couple of interactive exercises that you can use to learn about all the different kinds of symbioses that you need to know about for AP Biology. One thing that I want to emphasize now is that there's a symbol system that's used to describe their relationship, a kind of shorthand. And a plus sign is used for the species that gains, a minus sign is used for the species that loses, and a zero is used if there's no effect. So for example, in competition, it's a minus-minus interaction. In mutualism, it's a plus-plus interaction. With that in mind, we'll now go through the various types of symbioses that you need to know. Competition is a kind of symbiosis where two species require for the same resource and they're competing for that resource. It's a minus-minus interaction. Here we have the example of two predators, a leopard and a lion, and they're competing for the same prey. Here we have the interaction between two trees that live in the forests of the North American Northwest, and the Douglas fir tree and the coast redwood compete for resources such as light, water, space, and soil nutrients. Mutualism is an interaction where both species benefit from the interaction. It's a plus-plus, a win-win interaction, and here we have two examples. One is between clownfish and anemones. The clownfish lives amidst the tentacles of the anemone, and evolution has led to a situation where the anemone doesn't sting the clownfish. So the clownfish gets safety, that's its win. What does the anemone get? Well, the clownfish is a messy eater, so food falls out of its mouth into the tentacles of the anemone where they're digested. And when the clownfish defecates, the feces are also used by the anemone for food, a win-win relationship. Not all of these symbioses that are mutualistic involve anemones, but I couldn't resist sharing this other one. This involves the giant green anemone that lives off the coast in the intertidal zone of the northwestern part of the United States. And this anemone, which is in the same clade as jellyfish, stinging tentacles, just as we saw over here, kind of a predator, it harbors symbiotic algae that live within its tissues. Well, those algae get a safe place to live, and they produce food that the anemone can digest. So it's a win for the anemone, and it's a win for the algae that the anemone harbors. Predation is a relationship where animal species 1 kills and eats animal species 2. It's obviously a plus for the predator and a minus relationship for the prey. It's a win-lose interaction. Here we have a leopard killing a bushbuck, and here we have a kingfisher killing a tadpole. That's predation. In herbivory, an animal species eats a plant, and that's a plus for the animal, and it's a minus for the plant. It's a win-lose interaction. Here we have deer grazing on a tree, and here we have a sawfly caterpillar eating a leaf. Commensalism is a relationship where species 1 benefits and species 2 is unaffected. So it's a plus/0 relationship. Win, no effect. In this example over here, we have a cattle egret that likes to perch on top of the cattle. The egret gets a nice place to perch, and the cattle is pretty much unaffected. On the right, we have moss growing on a tree trunk. The moss gets a nice place to live and grow, and the tree is pretty much unaffected. Parasitism is a long-term relationship in which species 1, the parasite, lives in or on species 2, which is the host, and the host is harmed by that interaction. So it's a plus-minus win-lose interaction. Here are two examples. One involves viruses. Viruses are obligate intracellular parasites. In order to reproduce themselves, they need to infect cells, and those can be your cells, the cells of animals, the cells of plants, the cells of bacteria, the cells of protists. So the virus is the parasite, and you, or another organism or another cell, is the host. Here's another parasite. This one is Giardia. That's a single-celled eukaryote that infects people when they take in contaminated water, food, etc. The Giardia reproduces inside our intestinal tracts. It causes diarrhea, other kinds of intestinal upset, and then it leaves via the feces to contaminate other food, water, and spread to another person. Brood parasitism is a relationship in which bird species 1 lays its eggs in the nests of bird species 2. Species 2 feeds and nurtures species 1 at the cost of its own young. That's obviously a win-lose, plus-minus relationship. And here we see this little reed warbler that's feeding this cuckoo that has parasitized its nest. Parasitoidism is yet another variation on parasitism. It's mostly limited to insects, and it's where species 1 lays its eggs in or on the eggs or larvae of species 2. Individuals in species 2 are eventually killed, and that can take quite some time. This is a plus-minus relationship, a win-lose relationship. And here we have parasitoid wasp cocoons and a lime butterfly caterpillar. And the part of the story that you're not seeing is that a wasp came over to the caterpillar, injected in its eggs. The eggs developed inside the caterpillar and then emerged through these holes over here, and then the larvae formed cocoons around themselves. When the cocoons have fully developed, little wasps will emerge, and they'll go ahead and they'll parasitize other caterpillars. Just in case you're thinking that this parasitism niche is unique and unusual, it's not. There are more parasitic species than any other kind of animal niche. I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. Topic 8.5, Community Ecology Part 2, Competition and Co-Evolution. Before we get into competition and coevolution, I want to define a term that I used in the last section and I've used throughout this series. The term is ecological niche. You can also say niche, and it's how an animal makes a living. Here we have a bunch of dinosaurs and other Mesozoic reptiles. This one is an apex predator and a scavenger. This flying reptile over here was an aerial predator and a scavenger. This, the Triceratops, was a large herbivore. Here's an omnivorous forager. This one's an omnivore. This one's an herbivore or an omnivore. This one's a small predator, and here's an herbivore, also an armored grazer. Those are various dinosaur niches. Plants have niches as well. So an oak is a sun-tolerant tree. A rhododendron is a shade-tolerant shrub. A mangrove is a salt-tolerant tree or shrub. A cactus is a desert-adapted plant. A water lily is an air-requiring aquatic plant that lives on the surface of lakes or ponds. An ivy is a climbing vine. There are other plant niches as well, but this should give you the idea. An important idea related to competition is Gauss's competitive exclusion principle. This principle states that two competing species can't coexist in the same ecological niche. Gauss did experiments with various species of paramecia. So here are two species of paramecia, one of which lives on the bottom of lakes and ponds, that's their natural habitat, and the other is a surface forager, so it swims up higher. And you can see that by the position they would take in a test tube. When these two species were combined in the same test tube, they were able to coexist just fine. Why? Because their ecological niches, their ways of making a living, were different. But when Gauss took two species that had the same ecological niche, both of them were surface foragers, and when he combined them in the same test tube, what he found was that species A outcompeted species B, which became extinct. What did we see? We see two species in the same ecological niche can't coexist. That's Gauss's competitive exclusion principle. Over long periods of time, competition can be a powerful evolutionary force. One possibility is that species become extinct. The other possibility is that the competing species evolve through natural selection to do something that's called a resource partitioning. That's exploiting different parts of the resource that they were competing for. The way that that happens is through specialization. There's selection for different kinds of traits, and that results in character displacement. That simply means differences so that each species will dominate its sub-niche more effectively. Here we have the example of a variety of shorebirds, and you can see the character displacement, the different length of the legs, the different shape of the beaks. And what that does is that it enables each bird to exploit a different part of the resource, so that the avocets aren't really competing with the ducks because they're foraging in this part of the shoreline habitat, whereas the ducks are foraging over here, and the oyster catchers are actually on the sand and the plovers are higher up on the sand. So again, character displacements are the differences, and what they wind up with is species that can effectively exploit the resource that they were formerly competing for. To make this clear, I want to offer an economic analogy, character displacement and resource partitioning in the coffee niche. So here we have three companies that live in my part of the world, and they've all evolved through competition, slight differences. Starbucks, it's the mainstream everyday coffee experience with sugary specialty drinks and snacks, seasonal offerings. That's different from Peet's, which appeals to coffee purists, really high quality beans, dark roast, more robust coffee taste. And that's different again from Phil's Coffee, which specializes in pour overs, each cup brewed fresh to order. Customers are encouraged to customize their drinks. So again, we have three competing companies. Each is partitioning the resource for different parts of the economic market for different customers, and they've done that by evolving different characteristics. If you can apply that to animals and plants, you'll understand these concepts well. Resource partitioning can lead to what are called ecomorphs, and those are species that are morphologically adapted through their form to specific niches. So in the Caribbean islands, you have lizards that have adapted to different parts of the forest canopy. You have these very large lizards that are called crown giants, smaller ones called trunk crown, ones that live on the trunk, ones that live on the ground, and ones that live in the grass. Well, that's resource partitioning. That is all about character displacement. But what's really cool is that through convergent evolution, ecomorphs from different regions, different islands in this case, can develop similar traits. So you have crown giant lizards that can be found on Cuba, Jamaica, the island of Hispaniola, and Puerto Rico, but they're not closely related to one another. They evolve these traits independently through convergent evolution. And that's true of the trunk crown lizards, the ones that live in the trunk ground, and the grass bush. And this relates to a really cool case study from the Howard Hughes Medical Institute that most AP biology students experience, and now you'll understand it better. Here's a really interesting data set that could show up on the AP Bio exam or in your course related to beak depth in the Galapagos finches. Here are 3 of the Galapagos finch species that live on the Galapagos Islands, which are off the coast of Ecuador. The character that's been quantified here is the beak depth, the distance between here and here in the beak. Different size beaks are adaptations for exploiting different types of food. Here is what's going on. On islands where these birds live together, they're all inhabiting the same island, note that there's no overlap in terms of the beak depth. That's true of island set A and island set B. On island set C, that involves Floriana and San Cristobal, one of the bird species, G. magnorostris, is absent, but note that there's no overlap between bird species G. fulginosa and the other bird species G. fortis. But on these really small islands, Cuatro Hermanos over here and Daphne Mayor over here, only one bird species is present. And with only one bird species, note the overlap in beak depth. What I'd like you to do is try and explain that. So pause the video and see if you can come up with an explanation. On island set A and B over here, there are 3 species that are coexisting, and competition between those 3 is leading to niche partitioning, and adaptation to different types of food is leading to character displacement. So there's no overlap in the beak depth. We have the much larger beaks over here, the medium-sized, and the smaller size over here. On island set C, one of the species, G. magnorostris, is missing, but because there's still competition between these 2 species, G. fulginosa and G. fortis, there is character displacement and there's no beak depth overlap. On the smallest islands, D and E, there's only one species. And without competition, with no competition between different species, character displacement decreases. The beak depths overlap. So here we have a kind of natural historical validation of these ideas related to character displacement and resource partitioning. Another concept related to competition is the difference between a fundamental niche and a realized niche. A fundamental niche is the range of resources that a species could exploit in the absence of competition. A realized niche is the range of resources that a species actually does exploit in the competitive context in which it lives. An interesting natural history study was done in the intertidal zone involving 2 species of barnacles. That's a kind of crustacean that lives on the rocks of the intertidal zone. Here's species A, which lives closer to the high tide line. Here's species B, which lives closer to the low tide line. If species B is removed, which you can do by scraping these barnacles off the rock, notice how species A expands into the low tide zone where species B was living. But if species A is removed, species B pretty much stays where it is. So your task is for species A and B to compare their fundamental and realized niches. Pause the video, do that, and then go on. For species A, the fundamental niche is much wider than its realized niche. How do we know that? Because when species B is removed, species A is capable of colonizing this zone over here that's much closer to the low tide zone. Species B, by comparison, has a fundamental niche that's pretty much the same as its realized niche. And how do we know that? Because when species A is removed, species B is incapable of expanding into this zone over here, closer to the high tide line. Species B can outcompete species A, but it can't live in the area where species A is predominant. That's the difference between a fundamental niche and a realized niche. Let's end this discussion of symbiosis by talking about evolutionary arms races. These are positive feedback loops where adaptations in one species lead to counter adaptations in the second species, and they're associated with predation, parasitism, and herbivory. They result in extreme adaptations related to speed, sensing, camouflage, defensive and offensive weaponry. That can be physical weaponry or chemical weaponry. Why is a cheetah capable of running 60 miles an hour? Well, it's because the cheetah's speed has selected its prey for speed. And as the prey have evolved to become faster, that's caused a counter adaptation in the cheetahs, which has caused them to become faster. This obviously doesn't go on forever because there are physical and physiological limits, but when you see an amazing adaptation, it probably stems from an evolutionary arms race. Why is this insect so incredibly well camouflaged? Because there's some predator on the end which has incredible visual acuity. Why is this crocodile or alligator so incredibly well camouflaged in the water where it's swimming? Because on the other end, there's a prey organism which has acute hearing and acute vision. And so the alligator has evolved counter adaptations. And so it goes in the amazing pageant of life that we call biology. Topic 8.5, Community Ecology, Part 3. Keystone species and trophic cascades. What are keystone species? These are species whose action in a biological community structures the entire community. They're frequently, but not always, predators who keep a particular herbivore in check, and their effect is to increase the overall biodiversity of a biological community. Using the example of sea stars in the intertidal zone, explain how keystone species promote biodiversity. Sea stars are predators, and they prey on a variety of other animals in the intertidal zone. Here's the intertidal zone, the zone between the low tide and the high tide, and here are some of the many animals that sea stars prey on, including, importantly, mussels. When sea stars are removed from the intertidal zone, biodiversity can plummet. That's because by preying on the mussels, sea stars create ecological space, physical space, for other invertebrates to live in this community, and that keeps biodiversity high. So this is what this community looks like when sea stars are present. When sea stars are experimentally removed, which happened in a famous experiment by Robert Payne in the 1960s, where he took sea stars and threw them into the ocean away from the intertidal zone where they were preying on mussels, the mussels overgrew the entire intertidal zone, and that caused species diversity to fall. All the other species that were previously living in that zone couldn't live there anymore. So that's the effect of the removal of a keystone species upon the diversity of a biological community. Another example of a trophic cascade relates to the reintroduction of wolves into Yellowstone National Park. That reintroduction happened in the 1990s. Previous to that, wolves had become locally extinct in the Yellowstone ecosystem, mostly due to overhunting. When wolves were reintroduced, they started to prey upon elk. The elk, in the wolves' absence, had reduced the numbers of aspen and willows, particularly along riverbanks. So the aspen and willows were able to regrow. That provided habitat for beavers to use the willows and aspens to create beaver dams. That created great aquatic habitat, and that led to an increase in organisms like amphibians, fish, and songbirds. The wolves were also competitors with the coyotes, so their reintroduction led to a decrease in coyote numbers, and that led to flourishing of some coyote prey, including foxes, rodents, and antelopes. The main thing for you as an AP biology student is to be able to interpret a diagram like this and to know the basic idea, which is that the reintroduction of a keystone species increases biodiversity through a trophic cascade effect. To clear up a possible source of confusion, note that the top predator is not always the keystone species. This emerged from a famous study by James Estes in the waters off Alaska in the 1990s. And what Estes discovered is that when orcas changed their prey preference from seals to otters, then otters were no longer able to control the population of urchins. Urchins overgrazed the kelp, and that led to an overall decline in the kelp forest ecosystem. You can see this in this data set, and this kind of data set is something that could definitely appear on the AP Bio exam or on your teacher's next test. And what this shows is that a reduction in otters by orcas, these numbers show the various islands where Estes studied this decline, led to an increase in the biomass of sea urchins. Here it is in the late 1980s. Here it is just before 1997. That led to more intense grazing by sea urchins upon kelp. This is a measure of their grazing intensity, and that led to kelp forest decline, which you can see in the overall decrease in kelp forest density. Again, what's important is your ability to interpret these data sets and connect it to the idea of a trophic cascade. Finally, it's important to note that not all keystone species are top predators. For example, beavers are what are called ecosystem engineers, and their dams create habitats for dozens of other species, increasing biodiversity. Are you asking yourself, how am I going to get a 4 or a 5 on the AP Bio exam? It's a good question because it's a hard test, but we have a plan for your success. Go to learn-biology.com, sign up for a free trial, and complete our interactive tutorials and interactive AP Bio exam reviews. We guarantee you a 4 or a 5 on the AP Bio exam. See you on learn-biology.com. Topic 8.5, Community Ecology, Part 4, Ecological Succession. What is ecological succession? It's how a community undergoes predictable changes following a disturbance, such as a fire, flood, landslide, or volcanic eruption. Each array of species in each stage, so these are the different stages of succession, creates conditions that subsequently allow different plant and animal communities to thrive. So as the community ages, the plant and animal community here is going to be different over time, and it culminates in a climax community that will endure in relative equilibrium until a disturbance restarts the process. Succession is classified as being either primary or secondary. Primary succession starts from bare rock, and it follows an event such as a rock slide, a volcanic eruption, glacier retreat. And the most important thing is that primary succession requires creation of soil, which is the basis of plant communities. Here's a picture of my wife and myself in Maui, and we're standing in a volcanic crater, and you can see that there was a lava flow and plants are starting to come in. That's primary succession. On the right, we have secondary succession. The soil is still intact, and it follows a fire, a flood, a clearing of forest, etc. Because the soil's already present, the process can occur much more quickly. How does ecological succession unfold? It begins with pioneer species. Those are shown over here at stage 2. And those are organisms like lichens and algae that can live on bare rock, which was over here in stage 1. This is obviously a primary succession scenario. And that begins the accumulation of biomass and the creation of soil. After we have those pioneer species, we have sun-tolerant mosses and herbs. They're shown over here at stage 3, and they create soil. You can see the soil starting to develop over here. That sets the stage for sun-tolerant grasses and ferns that are over here at stage 4. That's followed by the introduction of small shrubs and then trees over here at stage 5. And those trees create a shady understory. That's simply the area below the canopy of the trees. And that creates a niche for sun-tolerant shrubs. In other words, plants that really can't survive in full sun, but are well adapted to live in partial sun. Finally, what we have is a self-perpetuating climax community, and again that can endure until a disturbance resets the clock. Some of those communities, like the redwood forests of the Pacific Northwest of California, those can endure for tens of thousands of years. What are the key trends associated with ecological succession? Note first of all that here we have succession not on a terrestrial ecosystem, but in an aquatic ecosystem. We have what's called pond succession, because successional processes occur there as well, and the process runs sequentially A through F. Overall, as the process unfolds, abiotic conditions, non-living conditions, are replaced by biotic conditions. Soil mass increases. You can see the creation of soil as this pond begins to fill in over here. Overall biodiversity increases. In other words, there are more species over time. The number of interspecific interactions increases as there are more species. And the community over time becomes more stable and more resilient. Topic 8.6, biodiversity. What is biodiversity? What are its components? Biodiversity is the variety and variability of life within an area, and it has several components. That includes ecosystem diversity. So if you think about an ecosystem like the one in Yellowstone National Park, there are mountains, there are meadows, there are aquatic ecosystems, so a variety of ecosystems in one area. You can also talk about species diversity, the number of different species within an ecosystem, or you can talk about genetic diversity, so if you're looking at that species, how much genetic variability is there among the individuals that comprise that species. An important idea related to biodiversity in AP Bio is that biodiversity increases resilience. What's resilience? It's the ability of a system to respond to change and to bounce back from difficulties. Here's an example. Here's a field of corn with one species, and in fact in today's world, this might be a genetically engineered field of corn where there's really only one individual that's cloned over and over again. So we have low genetic diversity and low species diversity combined into one. If there were some kind of fungal pathogen that were to enter the system, or an insect herbivore that were to figure out how to eat this corn in the face of pesticides and herbicides and all the other things that we do to protect fields of corn, well, this field would be extremely susceptible to damage. in a grassland like this with many different species, if there were a fungal pathogen that impacted one species, there are many other species that are in this same system. Am I saying that we shouldn't be converting grasslands into fields of corn? No, of course we need corn for food and for other products, but as we develop our planet, we need to do that with an eye towards maintaining biodiversity, ecosystem, species, and genetic at as high a level as possible because that promotes resilience. We just made a connection between biodiversity and ecosystem resilience. That's a benefit of biodiversity, but there are several more. One is that biodiversity is intrinsically beneficial. That means it's a good thing in and of itself. And to prove that, just go out into nature and you can experience the joy of the diversity of different ecosystems, the diversity of all the different species within those ecosystems, and the variability of the organisms within the ecosystem. So that is the intrinsic benefit of biodiversity. In addition, biodiversity provides many present and potential benefits to humanity. One example is the Pacific yew, which is a shrub that lives in the forest, and what was discovered years ago is that the yew contains a compound that has anti-cancer properties, and that's been developed into an important anti-cancer drug that's treating millions of people. Who knows what other compounds like that exist within the incredible biodiversity that exists in the rainforests or in coral reefs, so on and so forth. So we need to maintain biodiversity to gain those benefits. If all those species become extinct, those direct benefits won't be things that we'll be able to get. And finally, there are ecosystem services. Those are things that biodiversity provides to us for free that might otherwise have huge economic costs. The oxygen in the atmosphere, where's that from? It's from photosynthesis, provided for free by the producers of planet Earth. The oceans, when they're intact and healthy, can absorb carbon dioxide. That can mitigate climate change. That is an important ecosystem service. The bats that fly around eating insects, that keeps insects under control, another ecosystem service, and there are thousands just like that. The economic benefit of ecosystems that are intact and healthy is incalculable, and that's the benefit of biodiversity. Within the AP Bio curriculum, diversity comes up in two places. In Unit 7, we looked a lot at the importance of variability within species. And if you want to review that, you can go back to my Unit 7 video. In Unit 8, what we focus on is mostly species diversity. Here are three communities, Community A, Community B, and Community C. And what we're going to do is we're going to compare their diversity. And we're going to do that based on two components. The first is species richness, the number of species in an area. And the second is species evenness, how evenly distributed the members of a species are. So what we're going to do is we're going to rank these three plant communities from most diverse to least diverse. And to do that, we're going to do some counting and analysis. If you look at Community A, it has 3 species, 1, 2, and 3. And in terms of the number of individuals in each one, they're very evenly distributed. There are 4 individuals of species 1, 4 of species 2, and 4 of species 3. Community B has 4 species, 1, 2, 3, and 4, and they're evenly distributed. There are 3 individuals of each species. In Community C, there are also 4 species, but they're not evenly distributed. There are 9 individuals of species 1, and there's 1 individual each of species 2, 3, and 4. As we'll see in a moment, we're ultimately going to use some math to determine which of these communities is the most diverse, but we're going to start a little bit more intuitively. Community B is the most diverse. Why? Because it's tied with Community C for the highest species richness. Both communities have 4 species, and it's tied with A for highest species evenness. In both A and B, the species are completely evenly distributed. But how would we compare community A, 3 species evenly distributed, with community C that has 4 species, but they're not evenly distributed? To properly compare the diversity of different communities, we're going to need to use a formula. That formula is the Simpson Diversity Index. Let me tell you what it is, what all the symbols represent, and then we'll use it to figure out the diversity of community A. Little n represents the total number of individuals of each species. Big N represents the total number of individuals of the entire community. And this symbol means the sum of. So here's the diversity index. Let's compute it. So for species 1, there are 4 individuals. 1, 2, 3, 4. So we're going to plug 4 in over here. Then we have to figure out N, the total number of individuals in the entire community, and that is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. That's the total number of individuals in the entire community. So small n divided by big N for species 1 is 0.33. 4 divided by 12 is 0.33. Then we take that value and we square it. 0.33 squared is 0.111. For this community, it happens to be the same for all 3 species. So species 2 also has 4 individuals out of a total 12 in the entire community. So the values are the same. Again, n over n squared is 0.111, and that's true for species 3 as well. You add those all together, and that comes to 0.333. You subtract that from 1 minus 0.333, and the value is 0.67. That's your diversity index for community A. Let's repeat the same steps for communities B and C. For community B, there are 4 species, and let's count how many members there are of species 1. There's 1, 2, 3. So small n equals 3. The total number of individuals, again, is 12. 3 divided by 12 is 0.25. The values are the same for species 2, 3, and 4. There are 3 individuals for each of those species out of a total number of 12 individuals in the community. So 0.25 is n, small n divided by large n, and then we're going to square it. 0.25 squared is 0.063. The value is the same for each species in this community. We add that up, 0.063 plus 0.063, etc. We add that up, and what we get as our sum is 0.250. 1 minus 0.250 is 0.75. So we see that the diversity of community B is indeed higher than that of community A, and that's because the species richness is higher, even though the evenness of both is the same. They're completely even. Our question was about comparing the diversity of community C with community A. So let's run the numbers for community C. Like the other 2 communities, it has a total of 12 individuals, of which 9 belong to species 1. So we're going to do little n divided by big n divided by 12. We get 0.75. Then we're going to square that, and we get 0.563. So that's what this value is for species 1. Now let's do the same for species 2, 3, and 4, because it's the same number for each of them. There's 1 individual. 1 divided by 12 is 0.08. That's little n divided by big N. We square that, and we get 0.007, and that's the same for species 3 and 4. So we're going to add up all those values because of this expression over here, and when we do that, we get 0.583. We know that we have to subtract the sum from 1, so 1 minus 0.583 is 0.42. So we see that community C even though it has 4 species, has a lower diversity than community A, and that's because it had very low species evenness. So we can see how by analyzing each of these communities using the Simpson Diversity Index that community B has the highest diversity, 0.75. And again, think about why that is. It has high species richness and high species evenness. The next community in terms of diversity is community A, which also had high evenness, but its richness was a little bit below that of community B. And community C, even though it had a higher species richness than community A, its evenness was so low that its diversity index wound up being lower than community A. That's how you use the Simpson Diversity Index to determine the diversity of various communities. Topic 8.7, ecosystem disruption. Humans are causing biodiversity losses around the planet. In Unit 7, we talked about the 5 great mass extinctions. There's a 6th great mass extinction that's happening right now, and humans are unfortunately the cause of it. What we're doing is we're sending many species to the brink, down the extinction vortex, by changes that are creating small, isolated populations. Those populations are subject to genetic drift and inbreeding. They lose genetic diversity. They become less fit, less adaptable. That lowers their reproduction rate. That increases their mortality rate. That leads to smaller populations, and so the cycle goes. This is a positive feedback loop or a vicious cycle that's occurring around the planet right now. We're going to talk about 5 things that humans are doing that are causing these biodiversity losses that are generating this 6th extinction. The first thing is habitat alteration and destruction. There is nothing wrong with cities, fields, or golf courses. But every time we build a city, a field, or a golf course, we destroy the habitat of the animals and plants that live there. That's happening at a huge scale around planet Earth, where a huge amount of the planet's surface has now been in some way modified for human ends. That's one of the causes of species extinction. Another thing that humans have done is we've overexploited resources, and that can be through overharvesting or overhunting. In the case of the Tasmanian wolf and the passenger pigeon, these animals were hunted to extinction. In the case of the American bison, to the brink of extinction. Another major cause of biodiversity disruption has been habitat fragmentation, and that's taking a large contiguous area and cutting it up by roads or by development into smaller areas. What does that do? It creates isolated, disconnected populations that experience population bottlenecks and lose their adaptability. And it also creates situations where there's too much edge habitat. Edge habitat is exactly what it sounds like. It's the habitat on the edge of an area. And when you fragment an area, you create a lot of edge relative to the amount of habitat in the interior. And those edge habitats are disturbed areas that have different conditions from the interior conditions, and that makes it harder for the species that are trapped in the fragments to survive. Another cause of ecosystem disruption is the introduction of invasive species. Invasive species have the following characteristics. They're highly adaptable. They tend to be generalists in terms of their niches. They can figure out many ways to survive. They're good at dispersing themselves. They have high reproduction rates. Their effects include preying or parasitizing local species in the new area where they arrive, and outcompeting local species, and disrupting food chains and food webs. Again, invasive species, another main cause of habitat disruption. A final cause of ecosystem disruption is deforestation. Obviously, when you cut a forest, you disrupt those ecosystems, and forests, particularly tropical rainforests, are some of the most diverse habitats, ecosystems on the planet. Here are your next moves for AP Bio Success. Please subscribe to learn-biology.com and please watch this next video.